young colleagues. Thank you so much for joining this event. So I, on behalf of the organizing committee, welcome you all to the 33rd National Congress of Parasitology that's being organized by CSIR-CDRI and Indian Society of Parasitology. Today's event is a very unique gathering because we are celebrating 75 years of CSIR-CDRI and of course the golden jubilee of the inception of Indian Society for Parasitology. And to start this event, we are especially honored to have with us our chief guest, Professor N.K. Ganguly, who is like a family to CDRI. He's an eminent scientist, a visionary, an institution builder, policy advisor, and one of India's most reputed and respectable biomedical researchers. Thank you, sir, for being here. Your presence elevates this event, and more so because it tells us how a good and able leadership can lead India in the 21st century. We are also honored and delighted to have Dr. A.M. Khan, who is the president of the Indian Society for Parasitology and has spent decades in parasitology research in different areas. So we express our gratitude to both our guests and for gracing this occasion. And I now invite Professor Nirmal Kumar Ganguly, Dr. Khan, our director, Dr. Radha Rangarajan, and Dr. Satish Mishra, organizing secretary for the 33rd NCP, to please come and occupy their seats on the dais. Welcome, sir. So to officially begin with the function, I invite our director, Dr. Radha Rangarajan, to present a bouquet of flowers to our chief guest, Professor Ganguly. I request Dr. Radha Rangarajan to welcome our ISP President, Dr. A.M. Khan, with a flower bouquet. I now request Dr. Satish Mishra to welcome our director. I request the dignitaries to maybe give a moment and maybe start with the traditional lighting of the lamp. To officially begin the inaugural session, may I request our director, Dr. Radharang Rajan, to please welcome the delegates for the 33rd NCP.
Thank you, Mridant. Good morning to all of you, esteemed guests, delegates, colleagues, researchers, students, all those assembled here. I know it's a wintry morning, perhaps cold for many of you, but we hope this event warms you up greatly. I welcome you all officially to the 33rd National Congress of Parasitology, which we are co-hosting with the Indian Society of Parasitology. We are deeply honored by the presence of several eminent scientists and public health practitioners in our gathering today, whose contributions have shaped the landscape of biomedical and parasitic disease research in India and globally. I'd like to welcome Dr. A.M. Khan, President of the Indian Society of Parasitology and Director in Charge, ICMR, National Jalma Institute for Leprosy and Other Mycobacterial Diseases, Agra. He is an accomplished public health researcher, and his leadership in leprosy and mycobacterial disease programs continue to make remarkable impact. We are really privileged to have Dr. Nirmal Kumar Ganguly join us today, a Padma Bhushan awardee and former Director General of the ICMR. He is one of India's most celebrated biomedical scientists. His contributions to infectious diseases, public health, and medical education have been recognized nationally and internationally. Dr. Ganguly, of course, has a long-standing association with CDRI. He has served as chairman of the Research Council for over 10 years and has been a member of the Research Council of CDRI for another 10. So we are really, really happy to have him here with us. Welcome, sir. Dr. Bhupendra Tripathi is a senior member of the leadership team of the Gates Foundation. He leads the infectious disease and vaccine delivery initiatives in India and guides strategies to eliminate major parasitic and infectious diseases while strengthening immunization equity. Welcome, Dr. Tripathi. Parasitic diseases remain a formidable global challenge, disproportionately affecting vulnerable populations. In an era that is marked by drug resistance, emerging infections in new geographies due to climate chaos, increased human migration, and other global factors, research in parasitology continues to be highly relevant in the biomedical and public health arena. CDRI has had an unbroken tradition of working on parasitic diseases since almost its very inception. Many will remember the pioneering role we played in establishing various animal models for parasitic diseases and for our role in international and national efforts towards drug development and vaccine evaluation. In fact, one of our products still in the market today is a parenteral formulation of Artetha, which is now sold in over 20 countries. And in terms of the current pipeline, we have an advanced candidate in late stages of preclinical development. Given these research interests, we are particularly delighted to host the 33rd National Congress of Parasitology, along with the Indian Society of Parasitology. ISP is a distinguished scientific body affiliated with the World Federation of Parasitologists. The society fosters scientific exchange, encourages research, and promotes innovations in parasitology and tropical medicine. Its flagship publication, the Journal of Parasitic Diseases, published with Springer, is a testament to its commitment to disseminating high-quality research. In addition, ISP members and researchers actively support and conduct community-level initiatives, such as epidemiological studies, awareness campaigns, and health education programs. Incidentally, the ISP has its origins in CDRI 50 years ago. Actually, it's 1973, so mathematically, this would not be the 50th year, but if you exclude the COVID years, which we did, we're counting this as the 50th year. The founding members and the scientists were scientists in this institution. Dr. Ganguly served as one of the early presidents. And CDRI is marking its 75th year. So we are marking 2 milestone anniversaries through this conference. The 33rd NCP is focused on recent advances in parasite biology and drug discovery. The major themes are host-parasite interactions, parasite biology, transmission dynamics, mechanisms of resistance, new targets. There are also sessions on diagnostics, drug discovery, and target validation. While malaria is a key disease area, leishmanial, filarial, toxoplasma, and other veterinary parasitic infections are also well represented. While we deliberate on the biology of the parasites and the pathogenesis of disease, the ultimate goal, of course, for all of us is to get to better therapies. On this front, there has been some good news on drugs for malaria. At the recently concluded American Society of Tropical Medicine and Hygiene meeting in Toronto, the pharmaceutical company Novartis shared data on a new anti-malarial drug combination developed together with the Medicines for Malaria venture, MMV. The combination consists of a novel compound, ganaplacide and lumefantrine. The pivotal study enrolled more than 1,600 patients with malaria across 12 African countries who were treated for 3 days with either the new combination or coartem. Coartem is artemether plus lumefantrine. Researchers reported that 99.2% of those treated with the new combination were cured, compared with 96.7% of patients who received the artemisinin combination. This is, of course, great news and a major milestone for a disease that has not had a novel drug since 1999. Artemisinin-based combinations have served humanity well, but drug resistance has been worrisome, and now there is a sign of relief that a new drug combination is available. Should this be used immediately or should it be reserved for those who fail existing ARTs is a debate that will be resolved in due course. But the backstory to this is that this compound came from screening over 2 million compounds, and it took more than a decade. And even now, the mechanism is not fully understood. It is this backstory that reminds us that we still have a job to do. The question is how we can advance our understanding of biology and new targets, mechanisms, tools and technologies to improve the speed of discovering new drugs, because we know that resistance is inevitable. What are the underserved, vulnerable populations that still need our attention? I urge you all to be talking about these issues as you engage in discussions and deliberations over the next few days. I encourage all the participants to engage really actively, exchange ideas, and find collaborations so we can collectively push the boundaries of parasitology and drug discovery. I take this opportunity to congratulate the organizing secretary, Dr. Satish Mishra, who was ably supported by Dr. Mrigank Srivastava. Thank you very much for all of your efforts. This conference would not be possible without you being in the lead. I also want to thank the various subcommittees and their and the conveners of these subcommittees, Dr. Neeti Kumar, Dr. Anand Kulkarni, Dr. Amir Nazir, Dr. Namrata Rastogi, Dr. Amog Sahasrabuddhe, Dr. Rajkamal Tripathi, Dr. Bidyut Purkayat, and of course their respective committee members and volunteers for their dedicated efforts in shaping the event. I want to specifically also thank Dr. Saman Habib for her continuous involvement in shaping the agenda of the conference. I'd like to acknowledge the support of ANRF, DBT, and CSIR for enabling us to host this event. I also want to thank the Gates Foundation for their generous support and our major corporate partners, Themis Medicare and Premas, for their financial contributions. Once again, I welcome you all to CSIR-CDRI Lucknow. The next few days are no doubt going to be enriching and exciting. I hope you enjoy the time away from your laboratories and soak in the science and take back new ideas. I wish you all a fantastic learning experience. Thank you.
Thank you so much, ma'am, for your welcome address. I now invite our President of the Indian Society for Parasitology, Dr. A.M. Khan, to deliver his welcome address.
Good morning. Honorable Chief Guest, Professor Nirmal Kumar Ganguly, former Director General, Indian Council of Medical Research, New Delhi, Dr. Radha Rangarajan, Director, CDRI, Lucknow, distinguished guests, esteemed colleagues, respected mentors, young researchers, and friends. It is my honor and privilege to welcome you all to the 33rd National Congress of Parasitology, being held as part of the Golden Jubilee Year of Indian Society of Parasitology. This year's conference is especially meaningful as it coincides with the Platinum Jubilee celebration of CDRI Lucknow, our gracious host institution. As we gather here for the next 3 days, we celebrate not just 5 decades of scientific pursuit, but 5 decades of commitment, collaboration, and collective progress. Since its inception in 1973, the ISP has remained the leading national platform dedicated to advancing research in parasitology, education, and public health. Over the years, parasitology, both human and veterinary, have transformed dramatically. What we began as a discipline rooted in classical morphology, taxonomy, and clinical pathology has now expanded into a vibrant multidisciplinary unit or approach. Today, pathologists work at the cutting edge of molecular biology, immunology, genomics, metabolomics, bioinformatics, and artificial intelligence, which are enabling us to monitor and understand parasitic diseases with unparalleled accuracy. However, the foundation and support of basic parasitology still makes its relevance in the progress of overall parasitology. We take immense pride in the contributions of Indian parasitologists to national priorities. Our collective efforts have significantly contributed to the control and elimination process of lymphatic filariasis, malaria, and Kala-Azar as well, and also to continue support the management of various zoonosis and neglected tropical diseases. The ISP is also contributing to one health framework, connecting the health of humans, animals, and environment. Our work directly impacts public health, livestock productivity, food safety, and ecosystem balance. Our mission extends beyond laboratories and conferences. ISP members are actively engaged in community initiatives, field investigations, epidemiological surveys, health education, and awareness activities also. These efforts ensure that scientific knowledge is translated into real-world solutions that benefit the most vulnerable populations. As we look ahead to the next 50 years, our vision is one of innovation, inclusivity, and impact. The challenges before us are emerging and re-emerging infections, climate-related disease patterns, changing in vector ecology, and drug resistance. It demands fresh perspectives and interdisciplinary collaborations. We must empower young researchers, strengthen national and global partnerships, and embrace new technologies, particularly AI-driven analytics and rapid molecular diagnostics and predictive modeling. Over the next 3 days, you will hear from a diverse and distinguished group of scientists whose research spans the entire spectrum of parasitology. I am confident that the discussions, debates, and collaborations done here will spark new ideas and open new avenues for addressing parasitic diseases of national and international importance. On this special occasion, I express my heartfelt gratitude to all the past presidents, member secretaries, office bearers, and the entire ISP community. Your vision and dedication have built the strong foundation on which we stand today. Let us continue to work together with curiosity, compassion, and commitment to advance the frontiers of parasitology and contribute to a healthier parasite-free world. Thank you. I wish you all a productive and inspiring conference. Jai Hind.
Thank you so much, sir, for your inspiring words. I now request Dr. Satish Mishra, Organizing Secretary of the 33rd National Congress of Parasitology, to deliver his welcome address.
Good morning, esteemed Chief Guest Dr. N.K. Ganguly, respected Director Dr. Radha Rangarajan, and ISP President Dr. A.M. Khan, all guests, speaker, colleagues, and dear participants. It is my great honor and privilege as the Organizing Secretary to welcome you all to 33rd National Congress of Parasitology. So this year Congress is special because we are celebrating 75 years of CDRI and Golden Jubilee of ISP. Hosting this Congress during such an important year adds a deep sense of pride and responsibility for all of us involved in its organization. Our theme, Recent Advances in Parasite Biology and Drug Discovery, reflects both the progresses we have made and the challenges that still lie ahead. These diseases continue to pose major health challenges, and with emerging drug resistance, there is a growing need for new therapeutics and stronger collaborative research. This Congress aims to provide exactly that platform. We are delighted to host eminent scientists, young researchers, and students from across the country. Over the next few days, you will engage in an exciting scientific program featuring plenary talks, special talks, invited lectures, flash talks, young scientist awards, covering diverse areas of parasitology. A special highlight of the Congress will be Oration Awards. So this year 3 Oration Awards will be given. Dr. B.N. Singh Memorial Oration Award, which is awarded to Dr. Krispal Karmodia. Dr. B.P. Pandey Memorial Oration Award, which will be delivered by Dr. Sukhbir Kaur, and Dr. K. Hanmanta Rao Award will be delivered by Dr. Harpreet Kaur. And there will be another young scientist session, so winner will be decided there. Organizing a committee of this scale is a collective effort, and I am deeply grateful to everyone who has contributed making this Congress possible, especially our director, Dr. Radha Rangarajan. Her unwavering support, guidance, and encouragement throughout the preparation of this event. I would like to express my sincere gratitude to Dr. Saman Habib. Her key role in planning and phrasing the scientific sessions, and also designing the panel discussion which we are going to have on third day of this conference. And also we are celebrating 75 years of parasitology research at CSIR-CDRI. So thank you, madam, for this. And a very special thanks to Dr. Mrigang Srivastava for equally sharing the responsibility with me. And I also thank Dr. Sabir Ali for taking most of the responsibility on his shoulder, and all the organizing committee conveners. Of course, a detailed vote of thanks will follow after the valedictory, but all the conveners, Dr. Anand Kulkarni, Dr. J.B. Pratap, Dr. Niti Kumar, Dr. Rajkamal Tripathi, Dr. Sachin Kumar, Dr. Vidyut Purkat, and all the whole organizing committee. So I thank everyone, and also thank all the participants for their enthusiasm and contributions, which truly brings this Congress to life. I hope that your time here is productive, engaging, and inspiring. Thank you, and I wish you all a successful and enriching Congress. Welcome you all. Thank you.
Thank you, Satish, for your welcome address. So we'll move ahead with the function, and the next in line is the release of the abstract book of the 33rd National Congress of Parasitology, by the dignitaries on the stage. So may we have the abstract book, please? So a big round of applause. This has been possible because it's your abstracts and your hard work that makes this abstract book so pleasing and interesting. Thank you so much. So as we all know, the Indian Society of Parasitology has its official journal known as the Journal of Parasitic Diseases. So this is also the golden jubilee year of the release, or since the inception of the JOPD. And we have with us Dr. S.L. Hoti, who is the editor-in-chief of the Journal of Parasitic Diseases. I now invite Dr. Hoti to please come onto the stage and release the golden jubilee issue of the JOPD, celebrating its 50th year since its inception. May we have the golden jubilee issue, please? So JOPD, as you all know, is published in collaboration with Springer Nature. And so we have a very, very long partnership with Springer Nature. And this is a partnership award that was given by Springer Nature some time back to our editor-in-chief. I request our President ISP, Dr. A.M. Khan, to present these Springer Nature Awards, the long partnership awards for GOPD, to our editor-in-chief, Dr. S.L. Hoti. So thank you, everyone. So I request Director Madam and our Chief Guest, Professor Ganguly, to please stay on the stage as we begin with the inaugural address and the inaugural session. Yeah. I request to move forward. I request Dr. Saman Habib, Chief Scientist in CSIR-CDRI, to please join Director Madam and our Chief Guest onto the stage to begin with the inaugural lecture. So the inaugural lecture by Professor Ganguly, followed by that of Dr. Bhupen Tripathi, would be chaired and co-chaired by our director and Dr. Saman Habib. I request them to take forward the proceedings of this program.
I have the honor and privilege of introducing Dr. Nirmal Kumar Ganguly as the speaker for the inaugural event and our chief guest for the session for the event. Of course, Dr. Ganguly needs absolutely no introduction. Everybody in this room knows about him. He has had a distinguished career, held many roles. We know him for his various positions, including Director General, former Director General, ICMR, former Director, PGIMER Chandigarh, former Director, National Institute of Biologicals, former President of JIPMER and Asian Institute of Public Health, Bhubaneswar. So he's really had a very remarkable career in that he has not only been at the helm of, not only has he pushed the cutting edge of research, but also he has played many leadership roles. He's been either a chairman or member of several national and international committees, including Grand Challenges, Scientific Board of the Gates Foundation. He's been chairman of the Vallabhbhai Patel Chest Institute Stem Cell Committee and a member of the Regional Task Force on Diseases Targeted for Elimination. He's also been in the Global Working Group of the Advisory Committee to the Director of Centers for Disease Control in Atlanta. Professor Ganguly has published more than 775 research papers. He has supervised 130 PhD students or either as supervisor or co-supervisor. His major areas of research have been tropical diseases, cardiovascular diseases, and diarrheal diseases. In fact, one thing that strikes you about his career is that he's never hesitated to work on a different problem. And while talking to him yesterday, he basically said to me that, you know, if you're open to working on interesting problems in public health and disease biology, then essentially you have to be open-minded about learning new tools, and then you can start working in any area. So following that, his interests encompass disciplines such as immunology, biotechnology, and infectious diseases. Professor Ganguly is Fellow Imperial College of Faculty of Medicine, London, Royal College of Pathologists, London, International Academy of Cardiovascular Sciences, Canada, Third World Academy of Sciences, Italy, and International Medical Sciences Academy, New Delhi. He was President, National Academy of Medical Sciences, New Delhi, Indian National Science Academy, New Delhi, Indian Society for Parasitology, which I mentioned earlier. In 2015, he was selected for the award of Helmholtz International Fellow by Helmholtz Center for Infection Research, Germany. And of course, he's received the Padma Bhushan from the President of India. This was in 2008. So we're delighted, we are very privileged and honored to have Dr. Ganguly in our midst. I invite him now to deliver his lecture.
बैक ये पॉइंट है प्रेस करके रखा
Sorry. Honorable Dr. Rangarajan, it was absolutely very delightful experience to meet you and talk to you. I had met your father not as a friend or other, but as a spectator on the stands and while he was on the podium. So very nice to see you in person. And Dr. Saman Habib has been my favorite scientist here. used to work on epicoplast of the malaria parasite, which is also found in Toxoplasma gondii, and one of the very eminent scientists in our country. I also recognize here a lot of eminent scientists. Dr. Hoti, who started with the VCRC Pondicherry and was the director of the Belgaum Institute of ICMR. Dr. Khan, the president of the society. Dr. Shamal Roy, a very eminent scientist in the leishmaniasis. Dr. Bhattacharya, Dr. Singh, Dr. Das, whose father worked here, and he had taught me the axenic culture of he had two axenic cultures, one on Giardia and another is an amoeba. So he taught me how to do axenic culture. Later on, it helped me to establish the amastigote culture in the leishmaniasis in PJ Chandigarh. I think Dr. Bhupen Tripathi has already been mentioned. He has one of the major source of encouragement and funding in the particularly in the filaria and leishmania areas of the research and malaria, and so many other people. And I think Dr. Shailaja Singh is also here somewhere. And the Sukhbir and Harpreet were two my former students. So Sukhbir I can see here. So it's very nice to see all of you back here. As Dr. Rangarajan said, the Lucknow was my hometown, and this was the CDRI was my home almost, and it was great to come back here again. It is important that when we are talking of history, we remember the stalwarts of CDRI. It started with Dr. B.N. Singh, who brought in the amoeba research and was one of the very few earliest papers on the acanthamoeba, which infected brain, was published from here. And they were towering personalities. And so the other stalwart who used to work here was in the area of filariasis was Dr. A.B. Sen. Dr. A.B. Sen established the filaria model, and he had two assistants, Dr. Katyar and Dr. Chatterjee, and all of them were later on. This area was taken up by Dr. Bhattacharya, and then we have Dr. G.P. Datta, Dr. Puri, etc. in the area of malaria research, and Dr. Das, as I have told you, was working on amoebiasis and giardiasis. So the plate was full, all of these. And later on, the new crop of scientists who came up. And in the leishmania research, there were several people who were working on leishmania research. Even Dr. C.M. Gupta, as a director, went on and established his work has been recently recognized, and he established how the leishmania enters through the actin pulverization. So all of this work emanated from here, and as Dr. Rangarajan said, that the drug on malaria, which ultimately got the Nobel Prize, the Chinese lady who did it got Nobel Prize, but this there is a history about this. Dr. Nityanand stole some seeds from Artemisinin from China and brought them here, and that really didn't give the active principle which we needed for that. So here the research was started by the medicinal chemistry group to make endoperoxides. One of those endoperoxides had gone to Ranbaxy and now to Sun Pharma, and Sun Pharma is marketing that. But there are two drugs which have come up which give tremendous hope in the area of malaria, as Dr. Rangarajan said about the Novartis drug. Of course, now all drugs are ICTs, and there is one from GSK which is in the offing, so it will be very nice to have those drugs available. So there is a very rich history. It even now it provides a lot of challenges and opportunities to move forward. I will. The Leishman research was also in the biochemistry department over here, and Dr. Singh etc. Later on, Dr. Rastogi also tried his hand a little bit on that. So the large part of the institutes were working on those areas, and now Dr. Haldar said to me that there were groups over here who are carrying on the banner, but mostly on the basic research side of this. But basic research is also very, very important. So I was wondering, I had brought in two talks. One was the basic science research, and another was inspirational talk, which Dr. Tripathi actually identified me to go and give a 7 minutes talk in Sri Lanka, because this year there has been a very great good news about the leishmaniasis, the visceral leishmaniasis, the Indian subcontinent variety, has been eliminated from the entire Indian subcontinent. There was some good news about malaria also from the Indian subcontinent, that malaria almost has been eliminated many of the Southeast Asian regions, and China, the Maldives, even Timor-Leste, Sri Lanka. I met Kamini Mendes in Sri Lanka, and she is saying that there is some laxity which is coming up. So some imported, there is no local transmission, but some imported cases are coming in. So parasitology is exciting, and the society also took place. There are two more workers which Dr. Khan reminded me, and I know talking to them and visiting them often, Dr. Siddiqui and Dr. Shamim Jairajpuri, both of them were in the Aligarh Muslim University. Or Banaras, of course, was also a very important area where the leishmaniasis research was carried on. So I will identify the institutes, but there some or other I had missed the Aligarh Muslim University, but I recognize that its contribution was tremendous in the area and to the society also. But society actually took place here in that, and there was a meeting between, it germinated here. There was a meeting between 5 or 6 people, and ultimately it took place. I'm so happy that it's surviving and it's holding its meeting, and Dr. Khan, congratulations for holding this in CDRI in its very important moment. So when I discussed with Dr. Haldar that what should I talk, I have a basic science paper which was done by Ivanka Madan in my laboratory in Gangaram, and or should I talk, give an inspirational talk? He said, give an inspirational talk because it is an inaugural address. This morning he said that you can do both because you have 40 minutes, but after 10 minutes or 40 minutes, I went into the history path. So I will give this inspirational talk, but I have left my slides with him so that this could be taken up by those who are interested in this. So let me go. This very good news actually came after very hard work, and one of the heartening things to say that majority of this research was done in Indian subcontinent, mostly in India, but Indian subcontinent, and like the filariasis research was done at one time, majority of it was done in the Sri Lanka, in Bugya Pahangi and others. There were some leaders over there who carried out, and they were the first to also conquer this. So let me go a little bit about, since we are in the history path, let me go a little bit towards the history. So one of the world's first chemical drug the urea stivamine, was discovered in 1922 by Dr. U.N. Brahmachari in the Calcutta School of Tropical Medicine. This actually changed the whole scenario. This remained the bedrock drug for leishmaniasis for decades. At this given moment, in Africa, the SDG and paromycin combination is being used because amphotericin B doesn't work that well there. So this was what happens in 1950s. Kala-Azar nearly was controlled due to DDT spraying, and up to the time I was in medical school, although as a child I used to live in Gorakhpur as a child, so I was having malaria constantly and was given that very bitter drug quinine mixture in those days. That was the only drug available. We used to run around and we were pinned down and were given that drug. But after that, during my medical school, neither I saw malaria nor I saw leishmania cases. It came back with vengeance in 1970s, 80s, and at that time, the resistance to stibagluconate was also noted. One of the big personalities was in 1980s, 1990s, Dr. C.P. Thakur, who was a cardiologist. But in Bihar, he used to see these cases, and there was a great misery because it affected poorest of poor. At that time, it affected many others. So he devoted his time, and majority of these drug trials, which were almost world-class drug trials, he carried out. And he was drawn into this because some of these drugs created a tremendous cardiotoxicity. As a cardiologist, he saw these cases, and then he thought that he should devote this time. So we are very grateful to him. I'm also grateful to him because he came as my minister when I was the DGI, and both of them then got together to end this disease with a vengeance. And I will tell the story later on. He said that let us conquer this, and we got the miltefosine at that time. I will tell the miltefosine story a little more later on. But what he did later on, that he said that when we had this oral miltefosine, then we thought that we have now an oral drug. We could perhaps eliminate it. We could go to the grassroots because it affected the musahars. Musahars are the rat catchers, and they take the grains out of the rat burrows. They don't have a house, they don't have television, they don't have telephone, they don't understand what we say to them, and they were mostly unserved. And he said that musahars, they go to Bangladesh, India, Nepal, and these are the most affected countries. Let us do an agreement with all the three. So one of the very important cross-border agreement took because of him. And this miltefosine, we got it, myself and Dr. Thakur, although we saw all the adverse effects, but at that time I got the first lesson in my life in the preclinical toxicology assessment that even if you have some toxic effects, even then you can make into a drug. Like all antitubercular drugs have some toxic effects. Some malaria drugs have also toxic effects, but we could put this into the program in a record time of 4 years. I've never seen an NCE, although it is not an NCE, it was a breast cancer drug. Those who worked on parasitology can very easily move into cancer biology. It's a cell, and the cancer is also a cell. So I remember Dr. C.P. Thakur and miltefosine, paromycin. I talked about both of the development I was involved. The miltefosine was done by the Janta Kwabang. Her picture will come up somewhere. And the paromycin was with the One Health. It was moved by the Gates Foundation, but WHO was not involved into this. So Sudhir Bhattacharya, myself, and others put the paromycin also And of course, one of the central figure was Dr. Shyam Sundar. He will come up at a later period. So these were the two guys. There were three guys actually. The first one was a Scottish guy, William Leishman, who detected it in a case in a Dhamdham, Calcutta. But it was not a live case. He did it in the spleen of that. And then it was also detected. There is some dispute. It was first in Bangladesh, Jessore. So it was under a Schuster. Then Charles Donovan identified it in an actual sample and also in a postmortem sample, and these were known as Donovan bodies. So the identification of phlebotomous as a vector, the Leishman body, everything was done over here in this country. So I will also identify Dr. Bimal Bachawat. Shamal Roy has worked under him. He was a glycobiologist. So Dr. Bimal Bachawat, very few people, everybody talks about Gilead, the amphotericin B. He created the liposomal amphotericin B, because amphotericin B used to work very well. But Dr. Bachawat, being a glycobiologist, solved the problem, because this liposome need to be around 120 nanometer. If it becomes 200, 300, it becomes a problem. But when you freeze dried it, it enhanced in size. And the trick was that it need to get entrapped into a bilamellar membrane, and in this bilamellar membrane, it need to be released slowly. So this was the trick. Dr. Bachawat solved this trick, but he could not make it at that time. Very few companies were interested in this, and so he gave it to Dr. Kheedsagar. Nilima Kheedsagar, many people know, is a pharmacologist, was working in KEM Bombay. So Dr. Kheedsagar was given a fund to make it. And she did make it, but then it was not a commercial preparation. It remained a research thing, and in between the Gilead guys made it very well. After that, many Indians companies tried to make it, but they were not very successful, in the sense that when you, like Sun Pharma made it, several companies made it, but when you gave it to the animal died, because it was released too quickly. The trapping, which was one of and the other thing, to maintain its size and maintain that yellow cake in the freeze drying was one of the very major. I learned that freeze drying is a really major science, which I learned during the cholera vaccine. It was even in the Netherlands and many other labs, it was a challenge to freeze dry them. So, but now this has been solved. Indian companies are making it, because Gilead might exit. Gilead is promised to, and here the WHO negotiated with that, and amphotericin B, liposomal amphotericin B was available free to all Leishmania cases everywhere. Blood donations for the neglected tropical disease has been a great thing. Like Mexican donation helps in the filaria elimination program. So Dr. C.P. Thakur I have already talked about. There was another guy known as T.K. Jha. T.K. Jha was equally important person, but T.K. Jha was not a politician like Dr. C.P. Thakur. And but his evaluation of efficacy, toxicity, and the compliance, because they have to be given multiple doses, was exemplary. So he also established the relapse patterns. Relapse patterns in leishmaniasis was a very important problem. Then came Dr. Chopra. At that time, Dr. Chopra and Dr. Napier were in the heydays of the Tropical Disease Institute in Calcutta. And I remember that many people at that time went to study. Our director, Dr. P. N. Chutani in PJ Chandigarh, also went there. And I will give you an interesting story how I failed one of his interviews. He had a round scar on his head, and he used to ask, "What is this?" So we didn't know about this. This was the Leishmania tropica, the northern India. They used to. So he had taken a diploma from the tropical school. So he was very proud of that. And this is what he asked in the initial stages. Later on, everyone ran the secret, so they used to answer. But Dr. Chopra was really, he was if we look at this, so he not only one of the things he found out that these bodies were found more on the nodules, but were very scarce in the macules. And it is even true now. And the PKDL was all actually documented by him, and the mucosal and periorofacial involvement was also identified with him. Here I must remember, although his name has not come in this slide, Dr. K.P. Sengupta, who was a pathologist in the SSK. He set up the pathology of the whole thing very nicely, and later on their name are not coming up in the Safdarjung. Dr. Iyer set up the skin pathology. So all the PKDL cases around the country, they used to go to Safdarjung, and after Dr. Dr. Iyer, Dr. Siramachari, Dr. Mukherjee, all took up the skin pathology, and and they went on. So they did leprosy also, because leprosy also resides in the skin, and this one also. And later on, Dr. Poonam Salhota, she was the last one there who carried out this particular thing, and she created a lamp test, which comes as an important diagnostic test. So there is another one, Dr. Napier. Dr. Napier and Chopra at that time there were no diagnostic tests. They did the aldehyde population test. So aldehyde test was, and there was a guy known as Adler. He created another test where the promiscuous they combined to make a syncytium. So these were the two very early tests in the history of the leishmaniasis. And then comes Shamsundar. Shamsundar stood as a colossus. Almost every trial in the leishmaniasis has been done by him, and not in so much in Banaras Hindu University. He had his network in the Muzaffarpur, and he did most of his trials in the Muzaffarpur. He was favored by almost all the guys who wanted to do drug trial. Unethically, he did multiple drug trials on multiple drugs, which you can't do because but there was no other person than him. He was very good, so he did it, and he also had a laboratory there. And Dr. Shyam Sundar's major contribution is that he introduced 10 milligram amphotericin B as a single shot drug. Now some people are going for 20 milligram, but that actually changed the scenario. That brought in a first hope that you can use amphotericin B one shot to cure leishmania infection. Although leishmania and HIV/AIDS, which combined it together, put up a big challenge. It really this didn't work so well, but that's a different story. And Medicine Science Frontiers, they really created a lot of such studies around the world. So that particular thing also was very helpful. Farooq Modabbar must be remembered. Farooq Modabbar has just died. He was one of the pioneers who came many times in India, in ISCB also, when you used to do your things. Farooq's two contributions I remember. One was that he combined with me to do these combos was he. I established the first DNDI in India. DNDI, when it was established, the TDR guys came to me. I was in the board of TDR in the Geneva, that please oppose this. What is coming up like this? But I found that DNDI was, the TDR didn't do basic research in the drugs. DNDI did, and DNDA funded some proposal in CDRI also at one time. So the DNDI, through the DNDI, he created these combos: paromycin, miltefosine, amphotericin B, and all these trials were done here. And because of this agreement which was done by Dr. CM Thakur, we could do these trials also in Bangladesh and Nepal. So multi-country trials are important. So it was done. So Farooq was known for that. The Farooq was the first vaccine in leishmaniasis, which was done by Farooq Modabbar. And Farooq Modabbar at that time could do it in Iran. This was Shah's time, and it was possible to do it in Iran. And he did the first trial in Iran, but it was tropical leishmaniasis. So it was not but he remained engaged in that through DNDI and also the TDR. So his contribution is immense. Jantra and me got together, as I told. I was in the TDR board, and Jantra was also there in the TDR. So we got together for the miltefosine and several other major interventions. And one of the things which Jantra did, which was really very, very important, was she created a network of excellent. We did it in Bihar, and there was no laboratories there. So the laboratories, the field laboratories, the detection systems, and that created the active detection system for leishmaniasis, which was very, very important. So Jantra was a pharmacologist, a clinical pharmacologist from Thailand. And this was done at the world standard. The DSMB was an international DSMB, and it passed everything through flying colors. All centers, training, the samples, etc., the PD, everything was done world class. So Jantra should also be remembered. Now Jantra looks at the Suman Rijal, uh, is in WHO at the moment, Director Communicable Diseases. He's a clinician, and his contribution in setting up these systems in Nepal was immense. The Steve Reed, who was the ED director, Steve Reed created the first Leishmania vaccine. And myself, Sanjay Singh, and him, Sanjay Singh, incidentally did his PhD over here. So Altaf will be perhaps also here. He also did his PhD from here. Two stalwarts. And Sanjay Singh and me had several meetings with Steve Reed to find out a model where we should test the vaccine. So one of the challenges in the leishmaniasis is the asymptomatic infection, as it is anywhere, and about 3 to 4% people get activation, and so he said that let us do it in that, and let us see that whether this is eliminated or not. This will be possible to do it in a small number of people. And at that time, the GLA, which is one of the adjuvants, because adjuvant is the real misery everywhere, where the major adjuvants are occupied by the GSK and not available in other major drug companies. CDRI at one time had an adjuvant research center. I don't know whether it is there or not. So CM Gupta, etc., Dr. Nityanand, Dr. Khanna, they were working with the Muravil peptide, and these Muravil peptide, etc., with the other ingredient from mycobacteria were forming the basis of that. So adjuvant, and there was another group. There was a peptide group here. So the peptide group also created that peptide group. Dr. Katti was there, Dr. Dr. I'm forgetting his name. They formed the Hema Pharma, the first peptide company in India to making oxytocin. So they also started to create peptides as a adjuvant. So adjuvant research is still a very important part of the drug research. So so the Simon Croft used a very nice DNA kind of a thing which is known as E-Slim, which was made by Mologen. So he created a network in which Shamal Rai was part of that, and that Shamal Rai's discovery KMP11 is now part of many of the vaccines. So they were looking at the T cell responses to the different the setting in the clinical settings to see, because the immune system is a very, very important part of the Leishmania research. Because cell-mediated immune system is the protective system. There were no assay at that time. So in his the Simon Crowd's advisory board, I was part of it. Pradeep Das was part of it. Shamal Rai was part of it. We met in the tropical school one time. Or Shamal Rai, you were a part of the team in that. And there were two major challenges. One was that we didn't have Leishmanian antigen, which was known as the Montenegro antigen. This was actually a very good test which you do in skin and see whether the cell-mediated immune response has come to that. This has vanished. The Iran had this. I had a student there, Dr. Bahman. He did PhD with me. So I went to Iran to beg for some Leishmanian antigens, and Iran refused. Like Lalji Singh wanted to, the Indian cheetah has disappeared. Lalji Singh wanted one hair of a cheetah to clone a cheetah over here. Then I was the CCMB RC chairman, but they refused, so they didn't give it to. The Pasteur Institute, the Raji Institute had this. So now this has been solved. The Sanjay Singh has prepared a Leishmanian antigen, and this Leishmanian antigen is there. But what is very important, that there were no cell-mediated immune response, good validated cell-mediated immune response to Leishmania. So this was developed by Simon Croft with the help of a Swiss laboratory. This was on the pentamer assay kind of thing, so that the T cell integration was maintained into this, and this assay is now available for people who do the vaccine research. So then the David Moynihan, who was important because he criticized everything, whosoever did whatever. So he was a conscious keeper. He criticized everyone, everything. So David Moynihan was in the Liverpool School of Tropical Medicine, and then Saurabh Jain, Jose Alvar. Jose Alvar was a real rock. Jose Alvar in the Columbia developed the opening speech of the history, which I'm repeating over here. That different. He told different things. I'm telling different things. So mine is more India centric. So Jose Alvar and me and others, we investigated this Bhutan outbreak of in Indian culture. We investigated the Bhutan outbreak of the leishmaniasis, which is a very big challenge for the future. The climate change is a very big challenge for the future. It is seen in the malaria. Malaria now is in Jammu Kashmir. Malaria is now in the Bhutan. And this breakup was in the Bhutan, and then there was another breakup which was in the Shimla, and these were local transmission. Mind you, they were not just imported cases. So climate change will remain and is an important challenge for the future. And so Saurabh Jain, I forgot, he did one very important part. You know, the people from Bihar, Eastern UP and other, they go around Punjab and other places, and they are migrants. And the Musahars are also migrants. So what happened that they were he used the NSP, which was in the polio. In the polio, the migration was handled very well. So he handled that to ultimately track the Leishmania cases around. So at that time, the local office of the WHO became very, very important, and he created that. He is now in the WHO headquarters. So I have talked about many people. So these are some of the very important part of the. I have identified almost all the guys. RMRI Patna, of course. I have Antwerp and London School and Liverpool School. Jawaharlal Nehru University, Madhubala, Shalini Singh and others. They kept Poonam Sarotra in Safdarjung. School of Tropical Medicine, Calcutta. Dr. A.B. Chaudhary, who was my mentor, he was the father of parasitology in India. Uh, uh, Started there, but one of my PhD students, Dr. Nandi, went there for and carried out the Leishmania research. SSK Hospital, now there is Mitali Chatterjee there. She does on the biology, and Dr. Sen Gupta was there, KP Sen Gupta. National Institute of Medicine, NIPSOM in Bangladesh, Nepal Medical Research Council, Central Drug Research Institutes. I have put it in the last because if this was not there, Leishmania research would have gone. So I'm almost going to the end of my time, I think, and so I will hurry a little bit. So this is the graph which has come up. The same graph is in Nepal and Bangladesh, except with few cases in Mymensingh which was coming up. Bangladesh has passed through the elimination thing, and in next 3 years, hopefully we will also be. In 2023, we had the last case, so in another year or two, we might also be done with that. So PKDL remains a problem. The major problem with the miltefosine was this, that miltefosine, when you looked at the complete sequencing when you did, the area which does the drug resistance, it is same in the pentamonials. So there is a very big cross resistance. Shamal Roy, that's a very favorite area. Very strong cross resistance with the drugs in over here, and the pumps which push out the drug are very, very active over here. So there are several mechanisms which work. So miltefosine, there was the relapses were coming up. So that hope was particularly, it could not be given to pregnant women. It was also, if your hemoglobin, which was mostly 6 milligram dose less, it didn't work there. So the PKDL remains ongoing, but PKDL has come down, and it has come down in Africa also. I'm one of the reviewers in the Paul Case vaccine over there. So we wanted to do it in Sudan and Mohammad Musa there is one of the very reputed Leishmania scientists. He because of the Sudan problem, but even there the PKDL has come down. So it has come down, but there is a note of caution. It still the macular. We involved in India the dermatologists to really get all these PKDL cases and treat them. They were treated with miltefosine, and here I come back again with the toxicology thing. We found that miltefosine does diabetic retinopathy in the rat. But we thought that in humans it will not give because the doses which we are using, it also reduce the sperm count. And we did the sperm count retrospectively, we didn't find. But now when miltefosine was done for the PKDL, complete treatment, the diabetic retinopathy came up. So it was a big setback. So now these combos are being advocated because it's a long treatment. So PKDL will remain a challenge. So vector control, I will just say one thing, that these vectors were very witty, and with one of the major discovery was that there was a challenge that India went on using DDT, and Bangladesh and Nepal didn't use DDT. They went to the pyrethroids, and now India is using pyrethroid, but there is a challenge that pyrethroid you need a pressure pump because they are in the walls and not the not the foot pressure pump, a good pressure pump. You need a good training over there. And these guys are now living outside your house. So but this training was very, very important, and Kalakor guys, they did it. And another persons who should be remembered are the Kala Mitra, and we should always remember Bhupen and the Bill Melinda Gates Foundation, which provided tremendous partnership over there. This partnership was of many UN agencies, the DFID also, the CARE, all kinds of people. If these partners were not there, the Kaladar perhaps would not have been eliminated, and somehow or other we need the financing and these partnerships on. So the current landscape is there are new surveillance strategies and two diagnostics. One is RK39. There is one urine variant also, but the LAMP test, which should go, because RK39 doesn't give you the cure for the disease, but LAMP test gives early and also gives cure. And there were the xenodiagnosis was done by Dr. Mandal in the ICDDRB. He should also be remembered as one of the major worker over there. The xenodiagnosis actually showed that these macules can transmit infection. So this remains a real major thing, and introduction of better diagnosis which are now available, which are isothermic, and they could do it in the skin as well as otherwise, was important. So there is one major thing. There is a funding now through the Kigali Declaration. There is a funding now available. There are two Kigali meetings which have taken place for the neglected tropical disease. So financing has been done, and these are the important partners, the PCI, Global Health Strategies, because the communication and advocacy is very important in this. Because those who are the target, they don't understand our language. As I said, that there are a lot of government incentives. They give them house, they give them building. If you get treated, you get money. The person who treats also gets money, but they don't know how to use that. They become pauper by the time they are already pauper. They go to the people who are normally the private the registered medical practitioners, quacks, so they're treated by them. But it has been now a policy to involve them also. Both it worked wonder in tuberculosis, it worked wonder. Since I'm moving ahead of time, this is the last slide. I will talk about the vaccine development. Nobody talks about vaccines anywhere in the world because this is not every time it is a drug related control. So I will talk about three. One, I have already talked about the Salman-Cough Mologen. It really didn't get funding from the European Union, so it was dropped, and Mologen moved to cancer because the same platform was originally for cancer. The one which I will be talking about is the Paul Case one. I was the chairman of the Paul Case. Paul Case construct includes KMP1 of Shamal Rai. It also constructs a synthetic part, and it is in an adenovirus, adeno3 adenovirus vector construct. So it was a beautiful thing. Paul K. involved immunologists. He involved like the King White, as he did the wand for the malaria resistance. The Paul Gurin actually sequenced all the Leishmania things in this particular thing, which happened in Mekong Delta. This one happened in the Africa. So in Africa, there was Sudan, Ethiopia, Kenya, Uganda. These were the countries where we were doing it. They were doing the spinning puncture. It was officially allowed. In our country, it was not allowed. So the bone marrow puncture and this both were allowed there, and our model was very nice. That do the skin immunology, and what you do that you treat for 14 days with the SDG and paromycin, and after that give this and prevent PKDL formation, because PKDL with SDG, lot of PKDL comes up, and it was put as a therapeutic vaccine, but then PKDL disappeared. So then we had to design it as a preventive vaccine, and then Sudan crisis came and there were not enough cases anywhere else, but it's still going on. But the main thing which is now at the moment is the Hiranakasi's vaccine. So Selva Pandian joined me when I opened the Center for Molecular Medicine, American Financial Aided Institute. I had Selva Pandian from so it was a centrine, uh, double knockout centrine gene in the Leishman adenovirus, which prevents MS promastigote to form a mastigote. And it was a beautiful, everything was done in that. But then there was a problem. There was always a fear lurking that there could be a reversal. Because at that time, Nadira Kohanvira reported the Jaffna cases where there was a skin infection which had a visceralization. So so that particular challenge prompted Satoskar, Greg Matelowski, and Hiranakasi, and now and Selva Pandian also to create something in the Leishmania major. So there is a now I was the Indo-US Vaccine Action Chairman at that time. So I repeatedly got this funded some or other. So this vaccine is now there. This vaccine has now undergone dog because there is a dog leishmania vaccine. So this has undergone the dog testing. It has also has been tested in Tunisia. It has gone in Tunisia in humans. It is being made by the Genova in a large amount, awaiting that what will that when it will be tested in India. Nobody actually is supporting this at this given moment, because the only way it can be done in India quickly is a human challenge. So I will end by saying I will not deal by basic science laboratory data, but I just gave you a glimpse that it was the sphingosine sphingosine phosphate, which actually deals with the apoptosis, apophagy, and also the inflammatory immune responses controls. This was controlled by the by the sphingosine kinase. There are two kinases, but it is a completely parasite thing. It was the same as Chandrima Saha did for one of the molecules, which was found in humans also, but it is not found there. We have used the inhibitors for this. We have also used CRISPR-Cas9 to get by a vector to silence this, and what we feel that this will be a great target for the future, could also be a vaccine target, but may not get through regulatory pathway because a lot of things have to be cleared. So that whole set of slides are over there. So I finish it over here by taking you to a journey. Actually, Bhupen gave me 7 minutes in Sri Lanka. He couldn't talk about it, but I thought that I will talk about that in India. Thank you very much.
So thank you so much, sir, for your interesting and of course for taking the So thank you so much, sir, for taking the audience through the interesting journey of elimination of Kazar. Our director would facilitate you.
Good morning. It's a pleasure for me to welcome Dr. Bhupen Tripathi to CDRI and to this conference. Dr. Tripathi is a public health professional and currently works in the Gates Foundation as Deputy Director for Infectious Diseases and Vaccine Delivery. His focus areas are elimination of TB, malaria, lymphatic filariasis, visceral leishmaniasis, measles, and rubella. He also works to improve routine immunization coverage, including new and underutilized vaccines, and also participates in infectious disease surveillance. His current role involves working closely with the government health system, research organizations, academic institutions, and the private sector to design, develop, and introduce new diagnostics, drugs, vaccines, and treatment therapies towards control and elimination of infectious diseases. Before his current role at the Gates Foundation, he contributed to polio eradication with WHO, measles elimination with the UNICEF, and maternal and child health projects with USAID supported projects. He holds an MD from MLN Medical College in Allahabad and advanced public health trainings from the London School of Hygiene and Tropical Medicine and the University of Geneva. He also served as board member for the American Society for Tropical Medicine and Hygiene and now works with the Gates Foundation in India. Welcome, Dr. Tripathi. Please come here for your talk.
Yeah, thanks. You want me to be placed here? So you can. Hello, hello, hello. Excuse me. Excuse me. Hello, 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 hello. Okay? Huh? Is it okay? Yeah. Fine, thank you. Very good morning to all of you. Let me start by thanking Dr. Radha Rangarajan, Director of CDRI, for inviting me for this August meeting. Thank you so much, and thank you, Co-Chair Madam Sam, for the introduction. Thank you so much. All the eminent scientists sitting here, it's always a pleasure meeting you. And today's most important, I would say, is the young researchers who are sitting all over here. So very happy to meet all of you. And from the Gates Foundation, my best wishes to all of you on the 75th anniversary of this institution and the 50th anniversary for the society. So really congratulations to all of you. Well, friends, it's always a pleasure, listening to Ganguly sir. Like he's encyclopedia, and as a institutional memory, when you listen to him, I think we enter into a sort of film which is going the historical story of things. Sir, thank you so much for sharing your experience and all the things which we have. I think once elimination is achieved, we will need some sort of memory, and these recordings should be saved because the young generation will not listen what they had, you had done for the disease elimination. So with this, friends, I want to start my presentation. It's a very short presentation and largely for the young audience, honestly. So we will be talking about neglected tropical diseases. And the reason why I am raising this as a topic is because we need to prioritize the neglect. And the question is, okay, why and what is the reason we want to do this? Well, let's start. Excuse me, can you please help? Yeah, it's not changing. Excuse me, can you please help? Change नहीं हो रहा है. Yeah, sorry for the delay. Now, friends, this slide has pictures from 24 countries. Like not one country, 24 countries. And my question to all of you is there anything common in these pictures? If you notice closely, each of the picture is depicting something. Very good. What else? Very good. Next. Hygiene. Yes, very good. Very nice. Good. Very good. Anybody else? And it's a real picture of the current decade, not like a very old picture. So Gap Finder is a website. If you go, Gap Finder has pictures of all the countries based on every parameter: socioeconomic, urban, rural, age-wise, sanitation-wise. Very nice. So if you want to learn about any country, just go to Gap Finder, and just go through the clips. You will learn a lot. So well, friends, you are absolutely right. These people are living in conditions which are highly, likelihood for neglected tropical diseases. And there are around 2 billion people in the world today who are living in these situations, these conditions, including our own country, India. Now, as all of you must be aware that these are neglected tropical diseases, and WHO has coined them because they are mostly in the tropics, and we all understand tropics near the equator. The second is that they are not an immediate, health emergency for the developed nations. Why was COVID given so much importance? Because COVID impacted the developed nations as well. So mortality is high. In these cases, most of the cases happen in countries which are not developed, underdeveloped, or in the middle income countries. The next point is this happens mostly in the poor population. And poor population does not mean only poor because of the socio-economic situation. They are poor in their voice and their expectation with the government and society. Nobody asks them that, okay, what do you feel? They don't have space. They don't have place where they can tell their voices. So that's the second problem. The third problem in the same group is there are many diseases with stigma and discrimination. Think about leprosy. Think about lymphatic filariasis. These have so much stigma and discrimination attached. The next is, which is a promising thing, most of these diseases can be controlled, eliminated, and eradicated. Now, the good part, what has happened globally is, as part of the SDGs, these diseases have been brought up in the indicator number 3.3 and 3.3.5. One second thing happened 3 years back. WHO has come up with a specific day known as World NTD Day, and that happens on the 30th of January every year. Now, friends, all of you must be knowing these diseases. I just wanted to discuss two things on this slide. One is that we need to understand there are diseases meant for eradication, there are diseases meant for elimination, and there are diseases meant for elimination as a public health problem. And there is a very important distinction between elimination and elimination as a public health problem. And then finally, control. We only can control, forget about elimination or eradication. The most important thing is the bottom line, the color coding. If you see the color coding from the left side, green means we can achieve elimination or eradication, no problem, versus red where there are challenges, we cannot achieve elimination or eradication. Now my question to all of you is there any disease in green color, like the box green? The reason is we don't have either diagnostics or drugs for treatment or things for public health measures. If we go one by one, these are 20 diseases. WHO has added one more disease. There are 21 entities in these groups. Out of them, 12 are in our country, India. There are many diseases which are not in India. They are in the African geography. But for Indian geography, if you go through it, you can see that we were successful in eradicating yaws and guinea worm. So great job. It's all about because of you. All the stalwarts sitting in front, they are the people who ensured India reached that eradication. If you go to elimination, now let me come to elimination, and elimination is a public health problem. Eradication means there is no causative organism available in the environment. Done. Like smallpox. Done. Elimination means the organism will be there, but because of the measures what we play, there will not be a single case of that particular disease. But when we come to elimination as a public health problem, we recognize that there will be cases, more than a case, and there will be a threshold below which if we can keep it, we are very happy. So all the things what we discussed, lymphatic filariasis, visceral leishmaniasis, these are all elimination as a public health problem. The risk is that if we do not continue with our measures of prevention or control, these diseases will come back. And most of these diseases are linked with some sort of vector. For example, what sir mentioned? Sandfly. LF, all about mosquitoes and climate change, absolutely important topic. Things are changing and climate change will ensure that these diseases will be spreading beyond the endemic geographies. So that is very important. My focus to all of you is also on the control side. Friends, all the young researchers sitting over here, you have to look into it. Can we contribute towards this not only for the country but for the whole world? Now let me give you a challenge. Why are we talking about this in India today? Because in 2016 there was a very nice study on entities which was conducted, and at that time we tried to see, okay, where does India stand? At that time India was second in the population. China was in front ahead of us. But if you see the entities, India stood what? First. Is it a good thing? No. If China had more population than us, why is China nowhere in this picture? What is the reason we as Indians we are having these diseases? This is not a matter of pride for any one of us. But the question is, what is to be done? Can we come and try to see, okay, we will be doing something which will take us off from this list. Very happy to see that Kala-Azar at that time we were top. Today we are not at the top. I will speak about that as well. But the question here is, what is the problem? Let's go to the problem statement. So friends, this is a complicated slide, but I will take you through from the left side and that will be very easy for you to understand. So on the top, if you see, there are indicators for this slide. So point number 1, the color coding is that on the dark blue, these are the diseases which WHO has chosen that by 2030 we should achieve some good targets because we have to focus them. Others are still out of question and we are still struggling in these areas. Now the color coding essentially means if it is green, there is no bottleneck to achieve the target. But if it is red, that means there are bottlenecks for achieving the target. The second is about the dark color coding. If the specific disease gives more than 1 million DALYs, that is dark black. If it is 0.5 to 1 million DALYs, it is light gray, and the further light is less than 0.5 million DALYs. So this is over this place you can see over here. Now coming to the specifics. So if we see all over, these are individual diseases. Is there any single disease which is all green in terms of technical scientific understanding, diagnostics, intervention technologies available, strategy, operation, research, etc.? Is there any disease where everything is green? That is the reason. If we do not have the diagnostics, if we do not have the treatment, whether it is drugs or vaccines, if we do not have sufficient funding to implement the program, things won't reach the desired outcome. So my suggestion and request to all of you is, even if you choose any one disease, all the young scientists sitting over here, for your lifetime, I think you will spend the whole life in achieving elimination or eradication of a single disease, and you will be known as a champion for that elimination. The way we appreciate our Ganguly sir for eliminating Kala-Azar, you will be the people in the future. You can claim that yes, on one day the India and the world was at this level, and I have ensured that we reached from A to B level. That is what we want to do. So therefore, my request to all of you is, choose any one of them. There is no mention that your HOD or your guide is saying only work on this. You choose any one of them. And not only in India, you can go outside as well. Now, as compared to 2016, where do we stand today on NTDs? So this is a study of 2023 data. Well, friends, what is the percentage in India of NTDs? You can see and you can read there. 41.6% contribution is still from which country? India. We will say, okay, population is too much. That's why it's there. Where is China, by the way? Is China visible there? And it's not that China never had NTDs, by the way. They did had NTDs. But they worked on that and they have solved their problem. So as all of us Indians sitting over here, and if we aim for becoming, Viksit Bharat, Without working on entities, we cannot become Viksit Bharat. These are the proxy indicators of success. If tomorrow you talk to any politician, ask him or her, okay, pick up your constituency, let us see these indicators on entities. If they are great, you have really done development. Because entities don't happen by design. They are the culmination of sanitation, the environmental way you live, the drinking water, the vector presence, and program implementation, health system. So if the system is working well and the people are living in a nice, very good environment, entities will perhaps not be there. But if they are there, that is why we have to focus on them. Moving ahead, there was a very nice booklet which came up that if we invest on R&D, because we are sitting in CDRI, we should talk about what is the implication we can make in the world. So if we invest in R&D, 40.7 million lives will be saved and 2.83 billion dailies will be averted globally for a longer period of time, obviously. There will be 49.7 trillion in societal benefits. We are talking about Viksit Bharat. We say India has to become so many trillion economy. Well, friends, it's all linked with entities. If we do not work on entities, we cannot dream of becoming that specific, a champion. Then there will be requirements not only in India. It will be beyond India, South Asia, and many things will be required in beyond India geographies like South, Americas or Africas. So when we think about our scientific, rigor, it should not be limited to one country. You should start thinking about how can you influence the whole world going forward. Now, we are very happy to see that our government is, managing the research and development portfolio in a very big way. This is just a small graph to show how many institutions are there which are working on research and development. It is from ICMR angle, it is from all the CSIR, institute angle, it's also from medical colleges. There are n number of institutes working together. Now, the most important thing is collaboration. I may be working very good in my own silos, but I am not telling my neighbor or my, other group of members. Then we are failing as a collective. So the most important thing is that if we can figure out, if we can start talking to each other, we may speak during the conferences, that is okay. We may have some panel members who will be speaking in small committees, it's okay. But can we have a mechanism of sharing this widely? And sharing this widely not only within the scientific community, but also for prospective scientists sitting over here. That mechanism, at least today, I don't find. If we can design something where the science and the data or whatever your outcomes are coming, they are available widely. And if there is an amalgamation of data coming from n number of institutes, that science will be really helpful for all of us. On the left side, we have some data sets for South, like India and other countries. I will not discuss that. Let's move ahead. And sir has very rightly said, Kala Azar is actually going to be eliminated as a public health problem. I will repeat, as a public health problem, because if we take away our preventive measures, it will bounce back. There is no reason why it will not bounce back. Sandfly is everywhere. All across the country, sandfly is there. Whether we talk about south like Kerala or we talk about Himachal, Punjab or Jammu Kashmir. It only needs one infected person. That's it. What else? One infected individual. And sir rightly said, all the migrants of Jharkhand, Bihar, including our East UP, if they are traveling, if they are carrying the parasite, they are not aware they are carrying the parasite, and if sandfly is there, if they live in open environment, that huts which I showed, then they are going to spread this to that geography. Recently, there have been cases of cutaneous leishmaniasis in India as well, and we are worried. Because in Nepal, the western part of the Nepal, which is bordering the Himachal and the Uttarakhand area, we are finding a lot of cases there which are visceral leishmaniasis, and the situation is that they are expecting that India will also be having that. So if we work on one area, if you do not focus on another area, there is always a risk that the outbreaks will happen. So that's very important. Moving ahead, sir rightly mentioned, PKDL is still a problem. And this is my, humble request and, basically invitation to the young scientists. Think what else can be done. Whatever our senior scientists have done, we have been lucky to take the baton from A to say B. But from B to C, we all people will have to contribute. We can learn from them, all the things which were good as well as things which were not good. The way Ganguly sir explained where we failed, I think that's equally important in the scientific community discussion. That if we understand that, at least we will not do that mistake. Moving ahead, HIV co-infection does play a big role, and then we do have very good data today from the country where we can analyze the data and we can focus what we want to do. One more disease. Well, friends, I did not deliberately kept the name of the disease, anticipating everybody knows that. Is there anybody in the young audience who does not know which disease we are talking about? Please raise your hand. Everybody knows about the disease. Seriously. It's very rare nowadays to find these people traveling in the buses, trains, planes, or even malls. Have you seen anybody in the malls or shopping area or markets? Have you seen anybody? You will never find them. Why? Because they cannot move. One is the physical barrier they have. And these are, by the way, all alive people in our country in today's date. They are all living people. Imagine their life. Just for 1 minute, focus and think what will be the situation of the individual's husband or wife, individual's parents, individual's children, where they live. This is lymphatic filariasis, you all rightly understood, spread by what? Single mosquito. What was their, bad deed? Was there bad deed? Did they drink a lot of alcohol? That's why their liver went away? No. They just lived in that environment where the hygiene and the other protections were not there. And as public health, individuals, as the scientific, community, I think this is our moral responsibility to ensure nobody should get this disease. Nobody should get. Why? Because there are preventive therapies available. And this is the question to all of you. I will be, showing one slide and then the question will come up. Look at the map on the left side. There are colors. All the colors, red, green, yellow, are the districts where lymphatic filariasis is endemic. Okay, that is okay. Does Lucknow reflect in this? Or let me ask differently. How many of you belong to one of those districts which are color coded here? Please raise your hand. Please raise your hand. If you belong to any of the color coded, 51 districts of Lucknow, UP, all districts of Bihar, all districts of Jharkhand, all districts of Odisha, 3/4 districts of West Bengal, and further down. If you belong to any of those districts, the second question for you is, have you heard of Mass Drug Administration campaign, MDA campaign? If yes, great. Third question, how many of you have consumed MDA drugs in past 5 years? Please raise your hand. Very good, very good, very good. How many of you are living in Lucknow for last 5 years? Raise your hand. You are not living in Lucknow, guys? Coming from outside? Are you from any of these districts? No? Lucky. But not lucky forever. Because I will be traveling from Lucknow to your district, and if mosquito is there, if Culex is there, I will be gifting this to you. So, well, friends, the story is, if the campaigns are happening, if we don't know about the campaigns, or if we don't consume, this is annual campaign, not like once in 5-year campaign. So the problem here is people don't understand the problem. When we talk to people, I am telling you about people in the sense normal community as well as MPs, MLAs and IAS officers. Many people said, oh, this happens because of dirty water. Because if you drink dirty water, then it happens. Many said it's a generational genetic disease. Because the mosquito Culex lives in the same compound, same household. So if my grandfather was living there, my father was there, I was also born there, the same mosquito will keep on biting everybody. So once the mosquito bites, sucks up the larvae 3, gives to the new person, and the transmission pattern. We have seen so many families where the lymphatic filariasis flows in generations. And it's not because it's genetic disease, just because it's an area where the mosquitoes are rampant. And friends, do you have Culex in this CDRI campus or not? Is it there? I can assure you there are LF transmission patients around CDRI. I can tell you. Even I was carrying microfilaria. Forget about everybody else. I have been living in UP. I worked in polio for the WHO program and all that. When I came to the LF program, during the launch of the LF campaign, drugs were given to everybody who was sitting on the dais. I also took the drugs. At that time, nothing happened. I was fine. Okay, it's okay. After 3 days, I started getting itching across my body. And I was worried, what this is happening? I went to one of my friends who was a dermatologist. He said, you guys, you stay in hotels so often that you must be having scabies. He gave me ivermectin. I took ivermectin, the more itching happened. For 25 days, I was taking ivermectin and itching was happening. I went for a short course out of India where ivermectin was not available because it got finished and the itching went away. When I came back, read some more things in parasitology, then I realized, oh my goodness, I was carrying the microfilaria, and the moment these anti-parasitic drugs are given, one of the side effects is itching. Bottom line is, none of us is safe. And if you would have given time to Ganguly sir to speak on LF, I can assure you for 2 hours he would have given so many examples. How Gorakhpur area, how other areas we have these diseases. Point here is, those who are suffering in the sense having microfilaria, they should not spread to other people. And that will only happen with everybody takes anti-filarial drugs every year till the elimination happens. So my point is, it's not that because I am speaking or all these people are speaking. It's because you will have to start talking to your own community, your friends, your relatives, your neighborhood. Tell them that if you are living in a geography which is colored here, there is a risk of lymphatic filariasis. That is the story we have to discuss. Now, over a period of time, the program has improved a lot. The administrative reporting, WHO monitoring has improved a lot. We will not go into the details. And a lot of laws are actually getting eliminated as a public health problem again. It's not elimination. I wish we could eradicate. Ideally, yes, we can do it. But the way waterlogging is everywhere, the way all the engineers are developing beautiful drains for us with a U-shaped body, no cover, and then the water is always staying there, that is going to be a breeding ground for QLEX. So that's a bigger risk we have. And finally, a drug question to all of you. There is an institute in Kerala known as Institute of Applied Dermatology. They are using some local herbs and yoga therapy to reduce the swelling of lymphedema from say A to B with a very short span of time. I'll tell you, there is no modern science drug available to reduce the lymphedema swelling. There is no operation possible. We have tried debulking, we have tried plastic surgery. It does not work. But these guys are doing something wonderful which reduces the swelling. Now the question here is, what is that molecule? What is that drug? Can CDI take onus on this? Some of the researchers can invest their time, energy and brain to find it out and help everyone across the country. With this, I want to thank once again for this material. Thank you so much.
Thanks. Thank you, Dr. Tripathi. Just please wait for a second. So as a token of our appreciation, would like to facilitate the speaker. And I request Dr. Saman Habib to do the honors. I now request Dr. Satish Mishra to give a vote, to give a token of thanks to our chairpersons of the session. I request each of you to please join us for a group photograph right in the main auditorium. So please just come together. We'll have a group photograph and then we'll break for tea. And yeah, a little closer so that we can have everyone in the same frame. And we'll have a tea break after that. And once you assemble after 15 to 20 minutes, all those speakers who have their talk listed in the session 1 may please load their presentation in hall number 3, that is just across the main hall. Thank you so much.
चलो यार चार्जिंग लगा दें इसको।
चेयरपर्सन से एक रख दिया, सामने गुलदस्ता रख दिया, 2 माइक रख दिया। बजर
चेक कर लीजिए कि चल रहा है। यहाँ लगा ये भी हटा देते हैं। एक और बजट किधर लगा? ये सेकंड से करते हैं इसको खोल दे पन्ने खबर नहीं वो डिस्कनेक्ट नहीं चाहिए सर एक बार करिए सर हाँ दबाइए उसको बटन नहीं आ रही लाइट तो आ रहा अरे चल लगा तो रखा है
चेक कर लेना और माइक यहाँ पे दो नहीं है हमारे पास
टोटल चार्ज
चार क्योंकि तो 5 तो पहले बोला सब्जी पे ये
मैं ही आ जा रहा हूँ
आप दूसरा बता बताइए
ये ट्राई करिए बजट कहाँ गया ये वाला बजट ट्राई करिए भैया ये बजट ट्राई करिए अरे आओ इधर यार
फूल है ना पाँव में लगेंगे तो बड़ा खराब लगता है
अरे इसको हटा और दूसरा वाला
करिए ना अरेंज कर देना ऐसा करके इसको देखिए बताइए या 2 ग्लास पानी रख दी सेकंड वन फर्स्ट वो लोग आएंगे जी पानी में माइक को वहाँ पे पानी नहीं खुली है नहीं खुली है ठीक है स्टार्ट खत्म हो गया
और क्या लगा
understand. Actually, we will make them understand that we have to. But here for the chairman,
हाँ
and
तो ये यहाँ पे तो बेल ठीक हो गई है नहीं आप और भी काम कर रहे हैं
मेरी वाली तो और जो सब कुछ उसमें बहुत कुछ था ना वो सब कहाँ है?
अभी मैं पैक हाँ
Okay,
हाँ अच्छा तो who will make them understand will you convert
I can.
हाँ तो try to stand nearby here the moment they are here you can just take them
लाइव स्टॉप है ना?
तो आपको क्या करना है कि अल्दो आगे
नहीं
अरे ओके कर दिया
हो गया ओके हो गया।
ठीक है, ठीक है।
एक उधर
तुरंत हम ठीक है
हाँ ठीक है।
वो लाइव स्टॉप कर
तो ये इतना इन्फॉर्मेशन बट एक बार आप देख लेते हैं
लाइव स्टॉप रखें।
Just make sure कि भाई वो
नहीं,
बच्चे
ले-
सब्जी वाले से मांगे तो आपको वापस कर देंगे।
हाँ
तीली
का
बैठे देख के मैं
तुम चाहो तो रमेश सर का भी ले सकते हो क्या भी सोचता हूँ केला
हमें शाम से ले
यही दोगे तो
ठीक
हमारे पास में
हो गई, ठीक हो गई।
तो फिर आलू के लिए
हाँ
और
तो यही
मुझे
रखे
शाम को चाहिए तो तुम एक अपने पास रखो अभी। नहीं उसका बजट दोनों
यहाँ से गायब नहीं होगा कहीं और से गायब नहीं
इसके बाद बस मेरी है मेरे हाथ में चाहिए
ठीक
ये
है
और ये जाएगा हॉस्टल में इसके बाद टाइम
लेकिन
तुम्हारे हाथ
इसके
में
साथ में ये और वही था ना कोई और
हाँ
नहीं था
काला काला उसके
उसके
साथ
साथ क्या क्या था उस पैकेट में ये और कार
बस ये दो चीजें है ना
हाँ तो
बस ये
बॉक्स
ये
कहाँ है
कलेक्ट
बॉक्स मेरे पास
वो
हाँ
कलेक्ट
तो मैं
करके
शाम को मैं कलेक्ट करके मैं आपको दे दूँगा
ठीक है
हाँ
डन बस माइक सब चलते हैं बस शायद ये अच्छा ठीक है
माइक
अब यहाँ पे सिर्फ एक ही सेशन है उसके बाद लंच ब्रेक हो जाना है
हाँ
पूरे वैसे टाइमर चल जाएगा
नहीं टाइमर देखते
और
हैं
माइक
शुरू करते
और हर बार कुछ बच्चों को मतलब को अलग थोड़े मिलते हैं ढेर आए थे जो भी है 3 स्टूडेंट जिनका पोस्टर नहीं हो पोस्टर वोस्टर में ना भागना हो
अभी ठीक है अभी
बस ये देख लेना अब शो वगैरह कुछ नहीं
फर्स्ट सेशन के लिए आपने ये किया
हमने सब कलेक्ट कर लिया
फर्स्ट सेशन के लिए
मैंने सब कलेक्ट कर लिया
मैं कोऑर्डिनेट करूँ
हाँ क्या
मीडिया कौन देख रहा है
मीडिया इन प्लेस हो सकता है पहले होता आपको देखा होगा हाँ सतीश से पूछना पड़ेगा
हाँ I'll coordinate with the information. I'll just call the coordinator and then
मैं कर रहा हूँ
आप कर रहे हैं कितने बजे चालू होगा But I think we have to keep the timing. Yeah, I think these people बेटी जरा भारी ताली बजा दो so that people can come. मैं एक्चुअली बजाना चाहता हूँ then I said I बाहर से लोगों को बुलवाया wow. Thank you. मैं तो तुम्हें कह रहा था चल रहा है
Sir, should I announce?
हाँ सर, ये टाइमर बैकस्टेज मैनेज करो।
25 अलार्म है ना, 25 मिनट का
25 मिनट के बाद
हाँ, जिसका 30 मिनट का
सर वो पीछे मैनेज करो।
वो है।
ओके, थैंक यू सर। Welcome to the session on biochemistry and cell biology of parasites. We are honored to have our chairpersons, Dr. Pavan Malhotra, who is a renowned expert in malaria biology at ICGB, leading research in parasitology and the development of novel anti-malarial therapies, and Dr. Swati Patankar, professor at IIT Bombay. Dr. Patankar spearheads pioneering research in molecular parasitology and plays a key role in fostering global scientific collaborations in this field. I kindly request both chairs to be seated on the dais so we begin the proceedings. I would like to call Dr. Pavan Malhotra to introduce our first speaker. Sir.
It's our pleasure to introduce Hema Balram and other speakers of the sessions who have really done exemplary work in the field of malaria and also the drug development against malaria. The first speaker is Hema Balram, a true biochemist and who has done really great work to understand the structure function of various plasmodium proteins. And also, she has illustrated many biochemical cycles in plasmodium. It's our honor to really introduce him and request her to present the talk. Hema 25 plus 5.
Okay.
Okay.
Thank you, Pawan. Thank you for the introduction, and thank you, Satish, for inviting me to give a talk at this forum. So I'm going to talk about some aspects of the TCA cycle in the malarial parasite. Yeah, the two parasites that we have been working with to understand many aspects of both aspects of metabolism in the parasite. One is Plasmodium falciparum and the other is Plasmodium berghei. And in the Plasmodium falciparum, it's largely the erythrocytic cycle that we have focused on. And in Plasmodium berghei, it's the erythrocytic cycle, the gametocytes, and in vitro conversion of gametocytes into ookinetes. So those are the stages that I will be looking at. And way back when the full genome sequence of Plasmodium falciparum was published in 2002, this paper also had the metabolic chart that was published. And a large amount of this has been very well studied now, and we have better understanding of the complexities of metabolism in the parasite. Yeah, so as I said, I'm going to talk about aspects of the TCA cycle, and the parasite during the intraerythrocytic cycle is a microaerophile, so it needs reduced levels of oxygen and produces bulk of the ATP through glycolysis, and its dependence on TCA cycle is reduced. Other features that maybe I'll touch upon is that there is no amino acid biosynthesis. Certain interconversion pathways are there, but total biosynthetic pathways are absent, and there is no de novo purine biosynthesis in the parasite. Yeah, so since I'm going to talk about the TCA cycle to the students over here, this was discovered, metabolic chart published on the TCA cycle by Hans Krebs was way back in 1937 in a journal known as Enzymology, a journal which is no longer there now. And this classic paper shaped much of modern biochemistry. So the pathway itself was not a cycle, and well before Krebs could propose the cycle itself, you had by Martius and Knoop, all the steps starting from citrate to oxaloacetate was put down, and it was put down as a linear step of reactions. And the cycle was proposed by Krebs because he found that these intermediates, when you feed it into a tissue, were not completely utilized. They were not burnt up. So they continued remaining. And so he said it has to be a cyclic process. He had discovered the urea cycle before that. And this has to be a cyclic process, though the cycle from the step from oxaloacetic acid to citric acid was not linked up. So, when you look at the cycle, you would think of, what Shakespeare said. It says, "Thou hast spoken right, it is true, the wheel has come a full circle." This full circle wheel, or the cycle that we know today, is the foundation of many biochemical understandings that we have today. So, I'm going to talk of two things. The cycle is not in isolation. You have metabolites that feed into the cycle and metabolites that come out of the cycle. The inputs in the cycle is anaplerosis and output is cataplerosis. I will be touching upon both anaplerosis and cataplerosis. And in eukaryotic systems, these happen in the mitochondria, so you need transporters, which will take things in and out of the mitochondria. And the word anaplerosis itself was proposed by Hans Kornberg, who was there in UK. And in the obituary to Hans Kornberg, it says, where did the word anaplerosis originate from? It originated from Greek. With a love of the classical derived from his grammar school education, he called these anaplerotic reactions from the Greek. It means to fill up, and it's a topic which is widely prevalent in biochemistry today. So we were looking at the purine salvage pathway, and there are two cycles over here, and we were looking at this cycle, which is known as the purine nucleotide cycle. In the synthesis of AMP, from IMP, the cell produces, for every molecule of AMP that is synthesized, a molecule of fumarate or fumaric acid, which is a TCA cycle intermediate. And the parasite also has this purine nucleotide cycle, as shown over here. That's the cycle in which from IMP through succinyl AMP is formed, and for every molecule of AMP that is generated, fumaric acid is produced. And this cycle, if it keeps going, the flux of fumaric acid that is produced increases dramatically, and this is an important step in exercising muscles, because fumaric acid will now feed into the TCA cycle and increase ATP production. So that's what I said. You have the purine nucleotide cycle, and that feeds into the TCA cycle and can increase flux of ATP production. As I told you early on, ATP is largely generated through glycolysis during the intraerythrocytic stages. So during the intraerythrocytic stages, when you have active cell division and you have to produce AMP, what is the fate of fumaric acid? Is there anaplerosis, or is this just secreted out into the medium? We started this way back in 2008, and the simple experiments that we did were treating the Plasmodium falciparum parasites in culture with C14 labeled fumaric acid. These two carbons were labeled, and we found that the label gets incorporated into proteins, so proteins become labeled, and also into nucleotides in the cell, both RNA and DNA. And it is the pyrimidines which get labeled and not the purine nucleotides that get labeled. This was a preliminary observation. At that time, way back in 2007, we didn't have a mass spec. So we followed everything by NMR. We had ample time on the machine and it was allowed. So we fed it with not C14 labeled fumaric acid, but C13 labeled fumaric acid, because that's NMR sensitive and you can pick it up. So here, these two carbons, which are highlighted with a dot, are C13 labeled. It's 99.9% C13 labeled. And I could see from fumarate a conversion to malate, from malate to oxaloacetate, to aspartic acid, to pyruvic acid and lactate. These are the metabolites which we could pick up and assign from NMR. So that tells me this pathway, and I can annotate the enzymes. This is fumarate hydratase. This is malate quinone oxidoreductase, an enzyme that we humans don't have. We have malate dehydrogenase. Aspartate aminotransferase would convert here, and so on at each of these steps. We also established by endogenously tagging the copy of fumarate hydratase with GFP that this localizes to the mitochondrion. Malate quinone oxidoreductase subsequently was shown to localize to the mitochondrion. This we showed to localize to the cytoplasm, and these in the cytoplasm again. So we need transporters for carrying out many of these processes. So at this time, to take you back, what was known about the TCA cycle? When we had completed this work, just before that, there was a paper published in Nature saying that the tricarboxylic cycle in Plasmodium is a branch cycle with one arm going in the reverse direction in a reductive manner and the other arm going in an oxidative manner as it is there in the TCA cycle. What we found was this portion that we were studying was actually oxidative. This step and further into the cytosol were oxidative, didn't have any evidence further. But this paper got retracted in 2013, and just after that there was a paper published which said that Plasmodium has a conventional TCA cycle which goes in the oxidative manner like the way you read in textbooks, and you have all these steps which can be tracked. So thereafter the paper published in 2015 by Akhil Vaidya and co-workers showed that you can delete most of these, knock out most of these genes except for fumarate hydratase, which is the step which I showed you, which converts fumarate to malate, and malate quinone oxidoreductase are both refractory. You can't knock them out. So, so going back to my question of fumarate anaplerosis, that is from the purine nucleotide cycle, what is generated? You have fumarate generated in two steps, one within the TCA cycle and one outside in the cytoplasm through the purine nucleotide cycle. Can we ask the question whether this anaplerosis, is it essential or not? So the approach that we took was, at that time we had also an inhibitor that we had studied called hadacidin that is shown over here. This molecule, which will now impair the production of fumaric acid by inhibiting the step of conversion of IMP into SAMP. So the way we thought of doing this was, we did multiple things simultaneously. One of the things that we did was, can we rewire this purine nucleotide cycle itself and synthesize AMP either using adenine phosphoribosyl transferase, take the enzyme from yeast and do. We borrowed it from Akhil Vaidya's earlier approach of DHODH where he took the yeast enzyme. And also, can we convert it from adenosine to AMP? There are two steps, both are salvage steps which are present in many free-living organisms. Use the yeast counterpart and ask the question, now if I put this enzyme, can I knock out the adenylosuccinate lyase which produces fumaric acid? So anaplerosis of fumarate would be reduced or completely impaired. However, we found that both in P. berghii and in P. falciparum, despite introducing either APRT or adenosine kinase, we found that we could not knock out adenylosuccinate lyase which produces fumaric acid. And when we put this inhibitor hadacidin, after putting in yeast adenosine kinase or yeast adenosine phosphoribosyl transferase, the parasites continue to die. Could be due to two reasons. One, this rewiring upsets many other metabolic systems in the cell and it's not working, or fumarate is essential, or I'm not making sufficient AMP. At the same time, we were looking at, so the first paper which was said was fumarate hydratase is essential, MQO is essential, the next enzyme, malate quinone oxidoreductase, both are essential. But around the same time, little after that, we managed to publish a paper where we said that we can knock out fumarate hydratase in Plasmodium falciparum, but it was mouse strain specific. We could not do it with we could not get surviving parasites when we took BL6 mice. But however, in BALC mice, however, in C57 BL6 mice, we could get surviving parasites. This was also published by a Japanese group, again using C57 BL6 mice and not BALC mice. And they said it exhibits a slow growth phenotype. Subsequently, a few years later, there was a paper published where they showed that they used NF54 strain of P. falciparum, and here the strain used was D10 strain of P. falciparum, where they could not knock out these two genes, whereas using this, they could knock out, but this attempt was a very difficult attempt. And the conclusion in the paper is that there is existence of cell-to-cell metabolic variability with a very small subset capable of tolerating the FH deletion. So I'm going to tell you a small story about what is the consequence of deleting the pathway that I had shown, that is one starting with fumarate hydratase, which is in the TCA cycle, malate quinone oxidoreductase, the next step enzyme which catalyzes the next step in the TCA cycle, and malate dehydrogenase. I'll tell you why we have done that, which is cytosolic. And for looking at anaplerosis of fumarate, I would also need to look at transporters, and looking at the literature, primarily yeast carrier proteins and their functions, which have been well studied. We picked two transporters. One is called DTC, dicarboxylate tricarboxylate carrier, and the other OGC, or oxoglutarate carrier. The stages that we studied were the asexual stages, the gametocytes, and in vitro ookinete conversion to see the formation of ookinetes. We looked at two things. One is the phenotype of the knockouts and the metabolic consequences. Yeah, I'll go quickly through these. The only point where we saw that the phenotype was completely affected was in ookinete conversion. While the wild type formed ookinetes very well, the knockout parasite, which did not have fumarate hydratase, did not form ookinetes at all. It was completely abolished. Whereas the other stages were not in any way affected. The asexual, the gametocytes did not show any significant variation. We did this for the next enzyme, which is malate quinone oxidoreductase. We also knocked out malate dehydrogenase and made a double knockout of malate dehydrogenase and malate quinone oxidoreductase. So that's the step which I showed you earlier, where we knock out fumarate hydratase and I prevent the conversion of fumarate to malate within the TCA cycle. Malate to oxaloacetate is done by the enzyme malate quinone oxidoreductase. And if you look at metabolism itself, you can see that there is a possibility of a cycle that can exist, which is a malate oxaloacetate cycle, by which I would modulate the pools of NAD and ubiquinol. So we made the double knockout. So we made a single knockout of MQO, single knockout of MDH, and a double knockout of MDH and MQO, both in P. Berghii. And then the knockout of the transporters, one is DTC and other is OGC, and a double knockout, which is DTC OGC knocked out. To summarize again, these are all linked up with the same little metabolic pathway that you have. We found that again here, the ookinete conversion is completely impaired in just a single MQO knockout. MQO MDH knockout also completely impaired. DTC significantly impaired, but a single knockout of just the MDH, malate dehydrogenase here, which will affect the levels of NAD in the cell, that's not greatly impaired. It is possible that in the parasite, when you have lactate dehydrogenase, it is possible that this is sufficient to replenish the NAD pools that are there, that is required by the cell itself. Yeah, this is on the transporter, double knockout of OGC and DTC, completely impaired ookinete formation. So the net take-home message is that the asexual stages and gametocytes, the numbers were the same, the growth rate of the asexual stages were the same, but we have observed that with the TCA cycle enzymes, fumarate hydratase, malate quinone oxidoreductase, a double knockout of this and a double knockout of the transporters completely abolished ookinete conversion. How do I understand this? I can understand this by using two techniques. One is, both are using mass spectrometry. One is you quantifying the relative levels of metabolites between wild type and knockout, and other is an isotope tracing where I use C13 glucose and C13N15 glutamine to track the flux of metabolism. So these are the two methods that we use, and this is the data that we get. And this data, I'll show you, is largely consistent across these few knockouts that we have studied. In all of them, we find, I don't know how clear the colors are, fumarate levels go up. For the FH knockout, it is understandable because this cytosolic fumarate and this fumarate are not going to be metabolized. There's only one enzyme which can metabolize, that's fumarate hydratase. They're not going, so this level accumulates in the cell. But simultaneously, we find that succinated glutathione and succinated cysteine levels go up dramatically. These are the two levels. If you look at it from the context of glutathione itself, glutathione which is succinated, that is modified, you can see in the wild type there's hardly any modification. In the knockout, it goes up dramatically. Unmodified glutathione, that's the reducing equivalent in the cell, is present at high levels in the wild type, and it has dropped dramatically as a consequence of the modification that occurs. And similar things happens here when I have knocked out malate quinone oxidoreductase and MDH simultaneously, both of them, I can see that apart from accumulating fumarate and malate, which is expected, we see accumulation of succinated glutathione and succinated cysteine. Both of them have got a thiol group in it, glutathione and cysteine. Similarly, in these transporters, which we anticipate would transport either fumarate, oxaloacetate, or malate, we found that we have increased levels of fumarate, succinoglutathione, and succinated cysteine, in all cases. What is this molecule, succinated cysteine and succinated glutathione? This is the TCA cycle. I have put the structures of the tricarboxylic acid, these two, and all the other dicarboxylic acids, which are intermediates in the TCA cycle pathway. If you look at them carefully, you'll find that there's only one molecule here, intermediate, which has got a double bond, and that's fumaric acid. Others all have single bonds, CC single bonds. When I have a CC double bond like this, it's an electrophile, and it can react with thiols, which are electron rich. And one of the molecule that you have in abundance in the cell are thiols in cysteine, thiols in proteins, and thiols in glutathione. This is the modification that happens. This is succinocysteine, where there is this SC bond that is formed with cysteine. And in glutathione also, you have the second residue is a cysteine residue, and you convert it into succinocysteine. Way back when we started this project, papers came out saying that fumarate is a oncometabolite because it modifies cysteines in proteins, it modifies cysteines in glutathione and free cysteine also. So we looked at the proteome of the parasite with and without knockout of fumarate hydratase, and you can see here there are certain proteins which I have shown, not all of them, where there is a very high level of succinated or modified cysteine residue, a specific residue in these proteins. We do not know the functional implications, but it goes to show fumarate accumulation modifies this. And all these studies, I must say, were done on pure gametocytes, so that we are looking at a step where gametocytes are viable, ookinete conversion is not possible in these knockout parasites. The next thing was to look at flux, and there you use C13 and N15 labeled. And what do I mean by flux? If there is a traffic moving in one direction, if I knock out a particular gene or inhibit a particular gene, does it rewire and flow in a completely different manner? And what is the flux through the existing steps itself? I will not present all the mass spec data, but I'll just tell you how we do it. I've put example I've taken is fumaric acid. Fumaric acid, if it's all C12, which is what is most abundant isotope in our system, it's 98.9% abundant, and only 1.1% in our system is C13. But I feed these cells with C13 glucose, and when I feed these cells with C13 glucose, I can have, if the mass of fumaric acid is M, at one carbon which is C13, if it is introduced, my mass increases by 1 Dalton, M plus 1. So I'll have M plus 2, M plus 3, and M plus 4. This is 1 C13 is there in the molecule, 2 C13 atoms are there in the molecule, 3 C13 atoms, and 4 C13 atoms. So all my data will have M plus 0, where there's no C13, and M plus whatever number of C13s that are there. Yeah. So this is how, I start with, if I start feeding the cells with C13, I colored it C13 atoms in two colors, red and black. Black are those that will enter now the TCA cycle. Yeah, from pyruvate, it enters the TCA cycle, and these are all the M plus 1, M plus 2, M plus 3s, which I can get depending upon how many black circles, how many white circles are filled up with a black circle. But there's also metabolism happening in the cytosol, not just in the mitochondria, and those I have colored them red. But then by anaplerosis, they can enter into the mitochondrion, and now I'm showing you what would be what are called isotopologues. They're the same chemical, only the isotope is different. The isotopologues that you can generate. So these are the kind of isotopologues I can generate. The system can generate, and using this, I follow metabolism. The way the labeling goes on is what I use to follow metabolism. I've shown you only for glucose here, but I have done studies with both glucose and glutamine and the nitrogen being labeled in glutamine, and you will get isotopologues of all these metabolites. Yeah, I'll present only one data which was very interesting. If you take the purine nucleotide, inosine monophosphate, we found that it was M+5 labeled to a very high extent. So that means 5 carbons in this nucleotide have C13 atoms in them. Where will that be? The only way I can get M+5, because de novo biosynthesis of purines is absent in the plasmodium, the only way I can get M+5 is if the sugar ring is labeled. And where does the sugar ring come from? The sugar ring comes from PRPP, phosphoribosylpyrophosphate pyrophosphate. And that's hypoxanthine getting converted into the nucleotide IMP through the addition of the ribose phosphate unit from phosphoribosylpyrophosphate pyrophosphate. Where does PRPP come from? PRPP comes from the pentose phosphate pathway in the parasite, which is fully functional in the parasite. So there is an increase in flux of pentose phosphate pathway. What is the consequence of increase in flux of pentose phosphate pathway? Is that a large amount of NADPH is generated. How will the cell benefit from NADPH? I told you glutathione is modified. Free glutathione has been completely depleted in these knockout gametocytes. One enzyme in this glutathione metabolism pathway, which is there, which will increase the levels of glutathione, is glutathione reductase, which will convert NADPH to NADP. So, as a consequence of reducing the free glutathione levels that are available in the cell, that's a redox equivalent which is very essential for the cell, the parasite has upregulated the pentose phosphate pathway, which will produce NADPH. NADPH is necessary if glutathione reductase has to function and reduce oxidized glutathione back into reduced glutathione. So this is an aspect of metabolism that we did not anticipate when we knocked out these TCA cycles, and these clearly show that how you can have many pathways which are not directly associated but indirectly associated, which now get modulated, which completely change the flow of the traffic in the cell. Yeah, this is to show you that this IMP is completely upregulated, and this is my more or less last slide summarizing what happens when I knock out these TCA cycle enzymes. I increase fumaric acid levels in the gametocyte. The increase in fumaric acid levels leads to succination of thiols, both glutathione, free cysteine, and thiols and proteins. And this stress that is put on the cell is partly offset by increase in the flux of the PPP pathway, by which more NADPH is produced, which leads to reduction of oxidized glutathione to reduced glutathione. This is similar to an earlier work that was published, in which if you knock out just glutathione reductase or gamma glutamyl cysteine synthase, one which converts oxidized to reduce, another which produces glutathione, biosynthesis of glutathione, in both of them, asexual stages are not affected, but what is affected is mosquito stages. And what we find is the same thing. Asexual stages are not affected, but the mosquito stages are affected. We see no O-kinase conversion. So this becomes a possible target for transmission blocking drugs. We have studied fumarate hydratase as an enzyme by itself in great detail, including getting the structure of a homologue from getting the crystal structure of a homologue. And it is very different from the human enzyme in the sense that it has an iron sulfur cluster which is absent in the human enzyme. So the mechanisms and the way they function are completely different. So it is a possible target for transmission blocking. Just conclude with this one statement. I talked about anaplerosis and cataplerosis. Anaplerosis of fumarate, cataplerosis of oxaloacetate. Work together to ensure appropriate balance of carbon flow into and out of the TCA cycle. The beauty of this fundamental biological mechanism is undeniable in its simplicity and ponderous in its complexity. It's undeniable in the simplicity, because when you look at it, it is very beautiful. But it's complex because what we anticipated was not, the succination followed by PPP pathway increase in flux. And it is, the consequences are indeed very complex. And these are the students who worked on the project. Generating the single knockouts, double knockouts were very time consuming, and also optimizing all the conditions for doing metabolic flux analysis. Thank you for your attention.
Thanks, Hema. You will be agreeing with me. It's a really great classical biochemistry talk, which we used to study in our BSc days of the biochemistry. So I will request the audience that we should restrict on 2 questions so that we can maintain the timing. So Pushkar, please.
Nice talk. Just a couple of very quick questions. You looked at asexual development, you looked at exflagellation and mosquito stage development. Do you look at asexual to sexual conversion?
No, we didn't do it.
The reason for that is because, let's say, if it puts any stress on the parasite, does it impact asexual sexual conversion? And it will not be totally surprising if it has consequences on epigenetic chromatin remodeling and some of the enzymes involved in the stimulus way. So that is one question. The other one was look at the impact of diet in some of the mutants you made in Plasmodium burgei.
Impact of
Diet. Yeah. And because as a result of this, you can manipulate the levels of fumarate and so on if it has an impact on parasitemia and so on.
Well, the impact of one thing you can say is that during the asexual stages, they're all intracellular.
Yeah.
And the mosquito, when they go in, they're not intracellular. So glutathione is available when they're intracellular. There's host glutathione that feeds into the pathway. And once it goes out, it has to produce its own glutathione. And that's one major difference. We haven't looked at all diet things, no.
One more last question, please.
Ma'am, so I just have 2 things. One is on the strain that you have actually used. You made a point that you can actually delete this fumarate hydrate only in the bulb C.
C57, not in bulb
C. C57, but not in the bulb C. Have you actually tried to take the deletion strain and put the strain in the bulb C to see what is happening to that?
Yeah, so very good question. So when you transfect into bulb C after you electroporate the thing and you transfect in, what happens is initial southerns will show you that early on you have the fumarate hydratase gene also being present, and it vanishes, and then the only parasites that survive. So we actually looked there at, we did limited dilution cloning, and we looked at about 20 individual clones. The only clones that survived were ones that had undergone gene duplication and had another copy of fumarate hydratase present in it. Yeah, the rest were all going and integrating in a long locus and things like that. Whereas if I take the same mixture and I do it multiple times, one half of it goes into C57, other half goes into bulb C. Bulb C die eventually, but in C57 it will survive. You must also point out that in C57 BL6, you have one exon deletion in the gene called NNT, that is nicotinamide nucleotide transferase. What it does is it maintains a balance between, by transferring the hydride ion between NADH and NADPH. The hydride ion transfer, and in C57 BL6 mice, you do have elevated glucose levels because of these genetic problems.
Okay, and then second one.
Because of sake of time, I think we will stop here, because otherwise we will be. You can discuss with at other lunch or other. So yeah, come. So Hema, please.
Thank you, ma'am, for the insightful presentation. May I now request Dr. Pavan Malhotra to kindly felicitate speaker as a token of appreciation.
Thank you.
So thank you so much, Hema. So we have next Suman Kumar Dhar. Suman has been at JNU for a long time, and after that, he is now the director of TERI in New Delhi. And Suman has worked on DNA replication in Plasmodium for many, many years, and it's a great example of understanding the basic biology and how it can also give rise to an understanding of weaknesses in the parasite that we can exploit. So, Suman, looking forward to your talk.
We have an important announcement for the participants in poster session. Be prepared with your posters in the concerned area till 12:30 as the evaluation will start from 1. Thank you.
Thank you, Swati, for that introduction, and really thank Satish as well as Dr. Rangarajan and all the organizers for this invitation. And so, as you mentioned, that I was and I am still in JNU for the last 25 years and doing research as well as teaching in infectious diseases and parasitology. And last year I have moved to this place, which is TERI School of Advanced Studies, which is engaged in teaching and research in sustainability, climate science, environment, and the importance of climate and infectious diseases has been reaffirmed in my previous talks. So another point I would like to stress upon that it is not only the research on neglected tropical disease, it's also the teaching of parasitology and neglected tropical diseases are very important. The reason being, you will see very few universities or institutes which are actually teaching parasitology. Earlier, it was a special subject in zoology, but zoology with the for life sciences, biotechnology. So unless we strengthen the teaching more rigorously at the undergraduate level, no matter what you do at the upper level, the interest, even the students, even there are 5 students, 4 students will be interested in cancer biology. So therefore, with that note, that importance of neglected tropical disease as well as parasitology was there, it will be there and it will continue. So today I will discuss about one protein which is called proliferating cell nuclear antigen, and that is the number 2. There are 2 PCNAs and I will come to that in Plasmodium. I will focus mostly on that protein. Now, this has been also shown to you because the lab focus is mostly on DNA replication and cell cycle regulation. And you have seen this earlier also. But the point I want to make that during this life cycle of the parasite, either in mosquito host or in human host, erythrocytic or exoerythrocytic, there are 4 different stages where multiple rounds of DNA replication takes place. And this is one of the fastest rate of DNA replication known in any living system, because in gametocyte stages, there are in 8 minutes, 3 rounds of DNA replication takes place. So as I said, this is known as possibly as the fastest rate of DNA replication. Now, and as you know that what happens when the parasite infects, the nucleus keep on dividing. Although it's an eukaryote, this is the basic difference, because mostly in eukaryote, the cytokinesis is followed by nuclear division. But here, nucleus keep on dividing. Nucleus keep on dividing, and also there are organelles, apicoplast, as well as mitochondria. And after a certain time, and this is happening in a very small volume of RBC, so as you can see that after a certain time, the nuclear division doesn't take place anymore, and then the cytokinesis, which is more at the Sison stage, late Sison stage, takes place. So therefore, the focus of the lab is to study the proteins which are involved in DNA replication and the mechanism of nuclear as well as the epitope DNA replication, and identification of molecules that can inhibit DNA replication,
And also, many of these proteins are involved in non-replication function, and that is what will be the today's talk. Now, something basic about DNA replication, how it happens if you replace your biochemistry in eukaryotes. What happens is, and in yeast is the mostly studied system for eukaryotic DNA replication, the 6-protein complex ORC, which binds to the ARS consensus sequence. ARS is the autonomously replicating sequence, which then loads MCM227. MCM227 is the helicase with the help of CDC6, and that leads to opening of the replication origin, and that's the replication initiation, which is taken over by replication elongation. And elongation is that the leading strand as well as the lagging strand, and that's where the pCNA, proliferating cell nuclear antigen, comes. So that's the protein which is the processivity factor for DNA polymerase. This is like a stick for a blind person who cannot walk without that, and then pCNA shows the polymerase how to go forward. So pCNA is also, it was discovered as a proliferating cell nuclear antigen in cancer also. It is used as a marker because the proliferating cells, you will see that pCNA dots or foci are observed. Now, when we started to look at how replication starts in the parasite, if you look at the ring stage, which is equivalent to the G1 stage for eukaryotes, and then trop stage, which is the S phase, and then Sijorn stage, which is the G2M phase. What we found that pCNA, initially in the ring stage, it is not there, but remarkably it comes during the replication stage and forms this beautiful foci in the parasites, which is coincided with the ORC proteins. And as the replication goes on, now this disperse, and at the end, pCNA and the ORC proteins disperse from each other. So therefore, replication takes place in beautiful discrete foci in the parasite. Now this is what over the years which we have shown, that there are ORC proteins and pCNA are there. During S phase, all these proteins come together. They form this beautiful foci from N to 2N, 4N, and eventually, as I said, there has to be a signal which initiates, and there has to be a signal which turns it off. PFPK5, a CDK-like kinase, comes, and that phosphorylates ORC1. As ORC1 is phosphorylated, it goes to the cytoplasm and gets degraded, and that actually destabilizes the replication foci, and this cycle goes on. Now, what about pCNA? As I have told you, that proliferating cell nuclear antigen is the important protein for processivity factor for the DNA polymerase. Remarkably, Plasmodium has two pCNAs, pCNA1 as well as pCNA2. Now, in plant also, there are two pCNAs. In Arabidopsis has pCNA1 and 2, and pCNA2 in Arabidopsis has been shown to be involved in translation synthesis. So, during meristem development, endodeduplication takes place, which is also a very robust DNA replication, and that time more replication means there will be chance for more error. So therefore, there has to be a process by which that error can be corrected, and there pCNA2 has been shown, as in Toxoplasma also there are two pCNA, but pCNA1 but not pCNA2 was found to be essential in Toxoplasma. Now, Plasmodium pCNAs are majorly different. 30% identity in falciparum and 34% identity in Burgay, and this amino acid sequence level. So the functional role of pCNAs when we started was not known for falciparum. So what we did, first we look at the sequence, and we found that both pCNA1 and 2, they form this trimer. So they form this trimeric ring which actually goes through the DNA along with the polymerase, and that's how the processivity of the polymerases takes place. If you go to the yeast, take a pCNA knockout and try to complement with the pCNA1 and 2, only pCNA1 but not 2 can complement, suggesting that possibly pCNA1 is the replicating pCNA, and pCNA2 is also there, but it doesn't take part that much as pCNA1 does. We also express the protein as a GFP fusion protein. As you can see, this foci which I have shown you earlier by immunofluorescence, this beautiful foci you can see, these are actually active replication marks. Now, if you compare PCNA1 and 2 expression, both proteins come up at trough stage and Saigon stage it is there. But remarkably, there is a difference by way of subcellular localization of this protein. PCNA1, while it gives this foci both by antibody as well as by GFP tagging, whereas PCNA2, as you can see, it gives a diffuse pattern of presence there both in the nucleus in the cytoplasm. So PCNA1 more nuclear, dotted PCNA2 both in the nucleus as well as in the cytoplasm, but diffuse pattern. Now, if you do DNA damage, there is a robust increase both in PCNA1 as well as PCNA2. Both go up. And this going up is actually at the protein level because the transcript level doesn't go up. So therefore, in response to DDR, DNA damage response, both the proteins go up. Now, does it have any functional significance? So what we did then, both this overexpression PCNA1 and PCNA2, we did DNA damage and then looked at the survival. And we found both of them, and if you compare with 3D7 or only GFP expressed parasites, we found both PCNA1 and PCNA2 gave survival advantage to the parasites after DNA damage. So suggesting that Plasmodium has two PCNA. PCNA1 is primarily replicating PCNA. PCNA2 is an extra PCNA which helps in DNA damage response because the protein goes up as well as it gives the survival advantage to the parasites in the presence of DNA damage. Now the question is, and I have told you that PCNA2 is present both in cytoplasm as well as nucleus. DNA damage response I can think of for PCNA2 for nuclear function. Now the question is, what is the role of cytoplasmic PCNA2? We also tried to make the knockout of PCNA2, but we could not get the parasites, suggesting that this is essential. But then it is possible sometimes what happens is that, the 3' is inaccessible. So when a 3' becomes inaccessible, sometimes it doesn't happen. But we could make the 3' replacement by a GFP, suggesting it's not that 3' is not accessible. We did that. And we also looked at the localization of protein in all the stages, both in the liver stage as well as in the mosquito stage. It is present and expressed in all the stages. And also in Barghai, we could show that the protein actually goes up after DNA damage response. So not only in falciparum, the similar pattern happens in the Barghai also. Now the question is, what PCNA2 is doing in the cytoplasm? And also thank you very much, Professor Hema Balaram. She has introduced very nicely TCA cycle as well as the pentose phosphate pathway, and it's important for the parasites. We had excellent antibody against PCNA2. Also, we had the GFP PCNA2 cell line. So we did pull down and then LCMS. And if you look at, and we did for both PCNA1 as well as PCNA2, because PCNA1 is our control, and we are trying to find out the proteins which are interacting with PCNA2. To our surprise, even after doing IP with PCNA2, majority of the peptides were a pentose phosphate pathway enzyme. And that is glucose 6-phosphate dehydrogenase 6-phosphogluconolactonase. Now we'll see this is an also abundant enzyme. This could be an artifact. No, it doesn't pull down with the PCNA1, doesn't come with GFP, but it comes specifically with PCNA2. What is this enzyme? And she has already made my life easier. So, from glucose 6-phosphate, the enzyme glucose 6-phosphate dehydrogenase, and you know the importance of that for malaria also. And from 6-phosphogluconolactone, you get 6-phosphogluconate. So there are two enzymes for the first two, which then goes to the ribulose 5-phosphate. And why pentose phosphate pathway is important for the parasites or any other? One,
it gives you 5-carbon sugar, which is required for the nucleotides. It also gives you NADPH, which is very important for reduction purposes, oxidative pathway, which has been nicely mentioned by her. Now, interestingly, then we found there is a paper in Biochemical Journal in 2011 which actually studied the pentose phosphate pathway, and this enzyme, interestingly, is a fused enzyme. So these two enzymes, first 1 and 2, which you find the separate enzymes in all other systems, it's a fused enzyme in Plasmodium. And what are these? And they call it GLU4 because the glucose 6-phosphate dehydrogenase and the next ion. So this is the structure of the enzyme, 6-PGL and G6PD. And there is also an insertion there. We don't know exactly what is this insertion, very uniquely present only in the Plasmodium enzyme. So therefore, PCNA2 is interacting with this enzyme, which is a fused enzyme of first two pathways of the pentose phosphate pathway. So obviously, that was LCMS data. We try to do pull-down experiment in vitro. Yes, in the pull-down experiment, you can see that with all the controls, it's specifically PCNA2, not PCNA1, is interacting with the full-length protein GLU4. Does it interact with the G6PD part or the PGL part? So it does interact with the PGL part. Again, we did the pull-down assay, but very specifically with PCNA2 only, not PCNA1. This is the control. And the interaction with G6PD is also very, very low, not compared to the PGL, which we have shown earlier. So therefore, PCNA2 specifically interacts with PGL and not with G6PD part. If it is interacting, does it have any role in the enzymatic activity of G6PD? For that, first, this is GLU4 and G6PD. And it is producing NADPH. So you can take an absorbance at 340 nanometer and look at the enzyme activity directly. Now, interestingly, as you can see there, that the GLU4, if you look at the G6PD activity, the GLU4 activity in the fused enzyme is much more significant than if you take glucose 6-phosphate dehydrogenase separately. So therefore, this enzyme, which is a fused enzyme, is actually giving an advantage to the parasites by giving more activity for the glucose 6-phosphate dehydrogenase, which is the rate determining enzyme for the pentose phosphate pathway and the first. If that is true, and if PGL and G6PD, they are fused, if I can add them separately, whether they enhance the activity, the answer is no. So if you look at there, the activity is not enhanced that much. So therefore, the addition of PGL in G6PD does not enhance the activity. Next thing, does PCNA2 enhance the activity of GLU4? Yes, it does. So this is what we did. We added PCNA2 in GLU4, and as you can see, there's a significant increase, almost 30% increase in the activity of the enzyme, and this is the Vmax, and Km also goes down. So PCNA2 also enhances the activity of GLU4, which justifies the interaction of the PCNA2 with GLU4. PCNA1? No. So that's the control we are using every time. We use PCNA1 and GLU4. The activity doesn't, in fact, it goes down. It doesn't enhance. The last part, PGL interaction and G6PD interaction. Both we have shown that it interacts with PGL but not with G6PD. So PCNA2 and the G6PD, there is no advantage of the G6PD activation. So what we have got so far, if I summarize, GLU4, which is a fused enzyme, has higher activity than G6PD alone. Now, addition of PCNA2, the presence of PCNA2 in the cytoplasm, it leads to interaction with GLU4, and also it enhances the activity of G6PD further. Therefore, the interaction of PCNA2 with GLUFO and enhancement of G6PD enzymatic activity justifies the presence of PCNA2 in the cytoplasm and its possible role in pentose phosphate pathway and parasite survival under oxidative stress. Now, this is only observation. What is the mechanism? What are the domains? What are the residues by which it is interacting? We are still working on it. At present, we have done some preliminary study with the protein-protein docking, and we can see that the pocket residues which are highlighted for GLUFO as well as the trimeric structure of PCNA2, what happens stepwise, again in silico data, which needs to be validation experimentally, PCNA2 binds to GLUFO N-terminus via the IDCL, and IDCL is the inter-domain link of the PCNA of N-terminus and C-terminus, and it also shows it binds to the N-terminus of GLUFO where PGL is, that experimentally has been shown. Second, it stabilizes the NADPH binding pocket, which is 345 to 351, and this binding pocket of NADPH with its substrate possibly modulates the domain separation and G6PD activity. And finally, enhanced PPP plus and stress metabolic production can lead to better activity of a very important enzyme of the parasite, which is important for the oxidative stress. And this work has been done in collaboration with Dr. Arnab Mukhopadhyay. Now I will stop here. I hope I am on time. Yes. So this work was started by Dr. Pallavi Mitra, who is now a scientist in RGCB. She initially worked on that. Then Khadija Banu, she is not here, but she is listed here. Now, the GLUFO interaction was done by Khadija Banu, who is in USA doing postdoc, and the interaction of GLUFO with PCNA2 and enzymatic activation is mostly done by Steffi Verghese. She is now completing her work. Rina is here. She has her poster on another metabolic enzyme, phosphoglycerate mutase, and its relation with DNA replication. And also Poonam is here. She also did PhD, and now she has a poster, and she has moved to ICGB for her biocare. And I am indebted and thankful to all my collaborators, as well as all the students who have been in the lab and done the work, and all the funding agencies. And thank you so much for your attention. Thank you.
Thank you so much, Suman. So you're well within time, so we have time for many questions. Maybe we'll start with Puran in the back first.
Suman, yeah, fantastic talk and very interesting.
Thank you.
So I was trying to look at the other side of this interaction, because some of these metabolic enzymes not in plasmodium have also been reported in the nucleus in other systems. I don't remember whether this interaction also has some additive effect on the activity of PCNA2. Whether the interaction of PCNA2 with phosphogluco, whether it might also regulate or modulate the activity of PCNA2 in the nucleus, and whether these enzymes are also present in the plasmodium nucleus.
So pentose phosphate pathway is in the cytoplasm, and PCNA2 is present in the nucleus also. So we believe that its DNA damage response function, that is in the nucleus, whereas it has additional function in the cytoplasm, where it interacts with, this could be one of the proteins, there could be many other proteins, and they can modulate the metabolic pathway. We haven't checked whether GLUFO, because you nicely introduced this point, even some of the TCA cycle enzymes have been shown to go to the nucleus. And we see phosphoglycerate mutase, which is an enzyme in the glycolysis pathway,
that goes to the nucleus and it also binds to the DNA. And Rina has a poster on that. If you're interested, please go through that. Thank you.
What is the association constant, dissociation constant of GLUFO and PCN?
We haven't exactly calculated.
But you're able to produce all these enzymes,
Yes,
isn't
yes.
it? Then you have done this docking. You can do it another way. You can use your mass spectrometry, whatever you use for pull down. No? You cross-link these two by buying commercial cross. And then instead of just doing haddock, you give this mass spec information to the person who's doing haddock with you, and you can do guided docking. And that will give you a really reliable structure of the complex.
Thank you.
So, nice. Quick question. Is it this interaction specific to Plasmodium? Does PCNA interact with, if you tried GLUFO from other organisms? And if it does it interact?
Very good question, but PCNA2 is not there in most of the eukaryotes.
No, just in vitro.
In Arabidopsis it is there. We haven't checked there. But it is possible that they have evolutionary conserved function. And if that happens, that would be really, really interesting.
So I'm just curious that looking at the structure of PCNA2, can it behave like a transporter?
Transporter.
Because it is the
transporters are mostly in the membrane and also organelles also because of the specific reasons. PCNA1, even if it's a trimeric ring-like structure, but is it exclusively in the nucleus? Whether PCNA2 has some role?
Yeah, PCNA2.
Yes, we don't know, but that's a very novel way of thinking, and we can try to.
Yeah, Mrinal.
So you're suggesting that, I mean, earlier you have published that PCNA1 is the main replicating
replication
PCNA.
as well as GDR.
Yeah.
PCNA2 in the nucleus, as far as the evidence we have, mostly in the.
So I was wondering whether, did you look at whether PCNA2 is also involved in replication during the male gametogenesis, like where in 8 minutes 3 rounds of replication is happening?
Very good proposition, but we haven't looked at yet. But the expression of PCNA2 goes many-fold in the gametocytes. So that shows there could be some other functions of PCNA2 in the sexual stage.
Yeah, maybe I'll talk to you later on.
Sure.
So if there aren't any more questions from the audience, I have a very quick one. So Suman, that trimeric structure of PCNA2, one could see why it's useful for DNA replication, because it has to work like a clamp almost.
Yeah, so it's a clamp it moves through the DNA along with and you will know that when the DNA damage takes place, there is a switch of polymerase. So from replicating polymerase, the repair polymerases come.
Correct.
And they also have to interact.
Correct. So my question actually was that for the cytoplasmic function, is PCNA2 in the trimeric structure or is it a monomer? Because your last few slides, they suggest that they are even able to do the enhancement without being in that structure.
Yet, but good question. Thank you. But primarily, when you are expressing and purifying, primarily it is trimer.
Trimer. So that seems to be the...
Yeah, possibly it's also
trimer.
In the cytoplasm also.
Yeah, thanks. So thank you so much, Suman. Terrific talk. Very much in time. So we had lots of questions and really great to hear from your lab.
Thank you, sir. Now I request Dr. Swati Patankar to felicitate our speaker.
Our next speaker is Professor Binal from University of Hyderabad. Again, working on very fantastic pathway, DNA repair, DNA damage repair, and DNA repair pathways. And I think they are again very important pathway for drug targeting. So Professor Binal will be highlighting some of his work on these pathways. Very good.
At the outset, I'd like to thank the organizers, especially Satish and Saman, for the invitation. And this is a fantastic conference going on. So in ancient Chinese philosophy, yin and yang represent two opposing forces, yet interconnected and interdependent, and together they maintain the harmony. So today I'm going to talk on the positive and negative regulators of Plasmodium recombination and how they shape the outcome of homologous recombination in this parasite. So before that, let's understand why do you need to regulate homologous recombination. Homologous recombination, as you all know, that is a recombination between two homologous or identical sequences. Now, if this criteria is maintained strictly, then you do not expect any recombination between the mom's chromosomes and the dad's chromosomes because they are not identical. And then evolution will be stopped. On the other hand, if the recombination machinery can allow recombination between two non-identical or very divergent sequences, then probably you will see these creatures roaming around. So the bottom line is the homologous recombination machineries, it take some kind of middle path. It allows certain degrees of heterology during homologous recombination, and then it restricts homologous recombination if the sequence divergence is more. And hence you need certain proteins which will promote recombination and certain other proteins which will restrict recombination. So in the context of Plasmodium biology, homologous recombination has many different vital roles, but I'll just give two examples. One is antigenic diversification and another is multi-drug resistance. For both of them, you need the homologous recombination, that means recombination between not so identical sequences. So in earlier we have shown that PF RAT50 on the recombinase that positively regulate antigenic diversification, and work from Catherine Maddox's lab, they have shown that the rective helicases Bloom and WRN, they negatively regulate antigenic diversification. Now in other eukaryotes, in all cells, RAT51 is a recombinase. In mitotic cells, there is a protein called SRS2 that inhibits RAT51 activity, and another protein called RAT57 that inhibits SRS2 activity, and hence RAT57 is a promoter of recombination. Similarly, in meiotic cells, DMC1 that inhibits SRS2 activity, and thus it promotes RAT51 mediated homologous recombination. So in Plasmodium, RAT51 is very well characterized, but the other proteins which are the regulators of recombination, they are not well characterized. So in order to identify the positive and negative regulators of recombination, we took RAT51 as a hub protein, and we collaborated with Arijit Ray from TCS Hyderabad, and they did some analysis called RWR, random walk and restart, and identified the whole network, and from there we chose two such protein. One is SRS2 and another is NDR1, novel DNA repair protein. And today I am going to talk on these two proteins. So we have seen that SRS2, we made the antibody against SRS2, and we have seen that SRS2 is mainly cytoplasmic protein, but upon DNA damage, it goes to the nucleus, and when we knocked out SRS2 by CRISPR-Cas9, it appears that these SRS2 parasites are more tolerant towards DNA damaging agent than the wild type. And we have developed an assay system, a PCR-based assay system, where you can create breaks in the genome and then you can follow its repair. And we saw that, as you can see, this red line, so that is the SRS2 parasites. They can repair DNA damage quicker than the wild type. So which means SRS2 has an anti-recombinase activity. So if you remove SRS2, then the DNA repair is much efficient in this parasite. Similarly, we created Bargii knockout of SRS2 and saw that there also DNA is repaired much quicker and much efficiently. I am not going into the data. Then you wanted to understand that what is happening in the meiotic cell, because there more regulation is required, right? So in other eukaryotes, what happens that if DMC1 is not there, then HR does not take place and you get non-crossover product. If DMC1 is there, then HR takes place and you get crossover product. So you wanted to understand the interplay between DMC1, SRS2 and RAT51. So we created a mutant, but before that let me just remind you that when mosquito takes up the gametocytes, within the mosquito they are the gametes are produced and then the gametes fertilize each other to form the zygote. Their meiosis takes place and then eukaryotes are produced. So as a readout of meiosis, we wanted to score the oocyst because that is easier to score in the mosquito meat cut. So we created mutants of RAT51, DMC1, SRS2, and we found that in RAT51 and DMC1 mutant, the number of oocysts are drastically low. So there is 95% reduction in RAT51 and in DMC1 is 80% reduction in the number of oocysts. But interestingly, in SRS2 parasites, SRS2 null parasites, there is not a single oocyst, completely zero oocyst. Then we told ourselves, wait a minute, this is kind of anti-intuitive. You expect that if SRS2 is the anti-recombinase, if SRS2 is not there, there will be more recombination, there will be more number of oocysts. But we found there is zero number of oocysts. So hold on to that. I will come back to that in 1 minute, why it is happening. But then in RAT51 and DMC1, we found few oocysts and we looked more closely to them, and we found that in RAT51 there is heterogeneity in the oocyst size. Majority of them are petite oocyst, very small oocyst, and then few like 5% are of the wild type size. But interestingly, you do not see the sporozoites within the oocyst. You see that, look at the thread-like structures in the wild type, those are the sporozoites within the oocyst. But in RAT51 null parasite, there is no sporogonic development. Similarly in DMC1. So then we wanted to see, okay, there is no oocyst, let's go back one step at a time. So are there gametogenesis, are there gametes are formed, are there zygotes formed, are there eukaryotes formed? So we wanted to look at them, and we found that very interestingly that in RAT51 or DMC1 parasite there is no defect in exflagellation, there is no defect in male or female gametogenesis. But in SRS2 there is a defect in male gametogenesis, and that's why they are not forming any oocyst later on. And when you look at the eukaryote, there is a defect in eukaryote formation for RAT51 DMC1, suggesting that they have a role in meiosis, and that's why from zygote you are not forming the eukaryotes. But in SRS2 there is zero eukaryote, of course, because there is no male gametogenesis, right? So we wanted to look into deeper what is actually happening. So we did a couple of process. So here the idea is If you do a cross between the wild type and RAT51 or wild type and DMC1, you expect that there should be hybrid oocyst because you are having wild type copies of them, right? So in that cross, we found indeed there are hybrid oocysts, suggesting that if one of the parent has RAT51 or DMC1, that is good enough to do the meiosis and you get the oocyst. So both DMC1 and RAT51, they are haplosufficient gene. But interestingly, when we did with SRS2, here your expectation is, well, SRS2, there is a defect in male gametogenesis, but they form the female gamete. So the female SRS2 can be fertilized with wild type male, and you expect that there should be hybrid oocyst. But we do not find any hybrid oocyst, suggesting that there is additional defect not only at the male gametogenesis, but downstream there is an additional defect from zygote to ookinete. We do see hybrid zygote, but no hybrid oocyst, which means there is an additional defect and there is a meiotic defect, and SRS2 is haploinsufficient. So one copy does not support the meiotic role. So in summary of this part, in asexual development, SRS2 acts as an anti-recombinase. So if it is not there, then recombinase is much better, DNA repair is much better. But in the sexual development, it has two roles. One is during male gametogenesis, probably because SRS2 is a helicase, right? So as Suman was telling that during male gametogenesis, within 8 minutes 3 rounds of replication, so rapid replications, that probably produces lots of aberrant structures, and in other system SRS2 is known to remove those aberrant structures. So probably that's why it is a defect in the male gametogenesis, and it also has a defect in the meiosis from zygote to ookinete, because there also RAT51 forms a lot of aberrant structures between and SRS2 stop the illegitimate recombination at that point. If SRS2 is not there, then cell does not go to complete meiosis. So the same screen, as I said, that we have also identified another DNA novel DNA repair protein we named as NDR, novel DNA repair protein. NDR is also expressed mainly in the cytoplasm. Upon DNA damage, it goes through the nucleus, and NDR is an essential gene. You cannot knock it out. So what we did in the same assay that I have described earlier, we overexpressed NDR in the parasites, and we found that there is a kinetic advantage if you overexpress NDR. So NDR is a pro-recombination protein. It helps in the recombination if you overexpress NDR. So then what is NDR? When we did a blast search, we found that NDR is the ortholog of East RAT57, and RAT57 and human SRS3, they are the orthologous protein. So it looks like the NDR could be an ortholog of RAT57 or SRS2. Then we checked whether NDR can complement East SRS2 defect. We did 3 different kind of assays: MMS spotting assay, MMS return to growth assay, and UV sensitivity assay, and we could see that NDR can partially complement SRS2 defect. So what is RAT57? That is a paralog of RAT51 in East, and in human there are 5 paralogs. One of them is SRCC3, and that is the ortholog of RAT57. So it looks like that NDR that you are working with is a paralog of Plasmodium RAT51. So then we reason that if indeed RAT51 NDR is a paralog of RAT51, then it must contain those RAT51 like domain. Indeed, they have Walker A, Walker B motif, and ATP can bind very nicely to that. Earlier, we have identified one small chemical that can compete with ATP for a RAT51 binding, and it inhibits RAT51 activity. But interestingly, from our bioinformatics analysis, it looks like that B02 cannot compete with ATP for NDR binding. So the affinities are such that the B02 cannot bind to the NDR. So we thought that if these two are paralogous protein, then if we can overexpress NDR, then it may rescue BJO2 sensitivity. And we did that experiment, and we found that indeed, you follow this red line. So the black line is wild type parasites with BJO2, there is no DNA repair, and the red line is with BJO2 where NDR is overexpressed and it can rescue the BJO2 sensitivity. So that means that it is a ortholog of RAD57. Yeah. Now, what does RAD57 do? So when you have a break, then the exonuclease cleaves the DNA and it makes a single stranded DNA where RAD51 should be loaded, and RAD57 helps in that loading. Now SRS2 is trying to dislodge RAD51 from there, that is its anti-recombinase activity, and if RAD57 is there, then it finally wins the race and then it forms the RAD51 nuclear protein filament. So if this is true, then all this protein RAD51, NDR, if this is true for Plasmodium also, then RAD51, NDR and SRS2 must interact with each other, and we found that through H2 habit they physically interact with each other. So this is my summary slide. So today I have talked about the positive and negative regulators of RAD51, the yin and yang of Plasmodium recombination. I'd like to thank or acknowledge the group. So today I have talked about the work by Tanishka and Abhilasha, who are in the two extreme ends, and also I'd like to thank my collaborators and the funding agencies. So I leave you with this summary slide, and I'll appreciate questions.
Thanks, Mrinal. Great talk. And you know how many RAD family members are there in Plasmodium genome?
Plasmodium has only one RAD51.
No, not RAD. RAD family. There are so many RADs are there.
Yeah, there are many. Like RAD50 is there, RAD10 is there, but interestingly RAD52 is not there.
Okay.
Which is there in all other eukaryotes. Plasmodium does not have RAD52.
Okay. We'll take the questions from here.
Thank you, Professor Binwal, for your presentation. So going back to your concept you use, yin and yang, for doing this presentation, I want to know the yin of DDR2 and the yang of SRS2. Because you say the yin is DDSRS2, right?
Yes, SRS2 is in,
and the
yang negative regulator.
And the yang is DR2. So you presented the concept of yin and yang. So in the yin you have some yang, in the yang you have some yin. So I want to know if for this yin we have some yang, and from this yang we have some yin.
Okay, so basically that depends upon the context, right? The context of the DNA sequences. So depending upon that, so it is not completely known exactly how the decision is made, but depending upon that, the eukaryote they allows or disallows the recombination to happen there, right? So in strict sense, as you are telling that yin also has a little bit component of yang, so with SRS2 we can see that it has both components, but in NDR, so it is only, I mean, it has only positive, it does not have any negative. So far we did not identify any negative regulation by NDR1. Right.
Yeah.
So is there any RAD51 independent repair pathway?
I could not hear you.
So is there any RAD51 independent repair pathway in Plasmodium, or is there any preference of RAD51 to
RAD51 dependent pathway is the major pathway. There is no non-homologous end joining in Plasmodium. However, there is a alt EJ, alt end joining pathway, but I mean, people have shown that from Kirkdeich, Laura, Kirkman, their lab, they have shown that it has alt EJ, but none of the proteins which are involved in that have been identified yet.
So is there any preference for this RAD51 based repair pathway in the core genome or subtelomeric region?
Can you loudly, please?
No. So RAD51 dependent repair pathway has any preference towards the genome, for example, telomeric region or core region of the chromosome?
RAT51 works everywhere because that is the main pathway, right? But then again from Laura Karpman's lab, they have shown that in a particular context, RAT51 dependent pathway has major effect in the core genome over the telomere, subtelomeric region. But that is a artificial assay system. So we really do not know in the real plasmodium biology whether it has any preference. But yes.
Last question from Puran.
No, can you hear me? Yeah.
Yes.
So fantastic talk. So just a query I have. How did you determine the recombination frequency of your knockout parasites?
Recombination frequency?
Yeah. So you were saying the knockout parasites have increased recombination rates, right? Something like that, or the
like you are talking about the oocyst formation,
right? Yeah.
So we count the number of oocyst per mosquito after you infect them.
Okay. So I was wondering when you knock out these, whether the other side could be whether those parasites also have increased rates of mutations if you do genome sequencing. Is that possible?
Those parasites also have mutation. Yeah, you expect that SRS2 should have increased rate of mutation. Yeah, but we didn't look into that.
Okay, I know there are many other questions, but I think you can ask during lunch time or tea time, that time. So we end here. Thank you, Mrinal. Very nice talk.
Thank you, sir, for this insightful presentation. May I now request our chairperson to felicitate the speaker?
So our next speaker and the last for this session is Dr. Kalyaneshwar Mandal from TIFR Hyderabad, and he has been working on D-amino acids and proteins that can be used as therapeutics for the diseases that we've been talking about. So we'll start off with this talk right away. Kalyan, it's 16 minutes and 4 minutes for discussion. So this is 16 minutes and 4 minutes.
Okay. Okay. Thank you. Thank you, Professor. Am I audible? Yeah. Thank you, Professor Swati, for introduction, and I would also like to thank Dr. Satish Mishra for the invitation and putting together such a wonderful conference. All right. So see, as my title says here, that what we do, we actually engineer biomolecules, and here by biomolecules, what I mean, they are basically. Can you? Yeah. So what I mean by biomolecule here, they are either peptides or protein. So and what kind of engineering that we can do, we can actually do in atomic level precision. If you look at here, there are two peptides here, and let's say you have lots of nitrogens, right? And if I want to selectively just modify these two of those amino acids, we can actually do that. Let's say we want to make N15 level. What is happening? It has to be through this direction or something. Can you check? Where is that? Okay, that's not my fault. All right, I'll go back a little bit. Yeah, so let's say these two particular nitrogen, if I want to do N15 level, we can actually precisely do that, right, with our techniques. And one more thing, just assume there is a chiral center, additional chiral center in this molecule, right? And if I want to just switch that, if I just want to flip that, we can do that too, just by sort of chemical engineering, right? And I think these two peptides, Dr. Rangarajan will probably like it, because these two peptides actually represent CSIR and CDRI. And you know what? In this CDRI, luckily there is a cysteine at the N-terminus, which means it allows us to ligate together, and then we can form a full-length polypeptide. If you have multiple of these kind of segments, you can just make a full-length polypeptide, and then if you are lucky, it will be folded nicely and then give that proper disulfide bond, and then that leads to the folded protein, right? And just by this technique, we can actually make many different sizes proteins, and these are all synthesized in our lab in TIF Hyderabad. We have reached up to 208 amino acids, fully functional folded protein, right? And as I was saying, we are doing basically molecular surgery. Let's say I don't like that loop part, I'll just cut it out and then put something else which is probably better. And we can also basically connect that C-terminus and N-terminus together, make it a big cyclic molecule if I need it, right? But whatever engineering I just talked about, they are all happening in amino acids level, and then we are incorporating that and then making the full-length protein. Can we do engineering in protein level itself? So we can actually site-specifically conjugate multiple drugs or fluorophore as you want, right? Now, these are all about the chemical engineering of the peptides of protein. Can we also do genetic engineering? Yes, we do that too. Like you have a, let's say, bacteriophage virus, you can genetically modify them, and then you can basically display large number of library of protein molecules, right? So these are the kind of engineering that we do for what? To prevent malaria. Now, for this audience, I don't think I need to tell any facts about malaria. Just to remind you, as I speak, every child, I mean, like single child, one child always dying in alternate minutes. And what I thought, I will just put two sentences from this beautiful review written in 2013 by Miller. So what he's saying, that each sick child represents our failure to prevent malaria, and each death represents our failure to treat soon enough, right? So despite having the wonder drug artemisinin derivatives, we must always have new drugs in reserves. This is absolutely true, because from 2001 onwards, this is the real-time map of artemisinin resistance that is happening. And we are, India, there is nothing yet, but we are inch closer. That yellow mark is just in the northeast India. Now, so this again represents the same, and today in the early morning, Dr. Rangarajan was saying the resistance is actually inevitable. So you must have something else in arsenal ready, right? So what we are trying to do, we are trying to hit the Achilles heel of this parasite, which is the red blood cell invasion pathway. So what parasite does, it uses two protein, apical membrane antigen 1 and rhodopsin neck protein 2. These are the two proteins which interacts each other very tightly, and that helps the parasite to enter into the red blood cell. And the important fact here is, first of all, both of them are basically parasite protein, and this RON2, this extracellular domain that binds with AM1, that is extremely conserved within the strain. So what we are trying to do, we have multiple different ways utilizing the RON2 polypeptide and RON2 extracellular domain. We are trying to inhibit these interactions. I'll briefly tell some of these in the later few slides. So the crystal structure of this RON2 ectodomain with bound to that AMA1, that is already known. So if you look at that, this is the ectodomain which is 39 residue peptide, right? So if you just make this peptide and put it in the solution, it will actually have no structure. It's a completely unstructured region full of. There is a small beta sheet loop, otherwise it is fully unstructured, right? So what we thought, can we engineer this peptide such a way that it can mimic the bound form? And what we realized, that there is an alanine residue here, so we just mutated that to cysteine and then put additional disulfide, and we also connected the C-terminus and N-terminus just to give little bit of rigidity, just to mimic that bound conformation, right? And we are not sure how many glycines are required to have effective binding, so we screen basically 2 glycine, 3 glycine, 4 glycine, like that. So it will eventually going to look something like that, which will mimic the bound form, right? And if you look at that, when we screen that, we figured that 4 glycine is the right one. I mean, it is the optimum one. And if you now compare that IC50 value, that native ectodomain had like 240 nanomolar, whereas we got 3.5-fold better potency for this engineered peptide. Now, then we tried to understand like why particularly 4-glycine was optimum. So the structure, when you predicted structure basically, when you looked at, we see that there is one aspartic acid which is at the N terminus, which is making a beautiful interactions with the AMA1 receptor, where with an arginine and one with tyrosine, and then these interactions are becoming poorer and poorer when it is either large or short in length, right? So what I just told you, that we just we took this ectodomain disordered peptide but made it a little bit ordered to make it 3.5-fold better potency of the for the binding, right? Now, so the IC50 that we got nearly 60 nanomolar. So we are not very happy with that. We tried to make it even better. So then one of my postdoc in the lab, he just came up with a beautiful idea. So there was a paper which was reported long back that they found from phase display a small peptide that actually interact and bind with AMA1. So we superpose those two structures. So there it turns out that there are two peptides which uses the same groove for binding to AMA1. When you superpose that with the native RON2 peptide, they perfectly superpose actually. If you look at that, and then we analyze the structure very carefully, and what we realize that the tail part of this peptide, phase display derived peptide, which interacts much more tightly per residue energy contribution if we calculate, and the head part of the ectodomain of RON2, the native one, that binds much better, right? So what we thought, we again here we did some sort of molecular surgery. What we thought, that we'll basically take that head part of the native peptide and then tail part will conjugate with that, right? So it will remind you like a figure like centaur, where you have like it's a warrior like figure where you have human intelligence but with the agility of and the power of horse, right? And we just did that, and what you see here, that hybrid peptide, where you have the parent peptide has 4 micromolar or 1.8 micromolar IC50, but just by fusing that, what you got is 10 to 30 nanomolar IC50, which is extremely good actually. So we are very happy, excited. But there is one integrated PhD student in my lab. He was not really happy even with this good data. So what he did, he just took that peptide and put it in 20% volume by volume human plasma, and look at this curve here, and it turns out that within 15 minutes that peptide completely gets decomposed. So it gets proteolytically digested, right? So then he also came up with another beautiful idea. So the hypothesis was, look at that, this is the shorter version of that peptide, which is like 25 residue, and there is no structure again for the tail part, right? So we thought, so our hypothesis was, since this is a disorder region, so we thought that we can actually incorporate that. We can just completely inverse the chirality here. So we incorporate D amino acids here. We did not change the amino acid chirality of the proline because that is in the turn region. We didn't want to disturb that, but all other amino acids we replaced with the D amino acids, right? Thinking that it will accommodate, it will have that induced structure, so it won't hamper much because most of the interactions were basically backbone mediated. And just to make the L part, this is the L part, just to make it more rigid, what we did, we actually incorporated one more disulfides. Now look at the result. So the axis was, x-axis was before in minutes, hours by time. Here it is in days. So up to 3 days, we don't see even 50% decomposition. So significantly improved, right? And if you look at all this proteolytic digestion data, they are very, very much stable. So we had one more version, which is the ultimate one, where we also incorporated one isopeptide bond. So just to make it even more rigid, because this is the part which is L part, proteolytically still vulnerable. The other part is supposed to be extremely stable. And that is exactly what we observed here. If you look at here, even up to 5 days, it didn't even reach the 50% degradation. And so it is incredibly resistant to proteolysis. And so now the question is, did we compromise with the IC50? Not really. It is still 52 nanomolar. Partly we did, but 52 nanomolar is significantly good, the result basically, right? So we published that, we patented that, but then we don't know what to do. Basically, this is maximum I can do. Now the pharma industry has to come up, but as in morning we heard that industries are not much interested in malaria, so I don't know what to do. Anyway, but that was fine. But still, you look at that, there are L amino acids, partly D amino acids, right? What I wanted to have the whole thing in D amino acids. It's a completely D amino acid made protein. It's a small protein. So I don't want large protein like antibody, because then you are going to have tissue penetration issues. I also don't want a small peptide, because then you are lacking that surface area. You will not get enough surface area. So what we are looking at here, we are looking at a small size protein which will have enough surface area to interact. At the same time, that will not have any problem with the tissue penetration. And I am also talking about that the whole protein is supposed to be in D protein, right? So what does it mean? So just look at this picture here. So if you look at the two legs, there is one leg which is actually slanted. Because of that, this object is now the object is basically kind of chiral. Chiral meaning there is no the symmetry here, right? So the reflection that we are looking at here, that is now no longer superimposable. So that is exactly what we are talking about. So natural proteins are all inherently chiral, and they are all made up of L amino acids, right? Now if you can chemically make a protein which is made up of all D amino acids, the inverted chirality, what you are going to get, you are going to get a protein which is perfect mirror image of that fold wise. It is perfectly mirror image of the L protein, right? And as it is made up of D amino acids, all D amino acids, it is going to be 100% resistant to proteolysis and also non-immunogenic, right? Now the question here is, how can we come up with such a D protein from completely made up of all D amino acids? The technique that we are using, that is called mirror image phase display. So I'll just to give you a analogy with a lock and key. So let's assume this is your target protein. Now what we need to do, we first chemically synthesize the whole target protein in D amino acids form, and then screen that against a library of L protein library, which is displayed on the bacteriophage virus surface coat protein, to identify one small L protein binder that would go and bind to this D target with very high affinity. So once we have that, then just by putting the laws of stereochemistry, we can take the sequence information of this L protein binder and then chemically synthesize the D version of that L protein binder, which means now we got a D protein binder which should bind to the mirror image of the D target, which is the natural target, with equal affinity and specificity. So that is exactly what we are doing. And so basically what you got, let's say one of the partner, that is the RON2 we are talking about here, and then you got a D protein binder. If I inject that now, that will go and inhibit that interaction between the RON2 and AMA1, right? And so we basically tried with both of them, but for time constant, I will just talk about only the RON2 part. So here what we did, we actually conjugated biotin independently on the N terminus and the C terminus, and then screened them against that. You just take a scaffold protein. You can take any protein. We have taken this particular one because it has alpha helical nature, and then randomize the whole surface. We biased our library to tyrosine and serine because it is statistically observed that protein-protein interfaces are basically dominated by tyrosine and serine. And look at this data, phase ELISA data, that we got two binder which bound to our D target, but it did not bind to the L target, as well as any other random protein like streptavidin. So this shows the specificity that we observed from this phase screening. And now we took the sequence information and then chemically synthesized the mirror image version of it. This is the upside down CD. That data says that they are all D amino acid made. And again, when I said that these D proteins are proteolytically stable, nobody is going to believe me unless I show that, right? So what we did, we just took a random protein like BSA, and we also have that L protein binder, and then treated with pronase. Pronase is a cocktail of multiple enzymes, right? And if you look at that, within 2 hours, both the protein got completely degraded. Whereas the D protein binder that we have, even up to 48 hours, nothing happened. And I can guarantee you, if you leave it for 5-10 days, nothing will happen, right? So then we finally went ahead and then did that cell growth, parasite growth inhibition assay. Here we observed the indication we got that it is dose dependent manner. We are actually getting the inhibition. However, we are facing some challenge in the hydrophobicity of this protein, which is getting aggregated basically. So we are now working on putting some arginine tag, like solubility tag, and also multimerizing just to increase the avidity effect. And that's what we are working on currently. And so is it just the RON2 and AMA1 interaction, or any other interaction, any other protein that is responsible for this parasite invasion? So we figured out that there is another protein called RALF1, which is basically also indispensable. But I'm not going to talk anything about that. You can look at my student's poster where it is his thesis work basically. And then with that, I will summarize. I hope I could able to convince you that utilizing the chemical protein synthesis, mirror image phase display, and chemical peptide or protein engineering, we could able to identify some peptide or protein molecule which has that potential to interfere with the red blood cell invasion of malaria parasite. With that, I would thank all my students, because they are the one who does all the work, who do all the work, and I take all the credits. And obviously the collaborators, and many of the work that has been done, we got a lot of help from Professor Mrinal Bhattacharya, previous speaker. He's just in the Neighboring Institute. And some of the phase display part, I have my friend in University of Saskatchewan, Canada. He helps us with the phase display part, and of course the funding from Wellcome Trust India Alliance, and DAE. Thank you. Thank you very much for your attention, and I'll be happy to answer if you have any question.
We'll take some questions. We have Pushkar.
Nice talk. So, the target for this peptide is, as you showed in the last slide, is AMA1 interaction, and which is the blocking the tight junction formation, which is the critical process during invasion.
Yes.
So, all your growth in parasite inhibitory assays, are they looking at specifically a process, or just looking at the parasite growth?
So this is parasite growth we are looking at.
So I think you may get even a better insights if you do invasion assays, and which can be very well done with falciparum cultures and Bernal, and even do that for you, and see what is IC50 in terms of this thing. And then subsequently going forward, what R1 peptide does, it blocks the tight junction formation, but the rope trees keep firing. So you can rope trees, which are the organelles, which are also secretory in nature. So the rope tree bulk proteins are still being secreted inside the host, and beautiful pictures from many people. So it'll be very nice to see if you can recapitulate what R1 peptide does, or probably even better, when you block this. First, you have to demonstrate that it blocks tight junction formation, and subsequently, if it does, then do the rope trees continue to fire? Finally, my comment, I don't think you need to bother so much about cell permeability in this issue. As a matter of fact, it can be actually maybe better, because it may prevent the entering of these peptides into the host cells, because you are actually working at a process which are very extracellular in nature, and it's only when MA1 comes onto the surface, it goes and finds the tight junction formation.
Yeah, I'm not worried about cell penetration at all, because these are all happening extracellularly. There is no cell penetration.
You said somewhere that you wanted to put arginine rich to increase.
No, that is not for that. That is the solubility. Just a handling property I have been
so it
is getting precipitated.
So that's good. But my first two comments about the invasion rates.
Invasion, yeah, that is a good point. Yes.
Yeah, we'll have a couple more questions. Someone.
Yeah, very interesting. Oh, somebody else.
Yeah, anyone.
Yeah, please. Very interesting work. Yeah, but so it's, a question I'm sure you are asked all the time. So did you test this in vivo in any animal model for malaria? Because peptides, their bioavailability, their stability, the response you might have.
So the stability, everything we have shown It's not in vivo. Yes, I understand. The problem here is this is human specific, right? So I cannot directly take this peptide and put it in the mouse. So I have to do the mouse version of the RON2. So then the whole thing changes again. This is the problem with malaria research.
But you can do it in a humanized malaria model. There are available models now in the world where part of the, so in NSG mice, you can replace part of the mouse blood with human blood.
With RON2,
with human blood, and therefore you can use a P. falciparum infection model in mice.
Yes.
So you can do that in that model.
Yeah, okay. Yeah, we can do that.
Falciparum infection.
Right.
So we have 2 more questions.
I think Arun is doing probably, right?
No, we don't have it in India yet. Yeah, no.
We'll try to wrap up. As we have 2 more questions in the front, then Arun, very quickly, yeah?
Yep.
Mike, please. Okay, Arun, since you have the mic, go ahead. Yeah, you have the mic.
There is actually a suggestion.
Yeah.
You can also try to swap the RONs. Okay, Burgai may also have RON and AMA one and all.
That is
there is no need of going for a humanized mouse or
no, fair enough.
That is number one.
Yes.
The second one is there is a kind of a risk when you actually pick these proteins that are involved in megarozoid invasion and all this, because the entire process will be over by 60 seconds or so. So your drug should be really powerful enough to hold these megarozoids from actually invading the
but look at that, the parasite growth inhibition is actually showing that it is stopping the growth.
So there you actually do not have the stage, because you also made a point these are acting extra cellular and all this.
It is extracellular.
Maybe we can actually talk about it.
Yeah, I think, and actually we are over time. So Hema, if it can be super quick, sorry to cut you off.
I have one question. You showed a structure for these peptides.
Yes.
How did you determine them?
Which structure you are talking about? Just the cyclic one, right? That was AlphaFold predictor structure. We don't have the structure. The ISTA structure we don't
But
have.
they are small peptides. Can't you do them by NMR?
It is nothing to do with the peptide itself, because we want to see with the bound form, right?
No, but
So
you need to
this-
have a structure of the peptide too, isn't it? Because you're manipulating the peptide. So
That is known
there
already.
any structure, because you're putting one disulfide and then you're putting a second disulfide?
Right.
How do you know it is between these 2 specific residues? And after you make it, there is no disulfide scrambling. So these are common problems if you take any peptides with thiols.
So the way we do basically, we just proteolytically digest, suppose, and then get that connectivity by mass spec itself. So connectivity is right, that we can know by mass spec, but the structure of course will be better, definitely. But we don't have the structure of the peptide as such. What we did for the interaction studies, that we did the predicted AlphaFold predictor structure. There we see that the interval, the aspartic acid and arginine in the salt bridge I was talking about, they are from the predicted structure. They are not crystal structure. Of course, it is good to have that structure. We don't have that peptide structure, but the connectivity we can confirm from mass spec itself.
Yeah, so we're going to have to stop, but we must continue these discussions over lunch, which is coming up next. So thank you so much.
Thank you, sir. I request Dr. Swati Patankar to felicitate our speaker. Well, that marks the end of our first session. We thank both our chairpersons, and for that, I would like to invite Dr. Saman Habib to felicitate our chairpersons.
Thank you so much.
Thank you.
We extend our sincere thanks to all the speakers and delegates for a stimulating first session. The speakers of the next session are requested to kindly submit their presentations in Hall 2. And we'll take a short lunch break now, so everyone may proceed to the dining area and are requested to reassemble by 3 p.m. to resume our next session. Thank you.
कर दो तो।
and albumin or control for intra-nerve. There are various strategies which are available now that will increase the level of statin plasma, which is and then perhaps also a certain amount of nutrition first, you're looking at complicated malaria, so on and so forth. So you have to look at what you do, like penetration and the simplest one, and then look at that combination. There is a at the native peptide by itself. It will need to be massaged because it's not if you take a peptide, which is the peptide plasma state, it will go to the small, right? Under 12 months. Not that small, but yeah, so that is next. If you really want to pursue human translation for Arnab was your friend, so you'll find you a bar. Exactly. But we are meeting after 25 years. Oh. Thanks so much.
खुलेगा नहीं क्या? मैं जैकेट है वहां से भी तो बंद है लग रहा है बंद मेरा जैकेट में तो ऊपर से मैं किसी नहीं नहीं उसी में कूपन है
आ रहा है, रुको रुको, अब रुको, हेलो हेलो,
This is the file. So alternately, ma'am, you and sir would be giving the introduction. Okay.
Suman, you start.
Ma'am, this is you have to give for Pavan Malhotra sir, and we have to start. Okay. Yeah.
No. DC has to start. How is this job yet demanding? All the time. But you keep your lab in it.
Yeah.
But you're in the same city. Yes. So you commute.
Saturday.
Leaving it, right? This railway scene is not there, it seems.
You want me to start or please?
Start.
Good afternoon, everybody. So, welcome to this session on drug discovery and translational research for parasitic diseases. We are honored to welcome our chairpersons, Dr. Hemlata Balram ma'am and Professor Suman Kumar Dhar sir. Dr. Hemlata Balram is a distinguished scientist and leader from JNCAR Bangalore, renowned for her research in malaria parasite metabolism and protein structure function. Whereas Professor Suman Kumar Dhar is professor at JNU, who is also currently serving as Vice Chancellor of TERI. He is a renowned molecular biologist and leads pioneering research in malaria parasite DNA replication and cell cycle regulation. So I kindly request both the chairs, so they are already sitting on the dais, so we may begin with the proceedings. So I request Hemlata ma'am to kindly provide the introduction of our first speaker.
Okay, she said he's good afternoon. I think people are still at the poster session, but it's time to start this session. And our first speaker is Dr. Pavan Malhotra from ICGEB. He has been working on malaria and contributed extensively to the understanding of various aspects of parasite biology. His work has led us to understand a fair amount on host-parasite interactions. And he's going to give us a talk now.
Okay. This is next. Okay. Thanks, Hema and Suman, for the nice introduction. At the onset, I would like to thank the Dr. Radha as well as the Sushil and other team members who has really invited us to present here our work, what we have been doing, and I'm really enjoying this meeting with various different talks. So we have been working in various aspects of parasite biology, and today's talk will be on what parasite actually secretes during the infections. That's why we want to study that. We want to study the interplay between the parasite and the host immune system, how parasite gets established, how it's invades through the host cells, particularly the RBC, and how actually it manipulates the immune systems in a way that it can survive in the host cells. So my talk will be there. And another aspect is we are also trying to understand the surfactome of both infected erythrocyte as well as the merozoite. Why we want to study this? We want to study this because still after 40-50 years of research, vaccines have not been developed against the blood stages of the parasite. And I will tell you what is the main reason for that. Because there is a lot of complexity on the surface of parasite, unlike the viruses, where vaccines are available and against bacteria. But here the surface of merozoites is quite complex, and so we in the lab are trying to study the surface of these merozoite, that what kind of vaccine candidate which are important for the vaccine development against the blood stage of the parasite. So the first part of my talk is on the secretory proteins which are the parasite secretes at the asexual blood stages. And why do we want to study the secretory protein? Because they are important in various aspects. One is for the cell-to-cell communication, other is for maturing the uninfected RBC for their entry into RBC, and also for the immune modulation. So these are the various aspects why we want to study by studying the secretome as well as the surfactome of the parasite at the blood stage of the parasite. So first part of my talk will be on the secretome of the parasite. About 15 years back, we actually published a paper on the proteome of the secretory proteins of the Plasmodium parasite. And when we analyze this proteome, we were able to identify, actually you can see we were able to capture some of the pictures showing that there is a lot of beautiful secretions from the infected erythrocyte which is coming out. And when we analyze these proteins by proteomics, we could find many immunomodulatory proteins. In fact, in this data about 15 years back, we were able to identify 33 total proteins, but out of that 27 were never been characterized. So we thought we should characterize some of these proteins for their role, because even though we know P falciparum genome encodes about 4,500 proteins, but we hardly know the functions of about 50 to 60% of protein. There's still a lot to know about the parasite biology, and we believe that once we know about the function of most of the proteins, we might have a chance to develop vaccine at the blood stage of the parasite. So based on bioinformatic analysis done with our friend Dinesh, who will be presenting the data, we were able to identify a number of important proteins with important modules. And these are some of the proteins we studied based on this proteomics data. And these are the modules which in other organisms have been shown to play important role in immunomodulations. So I will present some of these characterization of some of these proteins. And in one of the protein we found was by chance. There was a study that was published that as soon as parasite enter into the body, NK cell gets activated. And these NK cells are able to eliminate some of the infected erythrocyte at the initial stage of the infection. How do they do? Nobody knows about them. So we thought, let's try to study how the NK cell do this elimination of the infected RBC. So there was a study which I showed in my previous slide that this is moving forward only. How to move back? Okay. Okay. So this study actually showed that there is a protein called lymphocyte functional associated protein 1, which is actually involved in recognition of the infected RBC. So we went ahead and we studied the features of this protein, and we found that this protein has an integrin binding domain, I domain. So one of my students, he expressed this protein, and then we studied the binding of this protein with the NK cells. And as you can see that this protein was able to bind the infected RBC beautifully in a dose-dependent manner. So this was a very good, exciting that we were able to identify a part of this LFA which was binding to infected erythrocytes. Then we cross-linked this protein with the infected erythrocytes and then immunoprecipitated using the antibodies to LFA1, and we were able to find a number of proteins. And one of the proteins we were excited to see was this protein called GBP, glycophorin binding protein 130. And this protein we also have identified in our secretome. So we were very excited that this protein is being secreted. So it means it is actually binding to NK cell, or it is probably activating the NK cell, and then the NK cells are getting activated. So we went further to show that. Then we expressed the small region of GBP 130. We raised antibodies to this and also showed that yes, this GBP130 is binding. It's expressed on the using the antibody. We showed that it is expressed on the surface of infected RBC, and then we studied binding of LFA1 domain, I domain, and GBP130, and showed that yes, indeed, by molecular modeling as well as by SPR analysis, these two proteins are interacting. To prove further these interactions, we studied the binding of LFA1 with both the GBP130 with two of the immune cell, THP1 cell and NK cell, and we were able to show this binding. And we could also show the specificity of this binding by siRNA. By putting the siRNA against the LFA1, we were show that binding was reduced, completely showing that the two proteins LFA1 and GBP130 are interacting together. And since GBP130 is expressed on the surface of IRBC, so this interaction is playing role in activating the NK cells. And then to show further that this is truly happening and how this is happening, we studied further and we could show that GBP130 is actually activating the NK cell by interacting with LFA1, and this activation is, these are some of the activation markers. We could show that yes, these activation markers are playing important role in activating the NK cell, the GBP130. To show that this is the phenomena which is actually participating in engulfing the IRBC or killing the IRBC by NK cell, we further did experimental studies to prove our point. Yes, indeed, the interaction between GBP130 on IRBC and LFA1 on the NK cell is mediating the killing of NK cell to IRBC, suggesting that the secretory element initially are very important for taking care of the parastemia in the host. So as soon as parasite enters, first the system innate immune response gets activated, and NK cells are part of that immune response that happens. So the secretory proteins gave us very good data that one of the secretory protein is activating NK cell, and NK cell are then killing the parasite. But parasite also has to survive, and it is also playing important role for it to survive in the host cells. Then we further studied another protein called complement control protein. Most of you must be knowing that one of the innate response of the human system is that the complement activation happens as soon as pathogen enter into the body. And parasite is making another protein which is called complement control protein. So how this control protein is playing a role, then we studied these proteins, this family of proteins in Plasmodium life cycle, and we were able to identify 6 of these proteins. And then we found that in literature that 4 of these proteins were already characterized at the liver stage of the parasite, but nothing was known at the blood stage of the parasite. Since we were studying and we identified one of these proteins, this one PFCCP1, at the blood stage, we thought this probably is also playing a role in modulating the activation of the complement system. So then we studied this protein carefully. We expressed the 2 regions of this protein. One is this CCP1 and CCPG region, and then we studied the role of these proteins in taking care of the complement activation. And you can see that we could see that LCCL domain of this protein was binding to complement factor C3b and C4b. And this binding actually was, later we found that it was actually down-regulating the complement activation. So this was very clear. The parasite itself is secreting some of the proteins which are down-regulating the immune activation so that parasite can survive. And then we studied this protein further, and we could show that if we passively induce antibody to this protein, the mice were surviving for longer time because now they were down-regulating the complement activation. So that's why the parasite was able to survive nicely in this, and mouse were dying little later, suggesting that parasite actually secretes some of the important protein to take care of the immune activation that happens as soon as parasite enters into the body. So based on this, we were able to suggest that at least this protein can be an important target for the new drug discovery, because one side the immune activation is stopped, other side you can give a combination therapy of the drugs which acts on the parasite development, and this combination therapy may prevent the resistance coming because of these two drugs acting at different levels. Then this work we published in a journal, and then we found another important protein called DACP. That also in our secretome. And this protein, we again thought that is a very important protein based on the domain organization. If you see that parasite usually in human, whenever you get infection of the malaria parasite, there is a big fluctuation of temperature. In the morning, there is a high fever. In the evening, there is a shivering. So almost 5-degree fluctuation is there. And people who have studied the transcriptome, they have shown that one of the protease, which is a serine protease, actually gets activated during this heat shock. And this was also shown in the in vitro studies that parasite actually doesn't survive at 40 degrees C, but whenever this heat shock proteins are activated, parasite is able to survive. So these proteins are importantly play important role in the survival of this thermal shock. So we actually characterized this DACP because this was the serine protease protein. It was never characterized, and we could find, yes, when we give a thermal shock in the culture, this protein is getting overexpressed. And also we could find a complex of these protein with various other superoxide mutase and other enzymes which are heat shock proteins. So we studied this protein further during the oxidative stress condition. Again, we found that this protein is overexpressed during oxidative stress. So we believe that this protein is secreted so that parasite can take care of the thermal shocks and oxidative stress that happens when the parasite enters into the body. And this we could prove that this is also an important target, again for the drug discovery, based on this result that when we put antibodies in the culture and gave a thermal shock, the parasite was not able to survive, clearly showing that this protein is playing an important role in taking care of the parasite survival during thermal as well as oxidative stress. There was one inhibitor for this kind of protease that were available. So we got this and again showed, yes, as soon as we put this inhibitor and then gave a thermal shock, the parasite doesn't survive. It means this protein is very important for parasite survival, taking care of the thermal shock at the time of infection. Along with this, we were able to actually study another proteins which is called cell1 and cell2, which found that it actually downregulates the Notch pathway, indicating that parasite is secreting many of these proteins to combat the activation of the immune system so that it can survive in the human host. And another set of protein which I am not describing, which has been shown in Leishmania, and we got that also in our secretome, that's called elongation factor 1 alpha and fructose. We have been able to characterize that they inhibit the pro-inflammatory responses, and that has been very nicely shown in Leishmania. So we were able to actually show number of proteins which parasite secretes at the time of infections and which actually protect parasite from the immune system so that parasite can survive and propagate. Now I switch gear a little bit about the surface proteome. We wanted to study the surface proteome of the parasite to understand why, if after 60 years research, vaccines have not been developed at blood stage. Now we know there are hepatic stage vaccines are ready and they will be launched in 2027, but we never understood the complexity at the blood stages. So we went ahead and we developed a protocol to purify merozoite at large scale and then strip merozoite surface to understand the structural organization of the merozoite surface and to know what kind of proteins and how proteins are organized. And we developed a team and got, in fact, the Welcome Team grant on that basis. And we studied the proteomics of this merozoite surface after stripping the proteins. And you will be amazed to see that besides the known vaccine candidate antigen, we could find large number of other proteins. In fact, there are about 300 different proteins. Unlike viruses and this one and bacteria, there are 300 proteins or 400 proteins which are on the surface of merozoites. We could identify them, and many of them, around 139 putative vaccine candidates which have been worked at different times, were identified from our proteome data. So we, with the help of our friends from IIT Delhi as well as UCLA, we have been now able to trying to understand the complexities of these protein vaccine candidate by cryo-EM approaches, and we have been able to get around 20 such crystal by 2D mapping, and now we have solved some of the crystals, and one of the crystal is a proteasome 20. You may be amazed why proteasome is being secreted out. Now we know very well that why it is being secreted out, because it prepares the uninfected RBC for the new infections. We were earlier puzzled that whether our protocol is working or not, because we got this in our cryo-EM data. We again repeated 2-3 times just because then we found out, yes, there is one study which shows that proteasomes are being secreted. In fact, beside proteasomes, we could identify many of the two of the important enzyme, aminopeptidase, leucine aminopeptidase, leucyl as well as aspartyl, and we have almost sorted out this native structure of these proteases. Though recombinant were published, but so we are trying to identify many of these protein complexes to understand why vaccines have never been developed against Plasmodium, and we believe that by studying some of these, we can approach to the vaccination approaches for the blood stage of the parasite. And another thing what we found which was not known up till now was the PTMs on the surface of merozoite. Because up till now we were making vaccine on in most of the E. coli system. There is a lots of PTM on the surface of merozoite. You can see phosphorylation happening. You can see glycosylation, which is not very well known, and you can see that. In fact, we have now completed the phosphoproteomics data, and we can show that about 200-300 peptides which are phosphorylated on the merozoite surface. So this was again a new finding based on our studies, and we could prove some of these important vaccine candidates that they are undergoing phosphorylation as well as methylation. So from our work on the surface proteome of parasite, we could identify number of different categories of protein. There are not simple one-to-one interaction that exist. We could identify protein which are immunomodulator. We could find proteases, we can find proteasome machinery besides the protein which participate in the entry of the protein into the IRBC. Similar complexity also exists on, so these are our partners in the Welcome Team Grant. Professor Asif, he will be presenting work tomorrow. Professor Manideepa, Professor Hongzu, Dinesh is also here. Sumit and from Rupesh from the AIMS, they are working, helping us to develop B cell epitope arrays and T cell epitope array to really develop effective blood stage vaccine if we can. Beside this, we are also working on severe malaria parasite. One of the questions we have asked ourselves was why some parasite, actually some human gets severe malaria and while many of them not. And the question was, it's because of host immune system. But we actually were able to establish few lines in our lab from the severe malaria patient who died. And these we wanted to study their surface proteomics of this parasite. And we were able to actually identify the membrane, and we were able to identify about 5 or 10 different proteins which were expressed about 1.5 to 2-fold more in this severe malaria parasite. Other than the mild, I can show you some of the pictures which were there. If you see this, first is the lab strain, other is the mild parasite, and other is the severe malaria parasite, which we have able to grow in the lab. And you can see that many of these proteins are overexpressed, which we have identified by quantitative proteomics, clearly indicating the parasite surface of the severe malaria parasite is greatly modified, and many of the proteins are overexpressed in these parasites. You can see this. This is mild malaria parasite, and this is 3D7, and these are the patient line from the severe malaria parasite. So they are very well expressed. We know some of their receptors also now. But then Krishan actually Cambodia met us some meeting, and he has beautiful essays to show that that they have the assay system. So he really helped us to identify that what are the elements of these overexpressed protein which are important for cytoadherence. So he developed this assay and could show that PFEMP3, GBP and ETRAM5 are important. They were never known earlier, that they are important for cytoadherence. So by proteomic approaches of this severe malaria parasite, we have been now able to identify important proteins. You can see this in next slide also. This is the cytoadherence normal parasite, and these are the parasite which are actually antibodies to these proteins which we have identified. Now the binding to the this one is almost gone, clearly indicating that these are the important elements which we can play to block the severe malaria. Earlier, the only 3 proteins were known, but now we have a set of proteins against which, if we raise antibodies, we can block the severity of the parasite disease and then block this. And so this whole work has not been possible without these grants with various agencies, and now we have this welcome team grant, and these are the collaborators at different stages of our work, and these are some of the publications we have got for last 10 years, and this is the team without which I don't think so I would have done and presented this work to you. So I take leave and take the questions.
Thank you, Pawan, for the very interesting talk. Yeah.
Pawan, yeah, great work and very interesting talk. I have a question on your last part. So when you talk of, severe infections in malaria, and of course we know that there will be human genetic factors that play a role, and there will be, of course, parasite factors that play a role. And in the adhesion question, we know that mutations in PCAM, VCAM, all ICAM1, all these, and then therefore levels of these receptors would influence, and they do influence. There are various fieldwork, and our lab also has previously looked at these mutations in Indian population. My question to you is, when you say that you see an overexpression of these surface proteins on these lines, are you talking of a subset of parasites in these infected patients that have overexpression? Or you think the entire parasite itself is modified and everywhere you have this overexpression?
So this was we actually took from patient.
Right.
We grew this in the lab, but not for many cycles, so that the surface doesn't change much. We were able to grow only for 2-3 cycles, and then study the various features. And this experiment of quantitative proteomics, we repeated around 4 to 5 times, and we were always getting the expression level change from 1.5-fold to 2-fold. But every time these sets of protein, 10 sets of protein, and there were certain proteins which are down-regulating also. That also we have data, but we couldn't work on them. But we thought first let's work on this up-regulatory protein, and they were beside the protein which have been shown, Riffin and polymorphic protein, and the data from Cambodia.
Yeah, so I was not wanting to ask a question, but then it's important to understand that whether the levels of the receptors also go up on the human endothelium.
Yeah,
I mean, so you have
both,
these proteins,
both.
of course, but
Yes.
then if the
Receptors are there.
numbers are not going up, it won't matter, right? So
No,
is there an upregulation also of the receptors in that particular individual who is infected with these viruses?
but important what I am trying to tell also was, when study the interactions, there were multiple receptor interaction we were finding,
So
and that we have not studied that completely. That I agree with you. Those has to be studied.
Pushkar?
Pawan, Pushkar Sharma here. Before you renamed me. Yeah.
Sorry.
So
Sorry.
yeah. So regarding the export, or which is I believe is different from secretion. So how are these proteins getting secreted? Some of the proteins are actually secretory proteins. Some of them are actually bona fide motor proteins, which are inside the merozoite and anchored to the glidosome. So obviously there has to be a different mechanism. But do you have some this thing? How are they actually getting secreted? And they have to be done in a timely way if they want to be utilized by the parasite for evading immune response, so on?
Yeah, I don't have an answer because I have not worked on that. But other people have worked there. They have shown that there are translocon complex, which is involved in secretion of some of these proteins. But if you ask me specific protein, I don't have answer. I have not studied that.
Translocon is actually for PTEX, and that is for Pexel motif. But here you're talking about the merozoites.
Yeah.
So that has to be very different, right?
Very different. I do agree, but I don't know the mechanism.
Here, can you give the microphone here?
I have a question from this side.
Oh, just a minute. Just a minute.
Okay.
Me or Pawan?
Yeah.
Pawan, nice talk. So you have about 400 surface protein when you did the proteome map. So if you do a sero proteome mapping, then you can get immunodominant protein, and that will help you better because
I do agree with it. So I didn't show the data. We have already have about the 139 putative. We have now done the peptide arrays with patient sera.
Okay.
And we got around very beautiful data at present. We are repeating it. And we got very good, very nice peptides which are coming. And among those peptides which are actually binding to those seras, we got about 30% or 20% which are actually recognized by 70% of the sera.
So as a function of HLA polymorphism, I would expect that you get wide variety of binding specificities. And how many of them are common?
Yeah. So we don't have a full map. It will be in next 2 months. Will be ready. Based on that, our aim is, can we prepare a chimeric protein based on that, as you said.
Okay.
That in fact, we also want to do a T cell epitope arrays based on the data.
But one thing I found missing is the NK T cell, which you have not studied, I guess.
We have not studied that. Immunology, we will study the later part of the grant. Initially, we are studying the proteomics and cryo-EM. And our aim is to find the best complex, the best or most important abundant complex of vaccine candidate which is coming out. And can we really develop a vaccine based on that? That is our aim.
Now, one last question.
I had a question, and this is regarding the secretome work. So, just a technical query: how do you separate between a secreted protein and versus something a dead parasite protein that is left back in the media?
So actually, it took us 6 months to do this. Sorry. So what happened was we grow parasite in the serum. First of all, how to prevent, how to really get those protein which are secreted was a major challenge. So last 4 hours, what we did was to keep without serum in a normal media and capture those proteins which are being secreted, because secretion doesn't stop at one time. So we actually, some of them, you are right, they're also on the surface and they're also secretory.
So thank you, Pawan.
Thank you for your talk, sir. So I now request Hemlata Balram ma'am to kindly felicitate Pawan Malhotra sir as a token of appreciation.
Oh, it's a very tough job. That was something which was, but then we proved by various ways that they are really secreted.
Thank you. So our next speaker is Mrs. Swati Patankar. She's a professor at IIT Mumbai, and she has made significant contributions to parasitology and therapeutic research. I don't think I need to read this, Swati. And today's talk basically insights on targeting nuclear import for therapeutics against multiple stages of epicomplex and parasites. Swati, please.
Thank you so much. And thanks, Suman. And of course, thanks to all the organizers, Satish Mishra, Saman Habib, Radha Rangarajan, everybody. Really happy to be here. Thank you so much. Put it here. Can you hear me? Audible? Great. So I'm going to tell you about some work that we've been doing in the lab regarding nuclear import and potentially how this could be a novel therapeutic target for apicomplex and parasites. So let's see if I can convince you of that by the end of it. Where? Here. So I don't need to introduce this to this audience. We know that both malaria and toxoplasmosis are very widespread human diseases. But I really want to make the point here, which is that they both have very complex life cycles, which have got differentiated stages. In this case, for malaria, it's the gametocytes and many others. And in the case of toxoplasmosis, there are tachyzoites and bradyzoites. There are also drugs, we know that, but we also know that there's plenty of drug resistance. So as we've heard today, there are many, there is obviously a need to continue this drug discovery pipeline. So in a very simplistic way, many, many decades ago, century ago probably, Paul Ehrlich described drugs as magic bullets. So what is the meaning of that? It means that they have to be very, very specific for the pathogen and they have to leave the human host relatively unscathed. So if there is a protein which is a drug target which is found only in the pathogen, then that drug is able to interact with the protein and inhibit its function. And there are many such examples for apicomplex and parasites, and I've put them here on this slide for you. So these processes like heme degradation, dihydropteroid synthase, these enzymes and processes are found only in these pathogens and not in the human host. In contrast, if you have a protein which is found in both the pathogen and the host, then your drug has to be this magic bullet, which means that it should attack the protein in the pathogen and not in the host. And antibiotics are a great example of this. So you need to find differences between these two. And in fact, a very unexpected example of this for treatment of apicomplex and parasites is actually pyrimethamine, which targets DHFR, which is found in the human host as well. So there are differences between the two. So I want to talk to you about nuclear cytoplasmic transport as a novel drug target. And in fact, nuclear cytoplasmic transport would fall into the second class of drugs because humans are eukaryotes, parasites are eukaryotes, so we both have nuclear transport. So we have to find differences between the nuclear transport of our apicomplex and parasites compared to our own cells. So what is this nuclear transport? I've just very quickly shown you the transport cycle. I want you to focus on importin alpha. It is a nuclear transporter. These are also called carioferins, but I'll be calling it importin alpha today. It binds to the cargo through nuclear localization signals on the cargo that are basic residues. Importin alpha also binds to importin beta. I'll tell you more about this. And this trimeric complex enters the nucleus where the cargo is delivered. And then there's recycling of these carioferins or importins back to the cytoplasm. So today we're going to focus on importin alpha. This is the focus of my talk. So in fact, dysregulation of nuclear transport does play very big roles in clinical disease. It has been shown for cancers, it's been shown in sepsis, neurodegenerative disease, and there are many clinical trials. There have been clinical trials in cancer for molecules that target nuclear export. There's also some interest in developing therapeutics for viral diseases because they actually have to send their proteins into the host nucleus. So if we block that pathway, can we target viral diseases? So can we target importin alpha as a target to develop therapeutics for malaria and toxoplasmosis? That's the question that I'll be discussing with you today. So this is a key nuclear transporter for cargo proteins that have this classical NLS or nuclear localization signal. In apicomplexans, there's a single gene for importin alpha, and it's essential, as you would expect. In humans, there are 7 genes for importin alpha, and they are expressed at different levels in different tissues. So the question that I'm going to spend about 5 to 10 minutes on, 5 minutes on in the beginning, is are there any differences between the importin alpha proteins of humans and apicomplex and parasites? And the reason I want to focus on this is because, as you all know, there have to be differences that can be exploited if you want to think about these kind of proteins as drug targets. So the importin alpha proteins from yeast and higher eukaryotes, they show this phenomenon called autoinhibition. So this is a schematic of a nuclear of the important alpha sequence. It has these armadillo repeats that bind to the nuclear localization signals. It has a Cas domain for export, and it also has something called the IBB, or important beta binding domain. So this is where this important beta transporter binds to important alpha. Now, in these organisms, Saccharomyces and higher eukaryotes like us, like humans, their important alpha proteins, they show something called auto-inhibition, which means that in the IBB domain, there are basic residue sequences which look like nuclear localization signals, and they actually bind to the ARM domain, and they block binding of the cargo. So for cargo to bind, in fact, important beta binding to the IBB domain of important alpha is essential, because this opens up the important alpha for cargo binding. And then the important beta also interacts with the nuclear pore complexes, and it's the main transporter which interacts with the NPC and carries the whole thing into the nucleus. So the IBB domain of many higher eukaryotes, important alpha, will have these NLS-like clusters which result in auto-inhibition. So when we started working on this project many years ago, we looked at the sequence of the IBB domains of Toxoplasma gondii, Plasmodium falciparum, and of course yeast, humans, and Arabidopsis. And you can see that there are these basic residues over here in the IBB domain, which is what cause autoinhibition. But you can see here that in Plasmodium falciparum, one of the key basic residue stretches, it has a serine rather than a lysine, right? Whereas the others have lysines and arginines. So we decided to study autoinhibition, and the way that we did it is a sort of standard way. You have an nuclear localization signal. It can bind to either full-length important alpha, which if it shows autoinhibition, this NLS will not be able to bind very efficiently because the IBB domain is blocking its interaction. You also check the NLS independently for its binding to something called delta IBB important alpha, which lacks this IBB domain, which will now expose the cargo binding sites. Or you mix important alpha with beta, which will also expose these cargo binding sites. So then what you do is you look for an auto-inhibition strength, which means there's a fold change. You'll get a KD over here, dissociation constant here, and a dissociation constant here. If there is strong auto-inhibition, your KD here will be very high, and this will be lower because now you get better binding. So for proteins that have strong auto-inhibition, this fold change will be high. You'll get a high number. So we did these experiments, and I'm summarizing all of them in this one table here. So we did these with wild-type Plasmodium falciparum important alpha and wild-type Toxoplasma important alpha. And this fold change number was very low. So in Plasmodium falciparum, it was just 1. And in Toxo, it was 3.5. In contrast, for humans, if you do this for some of the human important alpha isoforms, you will get numbers like 18 and 20, which shows strong autoinhibition. When we made mutants of these proteins, we saw that if you just replace this one serine with a lysine, now you get auto-inhibition strength of 13. So you can make PF important alpha strongly auto-inhibited by changing a single amino acid. However, for Toxo, this was weak auto-inhibition. When we changed this to KRR, which is similar to humans and yeast, we got about a 2-fold increase. So the bottom line of all this is that Plasmodium falciparum important alpha shows no autoinhibition, lacks autoinhibition, whereas Toxoplasma gondii important alpha shows weak autoinhibition. In contrast with human important alphas, which show strong autoinhibition. What are the biological implications of this weak autoinhibition? We are in the process of doing, knockouts and replacement studies in the, Toxoplasma gondii. So we have some preliminary data, which I won't tell you, but there seems to be a reason why the parasites have a single copy of important alpha showing weak or lacking autoinhibition. So then we started wondering, if there's a difference between these two proteins, can we target the apicomplex and important alpha with small molecules or some other strategies? And will these small molecules be able to inhibit these proteins differentially compared to the human proteins where they have a different strength of autoinhibition? Now, this could result from copy number. There's a single gene for important alpha in apicomplexin, 7 genes in humans. Binding site is exposed here, it is not exposed here, many things, right? So we asked that question. Can we use small molecules to target important alpha of apicomplexin parasites? So with David Jans and his team at Monash University, we did a semi-high throughput screen of the LOPAC and the pathogen box chemical library. So this turned out to be about 2,000 molecules. And using alpha screen, we screen for inhibitors of in vitro of Plasmodium falciparum importin alpha and nuclear localization signal binding. So we screen for small molecules that block this interaction. We did counter screens with Toxoplasma and mammalian importin alphas to find molecules that are specific for the pathogens. Then we also did anti-parasitic activity, and then we tried to see if there is a blocking of nuclear to of cytoplasm to nuclear transport. So this is a busy slide. I'm just going to tell you the key things and I'll summarize it quickly. But we found many such small molecules, and they had IC50s of the in vitro screening in the low micromolar. Some of them were targeting all sorts of different importance. Some of them were very specific for certain apicomplex and importance. And when we checked their activity in the cell culture, we found that that many of them also killed parasites at low micromolar concentrations. I want to highlight here this number because for Toxoplasma, we also did a cross screen against the host cells, which are the HFF cells. And you can get a number which is a selectivity index, which is the IC50 for host cell divided by the IC50 for the parasite. So this selectivity was not that high. It was for about 2 or 3-fold. And for this compound, Bay117082, it was about 11.7 or 12-fold. But please note here, this is pyrimethamine. And the selectivity index is much higher. It's 22. So we did find some small molecules. And, I'll just summarize them over here. We found some that worked both in vitro and in vivo. We found some that had a lower IC50 in vivo, which meant that they probably had many targets, and we found some that had similar IC50s in both. So I've just, sort of told you what might be the reason for all these different, phenotypes. Over here, it's actually cell permeability issues, possibly, and we are working with one of my colleagues, Rohit Srivastava, on trying to come up with carbon dot-based delivery systems. There are some which, some small molecules which showed much lower IC50s in vivo, so they may be having multiple targets. And we have some molecules which show similar IC50s in both. So along with Asif, we had a nice ICGB grant that we could work with him and his team. We actually made stable lines of P. falciparum with NLS fused to GFP. And then we treated these stable lines of NLS GFP with different small molecules. And you can see here that in this case, this is just 2 cells, but you have a pictorial graph of this. We did see some small molecules which gave a lower, number of GFP, signal which went inside the nucleus. So these molecules were blocking the NLS GFP from entering the nucleus. In my lab, Mansi, she did a similar work with Toxoplasma gondii. And she also saw some molecules which would reduce this nuclear localization of the GFP. I want to point out here that these are small effects. They are not very, very drastic. And I'll be happy to discuss this. There is a technical reason behind this, and I'll not tell you what it is just now, but I can tell you later. So we did find small molecules that also blocked nuclear transport in our 2 organisms. So the next question we wanted to find out was that we know that in the fast replicating stages, in the asexual blood stages, and in these tachyzoids, we are able to block nuclear transport and we're able to kill these parasites. But since nuclear transport is the crux or the underlying basis of differentiation, here in this case into gametocytes and in this case into bradyzoids. So for gametocytes, we have the AP2G transcription factors that play very, very big roles in differentiation. And they obviously have to be translocated into the nucleus to work. And in this case, for bradyzoites, this BFD1 is a transcription factor which has been found. That is a key protein that actually triggers bradyzoid differentiation. Again, you need nuclear transport for this. So we asked whether our small molecules will block differentiation of these 2 parasites because targeting nuclear transport should do that. So again with Asif, Mansi checked gametocyte formation and she did find that some of the small molecules, in this case CAPE and also oranophine, and ivermectin, they were able to reduce gametoside formation quite significantly. With bradyzoite formation, Mansi was able to show that some of these molecules, like BA11, they were actually able to reduce bradyzoite numbers, but again, not as much as would be very significant. I'll also point out that bradyzoite cysts have a very heavily glycosylated cyst wall, and getting small molecules to enter and penetrate through these cyst walls is also not very easy. So that might be one of the reasons. So, so far, I just want to show you the summary, which is that I think that we were able to show a proof of concept. So I'm not saying that we have found drugs. We definitely have not. But we have got a proof of concept that by inhibiting nuclear transporter, which is importin alpha, these small molecules do decrease nuclear transport and parasite viability. We also have proof of concept that some of them inhibit differentiation, which means that you can use the same small molecule to target different stages of the life cycle. And many of them do have a low selectivity index, while at least one of them does have a better selectivity index. So the next thing we asked was, if we know this is a proof of concept that nuclear transport can be targeted, can we try to improve the selectivity index? Because we were not very happy with that at all. And we thought, can we use a peptide-based approach rather than screening small molecule libraries? So this, even though I'm pitching it as if it was a very targeted and very well thought out question, the reality is it was not, and it turned out completely serendipitous. We had no idea we'll get this, and we got it. So I'll just point out over here. So in these experiments with Asif's lab, and when Mansi was working on her toxoplasma at IIT Bombay, we had an NLS, which was a classically well-studied NLS, which has been used in human cell lines for decades. And this is the SV40 large T antigen nuclear localization signal. It's absolutely classic. So we actually started these experiments fusing the SV40 large T antigen NLS onto GFP. And Mansi, when she spent a lot of time in Asif's lab, and she just could not get stable lines. So she tried very hard and it did not work. We then changed this strategy. So this is not the SV40 NLS. It is a different NLS which had been characterized from the protein PTEF. But when she came back to IIT, she again fused the SV40 NLS onto GFP. And again, she could not get stable lines at all, no matter what she did. So in fact, this was our cue that there's something strange about the SV40 NLS. And we actually discovered that this NLS on its own, when it's expressed by transfection on a plasmid, it causes loss of viability in Toxoplasma gondii tachyzoide. So this is 2 times GFP. And you can see that there is nuclear and cytoplasmic localization. There's no NLS, so it's passive diffusion. When Mansi put SV40 NLS fused to 2 GFPs, she used to see these pycnotic GFP signals, and often she would see nothing at all. However, when she put the SV40 NLS in the middle of the 2 GFPs, she now again saw live parasites. So that Toxoplasma experiment where she was able to screen the small molecules, this one, it's actually NLS GFP, but it's the SV40 GFP. Sorry, it's the SV40 NLS in the middle of 2 GFP. So we had to do a lot of maneuvering just to be able to get live parasites to do these experiments. So it was very clear to Mansi when she saw this that this SV40 NLS seems to be causing toxicity in the parasites when we express it by transfection. So these are all transient transfections. When Mansi looks, and these are all at 22 hours, when she looks at, 8 hours and 2 hours and 4 hours, she starts seeing these very unhappy, miserable GFP positive parasites, and they all end up looking like this, very small and pycnotic. So this asks, this begs the question, this SV40 NLS, why is it killing? So we first thought that it's because it's a very strong NLS and it binds very strongly to the importin alpha transporter. And that's why it is just gumming up the machinery of nuclear import. So Mansi did a nuclear proteome bioinformatics analysis, and she actually discovered that the SV40T antigen NLS strength, and this is predicted by online software called CNLS Mapper. This CNLS Mapper strength of NLS, there is about, I would say, 7 to 10% of the proteome is actually shows NLS is higher than the SV40 NLS in terms of the strength. So Mansi looked at these very carefully, and she was actually able to explain based on either the position of the NLS in the protein or whether it was exposed on the surface or not. She could actually explain why the majority of them would not be as strong as they appear by the mapper. So this SV40T antigen NLS seems to be a strong NLS, and when we overexpress it in toxoplasma, they die. So we then did this quantitatively, and what we've done here is, now you are seeing very small green fluorescent images. This is now a different magnification. It's much less magnification, and this is an entire Parasitophilus vacuole here. You can't see it that clearly, but this is a whole vacuole. So when we express 2F-GFP and 2X-GFP in cultures, we see lots of nice GFP-positive Parasitophilus vacuoles of tachyzoids. But when we express SV40 NLS 2X-GFP, we see 0. So this is the quantification of this, and we actually consistently are killing Toxoplasma tachyzoids if we express SV40 NLS. So again, the idea was that this NLS is preventing, is binding to the important alpha transporter and preventing the cargo which is essential for cell growth to actually do its job. So to test this, Mansi overexpressed Toxoplasma important alpha. So we have parasites now with 2 copies of Toxoplasma important alpha. And I told you that we had different strengths of autoinhibition. So we have wild type Toxoplasma important alpha as the second copy. Here we have a completely, an important alpha that lacks autoinhibition, and this one is one which shows slightly higher autoinhibition. So we then did the same experiment. This is important alpha. We are checking it with anti-HA tag. It's got a C-terminal tag. We are transfecting. So these are stable lines of important alpha. Now we are transfecting this SV42X NLS-GFP, which I showed you in the previous slide. When we do it routinely, we get no parasites. They're all dead. But now we started to see parasites which were expressing 2X, SV40 NLS-2X-GFP. These are green. And when you merge, you will see that there are no parasites which have only green. They are all dead. And any parasite that is green is also red, which means that important alpha is helping to rescue. So this is a quantification of this. I won't get into too much detail here, but the bottom line again is that when we express SV40 NLS-GFP, if we have important alpha present, we now see a lot of yellow parasites because the important alpha, we think, is rescuing this toxicity of SV40 NLS. So this tells us that this does seem to be going via nuclear transport and not some other mechanism. So I started this section about selectivity index. So what is the toxicity of SV40 NLS on human cells? And actually, this was a slightly strange experiment. We transfected the proteins into human cells. It's because the HFF cells, they are primary cells, and they're not that easy to do transfection with. We are still working on it. So we are going to do it, and we are going to do a transfection of DNA. But this is actually a transfection of SV40 NLS-GFP protein. So you put the protein in the cuvette and you mix your parasites and you transfect them, and you can actually get protein inside cells. So this was suggested to me by my colleague Samir Maji, who works a lot on phase separation and amyloid. So we know that we can get both of these proteins, GFP and SV40 NLS-GFP, inside HFF. And there's absolutely no toxicity whatsoever. The viability is 100%. We are repeating this with DNA transfections, but I should just point out that for decades and decades and decades, SV40 NLS-GFP has been used as a standard marker for the nucleus in human cell lines. So we want to repeat those experiments but do quantitation. But our preliminary experiments do really indicate that when we put this peptide or when we put this SV40 NLS in human cells, they're perfectly happy and they don't die. In contrast, when we put them into Toxoplasma, they die. And as an extrapolation, because Mansi could not make any stable lines with ASIF, with P. falciparum, we are anticipating that this NLS will also kill P. falciparum parasites. So this is my last slide. I hope I have shown you some data where I've told you that importin alpha is a feasible drug target that one could try to block for apicomplexan parasites. The reason is that it does have different properties from human importin alpha, which we can try to exploit. We found some small molecules that block P. falciparum and T. gondii viability. And we've also found this SV40 NLS, which is at this point being transfected as a DNA plasmid. But I did indeed talk to Kalyan about peptide strategies, and we might want to move ahead with peptides and see how we can deliver them. So I want to thank everybody, which is my students. And Mansi's done a lot of this work, so she's been driving this. Sujata has done a lot of the screening with David Jans. Vishakha did all the work on importin alpha of P. falciparum, and Diksha is doing structural biology of this. I've also put in red Anupama, who is here today, and she has a poster about bradyzoites, so it would be great if you could drop by her poster tomorrow. So that's it. Thank you.
Thank you, Swati. Time for questions. Yeah, Minal.
Swati, so the IBB domain is already part of the importin alpha, and that is already engaged with that ARM domain of importin alpha, right? So when the cargo comes, how does it disengage the IBB domain and then it get access to and bind to that importin alpha?
So where there is strong auto-inhibition, which is with human and yeast, not in apicomplexans, the KD, so the dissociation constants of the cargoes, they are also in some of the cases they are higher. So they knock it off. So they actually sort of knock off that interaction. So the IBB domain has a very strong binding to the importin beta. So your question is that how does the cargo bind if
right.
Yeah, so it's the importin beta is also binding with its own KD, which is going to remove the that auto inhibition. Then there are times when the cargo has very high binding affinity, so then they can knock it off.
So why
But
is it,
having said that, I'll just say
yeah.
quickly that there is a regulation that happens there, right? So the importin beta binding and release is part of the cargo regulation cycle.
So why is it like auto-inhibited at the first place?
It is actually regulation. So you don't want the importin alpha to be taking cargoes all the time without any control of how much cargo is being taken. So you need importin beta because that's the one that binds to the NPC. That is essential. Importin alpha beta complex is essential. But this cargo binding has to be regulated. So the importin beta brings in a level of regulation, that auto inhibition.
Pushkar and then Trinity. Yeah, nice talk. Swati, I mean,
Yes,
just if I can extrapolate Mrinal's question, I think there is a competition between the cargo and IBB for itself because the binding site motifs are very basic. So I think once you have the cargo, it replaces it, and that's why the auto inhibition is released. Right? Is that correct?
That is right. But again, I just want to remind you that this is in case of yeast and humans.
Right.
In case of apicomplexan, it's wide open. So then you have to start asking what is the regulation for cargo of apicomplexan parasites. So the biology of all this is something which we don't know yet.
So my question was, most of the nuclear localization studies have been done by epizomally overexpressing NLS with GFP, and that's where you have done some quantifications. But if you did, let us say, with an endogenously either tagged or a protein or endogenous protein, you're likely to get even better numbers because again, stoichiometry issue.
Yeah, that's true, Pushkar. And so again, it would depend on which protein we chose because it would have NLS of different strengths. But you're exactly right. We did NLS GFP for obvious reasons. It's easy. But you're right. We would get different numbers if we use different proteins.
Better numbers NLS in that
strengths.
regard.
Yeah.
Quickly, did you look at asexual to sexual conversion, or did you look at gametocyte development? When you say stages 3, 4, 5, so did you vis-a-vis asexual to sexual the percent conversion?
Conversion. So Mansi looked for stage 1, 2, 3, 4 gametocytes after inducing gametocytogenesis by removing serum.
Last question.
Yeah. So Asif had his cultures in growing, and then he.
Yeah, very nice talk, Swati. Just a quick thing. Did you look at the canonical sequences of NLS and whether the nuclear proteome has more preference to N or middle or C termini sequences?
Yeah. So, thanks, Niti. So, actually, for that, so the only place where Mansi looked at the local, where is that NLS found, was for the proteins which showed higher NLS strength than SV40, NLS, GFP. So, those were the ones she focused on. And in fact, in those proteins, the NLS was not at the N terminus, right? So, having a strong NLS right on the N terminus was something that the parasite nuclear proteome did not show at all. So if it's a strong NLS, it's usually in the protein, which will probably reduce its strength. Yeah. So we did do that, and we do have data.
Thank you. Thank you, Swati.
Thank you.
Thank you for your insightful presentation, ma'am. I request Professor Suman Kumar Dhar to please facilitate our speaker as a token of appreciation. A quick announcement. All those who have participated in Flash Talks, you are requested to submit your presentations in Hall 2. Thank you. I request Hemlata Balram ma'am to introduce our next speaker.
Yeah. Our next speaker is Arun Nagaraj. Arun is a scientist at Institute of Life Sciences in Bhubaneswar, and he has worked on all aspects of malaria for many years, including the parasite biology, diagnostics, novel therapeutics, etc. And today he's going to talk about his recent work on parasite biology. Arun.
So the pointer is actually this, right? It doesn't seem to work. Okay. Slide move. What is this? First, I would actually like to thank Dr. Satish and other organizers for asking me to deliver a talk in this. So the title of my today's talk is Plasmodium Metabolism, the Paradigm of Take It or Make It. So we all know that the genome editing is pretty tough in the malaria parasite because of the haploid genome. Sorry. Because of the haploid 80 genome and also the lack of NHEJ. And we can also do this only for the blood stages. So always the starting point would be the genes that are non-essential for the RBC stages. And we have made a fair amount of progress. This is mainly because of the recent advances in the genome editing. And what we know at present is out of the 5,000 and odd genes, protein coding genes that are present in the malaria parasite, 40% of them are not essential for the survival of RBC stages. And if you take a glimpse on these genes that are non-essential for the survival of RBC stages, you can broadly classify them into three. Those which are essential only for the sexual and the liver stages, for which the genes are not being expressed in the RBC stages. And then you have the multigene families that are involved in antigenic variation, modulation of host immune responses and all this. Probably a subset of the genes is being expressed in the RBC stages, and one will be able to delete these genes very well if you grow the human malaria parasite in the flask. And the one which I will be talking today is the genes that are involved in key metabolic pathways like heme synthesis, amino acid metabolism, fatty acid synthesis, and TCA cycle. If you actually delete these genes, you will find them that the genes are actually needed for the survival of sexual and the liver stages. But
These genes have been expressed in the RBC stages and the pathways are operational. People tend to write them off by telling that these are minimally active in the RBC stages or so. And what I will do today is I will actually present two findings from the lab. At the end of it, I will leave it to you to judge whether these are just the futile leaky metabolic pathways or they do carry a physiological relevance. Many of the protozoans are hemoxotropes, and people actually thought that malaria parasite will be a hemoxotrope for the simple reason that is present in human RBCs. But that was not the case, and we actually deleted the first and the last enzymes of this pathway and showed this is non-essential for the RBC stages, but is actually needed for the formation of sporocytes in the salivary glands and exoerythrocytic stages in the liver. But then you also need heme for the synthesis of cytochromes and also the function of electron transport chain in the RBC stages. And we showed that the malaria parasite can actually take the host heme that is released during hemoglobin degradation. There are also a number of other backup mechanisms. But then, why do the RBC stages synthesize all the proteins that are actually needed for the synthesis of heme and make a small amount of heme? We and others have shown that a small amount of heme has always been synthesized in the RBC stages. So this is what we had at the back of our mind. And we got the answer for this when we used Burgay as an in vivo rodent parasite model. Like many other labs, we actually grow the human parasite in the flask, but it actually doesn't help us to study the disease pathogenesis. And Burgay Anka is a lethal strain. It won't even spare a single mouse. And if you take the C57BL6 mice or CBA mice and give this Burgay, 60 to 80 percent of the mice will die due to cerebral malaria around day 7 to 8, when the blood parasitemia is around 5 to 10% or so, and the rest of the mice will die due to anemia. You will find all the typical symptoms of cerebral malaria. When we actually did this for heme pathway knockout parasites, it was a surprise. Not even a single mouse died of cerebral malaria, and even the death due to anemia was also delayed. There are a number of readouts people can do for cerebral and severe malaria assessment and all. I don't want to spend time on this data, but what I would like to tell you is none of the hallmark features of cerebral and severe malaria could be detected in the mice that are injected with heme pathway knockout parasites. And if you take a look at a hemoglobin degradation, it is a massive process. It is 7,000 molecules of heme is being released per second, and the vast majority of the heme is detoxified into an inert pigment called hemozoin. And remember, hemozoin is actually inert for the parasite, but it's not for the host. You name any aspect of the disease pathogenesis, there will be a hemozoin link. And to our surprise, what we found was the heme pathway knockout parasites synthesized a very small amount of hemozoin. And given the extent of heme that is released through hemoglobin degradation, and a small amount of heme that you need for cytochrome biogenesis, utilization of the host heme can never be an explanation for 70 to 80 percent of the reduction that you see. So we further went ahead, and finally what we found was the heme that is made by the malaria parasite is required for the synthesis of oleic acid. And this is a major unsaturated fatty acid that is present in the food vacuole, and it is known that this actually plays a very important role in hemozoin formation. And also the fate of the oleic acid, if you actually take it, is similar to what we have found with heme. The malaria parasite can synthesize it, and it can also take from the host. And in Arabidopsis, it has been shown that oleic acid can regulate the function of beta proton ATPase, and it is a major proton ATPase pump that is actually present in the food vacuole. And in agreement with that, we actually saw decrease in the pH, and also we saw very less abundance of V-type protein ATPase subunits in the food vehicle heme pathway parasites. So the summary of the story is it's the small amount of heme that is non-essential for the survival of RBC stages is the one that actually regulates the food vehicle integrity and the massive process of hemozoin formation. And it is the heme that is the non-essential heme that actually regulates the disease pathogenesis. And also, although the malaria parasite can take the host heme in the RBC stages, the host heme can never be the complete functional equivalent of the parasite heme. And we never had a molecule to inhibit heme synthesis, which can be taken forward for clinical relevance and all. Then I came across this paper where they have used antifungal griseofulvin to inhibit the host heme synthesis. Griseofulvin is an FDA approved antifungal drug. It's very safe drug, and one can actually use to an extent of 1000 grams to 2000 mg to 2000 mg per day in adults, and it can be used in the children also for weeks together. Griseofulvin binds to the fungal tubulin, and it actually prevents the growth of the microtubule. But it can also give rise to N-methyl protoporphyrin in the liver. And two mechanisms have been proposed for this. One is the cytochrome P450 and another one is free radical based. And this N-methyl protoporphyrin is a powerful inhibitor of ferrochelatase. But there was a catch in this because the dose that they have used to inhibit the host heme synthesis was 5 times than what is being provided for the antifungal treatment. What we actually did was we actually placed the bet on the organism because the malaria parasite is known to scavenge the molecules aggressively from the blood. And it also has the cytochrome P450 present, and it also has plenty of heme that is present as a free radical, as a source of free radical. And surprisingly, when we saw the griseofulvin could actually prevent the cerebral malaria in the mice at half the human equivalent dose. And we showed it using Burgeye and clinical isolates that it can inhibit the heme synthesis and hemozoin formation and all. And in agreement with the non-essential nature of the heme synthesis, you can see there is no effect on the growth at all. This we have done through oral route, intraperitoneal route, before the appearance of the cerebral malaria symptoms. After, it actually works well, and it has a huge potential to become as an adjunct drug. Why do we need an adjunct drug when we are having 6 artemisinin-based combination therapies? Because malaria mortality due to cerebral and severe malaria takes place even after the parasites have been cleared from the blood using ACTs and all. So there is a need of an adjunct drug. And morning we heard about the number of deaths and every child we actually lose in a minute or so. We have actually taken this finding forward for clinical trial. So all this data will actually look like it's been done in the rodent parasite and all. If you take a look at this, when we delete the heme pathway in falciparum, you see the same. You see a drastic reduction in the synthesis of hemozoin, and the story of heme, it actually doesn't seem to end. Another 6 more minutes, what I will do, I will actually talk about the second story. Here it will be more on the way a protein has actually evolved. Morning we heard that the malaria parasite lacks the canonical pathways for amino acid synthesis, and the different stages can actually take the amino acids from the extracellular sources, and in RBC stages you have an additional source of hemoglobin degradation that can give rise to amino acids. And the amino acids have been released so much that the surplus have been secreted out. At the same time, if you see, the parasite genome has actually retained a gene for glutamine synthetase, and glutamine is the most abundant amino acid in human blood or in the hemolymph or in the liver. Why does an organism have to have an enzyme for which the amino acid is available plenty from the host? And the labeling studies that have been carried out for the TCA cycle and have shown that the malaria parasite can actually take glutamine from the extracellular source also. And if you take a look at this enzyme is been abundantly expressed in the cytosol, be it whether it's RBC stages or the sexual stages or the liver stage. And if you glutamine synthesis is a very old enzyme. It has actually evolved a lot. You will find 3 types. The type 1 you will find it in proteobacteria, type 2 in humans and plants, and the type 3 in archaea. And they actually differ in the structure and also the sequence and the length. And the type 1 is being strongly inhibited by methionine, sulfoxamine and the phosphinothrycine. These are all glutamate analogs. And we did a number of enzymatic studies with inhibitors and all, and we found that the malaria parasite enzyme is actually belongs to type 1. At the same time, this is something we all might have read in our textbook. The type 1 enzyme is actually known for allosteric regulation and covalent modification. And all other amino acids that you are having and the products that have been derived from glutamine can inhibit the activity of glutamine synthetase. And adenylation, it actually regulates the activity of this enzyme, and it's been regulated by the TCA cycle intermediate alpha ketoglutarate. And if you take a look at the way malaria parasite enzyme has evolved, it actually lacks the two. There is no allosteric regulation at all, and there is no covalent modification. Why so? Because there is a massive amount of amino acids that's been released through the hemoglobin degradation. For an enzyme to stay active, it should not have the feedback inhibition. And this is also true for the second one that I actually spoke about, because the TCA cycle is minimally active. And what actually will happen is alpha ketoglutarate, the levels will be always low in the RBC stage. So the protein will always remain in an inactive form if the levels of alpha ketoglutarate are low. Not just this, there are 2 peptide inserts that are present. You see one of around 20 amino acids or so, and another one of around 40 amino acids, and no other organism has except for the hepatocystis, that is the close ancestor of the plasmodium, and you do not see even in other epicomplexans like the toxoplasma and all. And if you take a look at this, these 2 are actually occupying the strategic positions. If you see the first one, that is actually present in the Dover-Wetterkamer channel, making it much more compact. And the second one is present just above the active site. And when we deleted the first one, the protein was active. It actually behaved like the wild type. Only thing that we saw as a difference was it actually could not retain its activity when we changed the temperature from 37 to 42. And we know that the malaria parasite is exposed to febrile temperatures and all. It actually looked like the enzyme has actually evolved to retain its activity under febrile conditions. The second loop, when we actually deleted, there was no activity at all. We tried several times with different lengths and all this. If you actually touch the loop, there is no activity at all. When we tried to do the MT simulations, what we found was there are a large number of changes that are happening in the C alpha backbone and so on, and there was a drift in the structure. So that actually suggests that the second loop has evolved with additional changes and all this, so that it can be actually, it cannot be removed from the protein. And not just that, we also found that this enzyme is needed for falciparum, but it was not essential for Burgay and Bix. We tried to delete this enzyme, but it did not work, and finally we tried with the NOG sideways and showed that this is actually needed for the survival of the falciparum. And if you see, the methionine and the sulfoxamine can reduce the protein synthesis in falciparum but not in Burgay. You see a change in EF2 alpha phosphorylation in this, but you do not see that in bird. And surprisingly, glutamine plays a large number of roles. It is actually needed for the synthesis of nucleotides, amino sugars, glutathione, and so on. But the inhibition of the protein synthesis took place much ahead of all this. This was actually found to be because of the asparagine-rich nature. What we showed was glutamine that has been formed by the glutamine synthetase is actually needed for the synthesis of asparagine. And if you take a look at the proteome of falciparum and the rice novi, they are all asparagine-rich proteome it is. So this is one of the reason why we found that this glutamine synthetase is actually needed for the survival of falciparum. At the same time, we still do not know why Vivax, Burgay, they all have actually retained this protein. And as I told you, we could see a decrease in the asparagine levels when we treated the falciparum with methionine sulfoxone. So this is the last slide for this. Not only this, the glutamine actually plays a very important role in artemisinin exposure. It actually acts as a nitrogen reservoir. And RNA-seq studies have shown that the protein, the gene is being upregulated when you expose the artemisinin resistant parasites to artemisinin. And if you take a look at this ring survival assay data, even in exposure for just 6 hours, you can see, I think you can actually see there is a kind of a very prominent growth inhibition that is actually taking place. So it actually suggests that the glutamine synthetase is actually needed for the survival of artemisinin resistant parasites. So this is just the summary of what I actually told. What we do not know at this point is whether the make it and the tick it are independent of each other or they actually interlinked. And also whether the malaria parasite utilizes a metabolic switch of make it to control the disease outcome. And all these things like taking from the host can happen after some time once the malaria parasite can invade the human RBCs. And it's not clear at this stage whether the early stages they actually depend on its own synthesis. And at the end, are there hidden targets that are present in this non-essential pathways? Thank you. And the credit should go to the team, 5 of my students, and the fundings from the DBT, SCR, BIRAC, and others. Thank you.
Thank you, Arun, for that very interesting talk. Just one question. Any questions?
There is no question from the audience. Okay. You were going to talk now.
Yeah, so I thought I will ask question before.
Okay.
You were finished. Arun, very nice talk. So this griseofulvin, right? So I'm sure you would have thought about it. Did you look at whether when you inhibit the parasite and they transit to insectostase as?
See, in Burgay it is actually known. We have deleted the genes, right? And it can undergo up to the formation of eukaryotes and all this. So when you treat these mice, you do not see a decrease in the gametocytes and all this. I think that is what
my question is whether this is completely blocking the parasite mediated heme synthesis.
Yes. Right.
Okay. Right. So and then I don't know if you've checked this. So and it has no effect on. But you said I think it inhibits hemogen formation as well, no?
Right.
So then, which means that might enhance effectiveness of artemisinin.
We have actually done it.
You did that? Okay.
It doesn't seem to change the efficiency of artemisinin. Okay. So it is the same. Chloroquine, it actually does.
Thank you.
Recrudescence is seen.
I'm sure there are other questions, but please save them during the tea. Thank you, Arun, for that excellent talk. And
thank you, sir. Now I request Professor Suman Kumar to please facilitate our speaker, Arun, sir.
Felicitation. Please come.
A gentle reminder, all those who have their presentation running in the parallel sessions, please submit it in hall 2. Thank you. I request.
Yeah, he's already here. Our next speaker is Dr. Puran Singh Sijwal from CCMB. And
Suman and Hema. And of course, I would like to begin with thanking Satish, Saman, Dr. Rangarajan and other members of organizing committee for Okay, sorry for the interruption by me. Okay, so yeah, I was saying that I would like to thank the organizing committee for having me here and giving opportunity to talk about some of the work I'm doing in my lab at CCMB. So today I'm going to talk about an ER resident protein which functions as a lipid assemblage. So this is the life cycle of malaria parasites, all of you know. So the reason we ended up, the story I'm going to tell today was not what I wanted to do. What happened? The invasive stages of the malaria parasite here, and then the nookinete, the sporozoites, they have a set of secretory organelles which are required for invasion of the host cells. And as the parasite differentiates into the non-invasive state, in this case, let's say from merozoite to ring, and these secretory organelles disappear. So we were studying autophagy to investigate whether autophagy has a role in turnover of these organelles, because autophagy is known for turnover of cellular contents, including membrane organelles. So this is an intra-ectocytic parasite here. Okay, here the green signal here shows the labeling of the parasite with ATG8, which is a canonical autophagy marker. And we are interested how are these autophagosome-like structures formed and where do they originate. So to investigate that, we are investigating some of the proteins of autophagy which are involved in early stages of the formation of these autophagosome structure. One of the protein is the VMP1, which is vacuole membrane protein 1. Srinivas was a PhD student at the time in my lab who has finished this PhD, now working in Thermo. And he took up this project to study VMP1 to find out whether it has a role in organelle autophagosome biogenesis. And what VMP1 is, so VMP1 has been studied in model organisms. So it is an ER resident multipass transmembrane protein, which you can see here. And this portion of the molecule is called dead domain. It has been shown to have roles in autophagosome formation and various other functions. And it has members in all the life forms from bacteria to humans. And these homologs are put together in this superfamily called dead A. So now coming to the secretory organelles I was talking about. So the invasive stages of all the apicombustion parasite, in this case here is a plasmodium, here is a toxoplasma, contain the secretory organelles. These are rhopteries, here are micronemes, and here are the dense granules. You can see here as well in the merozoites. And these organelles secrete a set of proteins which are involved in motility, invasion of the host cell, parasite development, egress from the host cell, as well as remodeling of the host cells. So now our autophagy investigation ended there. What we found that it did not have any role in autophagy. Instead, what we found that it has a role in biogenesis of some of these organelles, which I will talk about. So this is the predicted domain architecture of human VMP1, and this is Plasmodium falciparum and Toxo, and this is the multi-domain, the Dead family superdomain. Our usual approach is to start with generating parasites in which the desired protein is tagged with GFP, MYC or HA. Try to generate knockout or knockdown and look at the effect of the knockout or knockdown on different stages of the parasite. So here this is Western blot of Plasmodium falciparum, asexual erythrocyte stages. What you see here is the parasites are expressing VMP1 endogenously tagged with GFP. Here we are using GFP antibody, you see expressed in all stages. This is live cell fluorescence microscopy. So VMP1 co-localizes with the ER tracker, indicating that it is ER resident protein. Repeated the same experiment in toxoplasma as well, in which we slightly change our strategy because toxoplasma allows conditional protein depletion using AID, oxygen-induced degron here. So we fuse this AID with HA to the C terminal of the TZVMP1. This is a Western blot showing expression of the fusion protein. And here the VMP1 again co-localizes with the ER marker here. And it also partially co-localizes with the Golgi reporter grasp in this case. Again indicating that in both the parasites, the VMP1 proteins are ER localized. So next question, what are the roles of these proteins in these parasites? So our lab is primarily a malaria lab. We generated 2 knockdown lines of Plasmodium falciparum VMP1 but did not see any knockdown. And that is about when Srini was within 3rd year, and he thought, well, okay, there's nothing much we were going to do. But then we talked to Avijit, who is a scientist at NIAB. So he helped us setting up toxoplasma in our lab, and we did further work using toxoplasma VMP1. So again, what we see here is, as I said, that here TZVMP1 fused with EID HA. This Western blot shows time-dependent protein depletion of TZVMP1, and nearly complete depletion is about 3 hours. And in IFA as well, here's the VMP1 signal in the control parasites, which is minus IAA. When the oxygen is added here, which is plus IAA, this signal is gone, which means we have a system, we can deplete the protein as and when we want. So first we looked at the effect of the protein TZVMP1 depletion of parasite growth. This is a plaque assay. What you see here is the control parasite, you see nice plaques, and in knockdown parasite, the plaques are gone. That means the protein is essential for parasite development. It is essential for parasite replication as well. Here the knockdown parasites, most of the parasites are arrested at single stage in the vacuole. It is also crucial for the in vivo development of the parasite in mouse. So here are the control parasite, in which the infected mice die within 10 days, whereas only 50% of the mice infected with knockdown parasite die and rest survive. Again now, so what could have caused this growth defect of the parasite? So if you look at the different features of the parasite, it comes the motility, invasion, egress, and development. Development I already showed you that when we deplete the protein, the parasite development is inhibited. And the depletion also caused defect in motility here, defect in the invasion of the host cells then compared to the control parasites, and defect in egress as well. What you see here is, if the egress is affected, so the released parasite form clump, whereas in case of the control parasite, you see nicely scheduled parasite. So the number of clump in case of knockdown parasite is much higher compared to control parasite. So these different defects would have contributed to the overall growth defect of the parasite. But these are the phenotypes. Now the question comes, okay, what is actually happening to the cellular structures of the parasite? So we focused on micronemes, rhoperies, and dense granules. So first we looked at the micronemes. What we see here is, so here we are using a microneme protein EMA1, and these are the control parasites and these are the knockdown parasites. I don't know if you can see this nice green signal in the back, but believe me that both knockdown parasites and the control parasites have nice labeling for EMA1, indicating that the micronemes are not affected at all. So micronemes secrete their contents which are required for parasite motility and invasion as well. So when we come to that, I will show you here. So these are the nice 10 micrographs. In control parasite, we have both apical micronemes as well as the lateral micronemes. In knockdown as well, we have both apical as well as the lateral micronemes here. Now coming to the secretion. So microneme contents are secreted, and while these are secreted, these proteins also processed. So we induced microneme secretion and then did Western blot of the extracellular antigens collected from the control parasite as well as knockdown parasite. So what you see here, this is MIC2. The protein gets processed in the secreted content. This is the precursor form. Whereas in case of the knockdown, you see, first of all, you see the amount of secreted protein is less, but you also see the presence of precursor form, indicating that VMP1 depletion affects microneme secretion, which could be the reason why the parasite showed reduced motility. Next, we will look at the rhoptry. So rhoptries are elongated structures at the apical end of the parasite. So in control parasite, you see nice elongated structures, which are pretty much absent in case of the knockdown parasite. Here is the quantitation. So this is stem, which clearly shows that in control parasite, you see very nice elongated rhoptries here, pretty much absent in knockdown parasite. So here this data indicated that this protein is required for rhoptry biogenesis. Now what about the rhoptry secretion? So one can induce rhoptry secretion, or rhoptry secretion is induced when the parasite comes in contact with the host cell and the rhoptry contents are secreted in the host cell, which you can do by IFA. So here in the control parasite, this is the parasite, and the secretion happens at the apical end into the host cell, whereas in the knockdown parasite, the secretion remains inside the parasite, and you can quantitate it, indicating that the knockdown of VMP1 reduced rhoptry secretion as well. So next we looked at the dense granules. So dense granules are cytoplasmic electron dense granules which secrete proteins into the PV, which are required for formation of this intravehicular network or IVN. So this is the IFA using dense granule markers like GRA2. This is what you see, which is clear here. So in control parasite, you see nice cytoplasmic green signal which represents dense granule. You also see cytoplasmic vesicles in knockdown parasite, but a large number of these proteins are stuck in the ER, indicating a defect in dense granule biogenesis, which is clear here. So here we have 3 parasites, and the space between the 2 parasites is called parasitophorus vacuole, which contains IVN. IVN are tubular and vesicular structures. Okay, when you zoom in, you can see nice tubular and vesicular structure in the control parasite, and these are completely absent in the knockdown parasite, indicating that the TZVMP1 is required for biogenesis of the dense granule as well as for the formation of IVN. And since some of the proteins of this dead A superfamily have been shown to have a role in lipid transport, so we stained our control parasites and the knockdown parasite with lipid stain, Nile stain. What you see here is the number of the green signals is more in control parasites than the knockdown parasite, but the size of the green signal is much more than the control parasite. It's quantitated here. More clear in these transmission electromicrographs. In control parasite, you see these are lipid droplets, okay, zoomed here. But now look at in the knockdown parasite, they are way much larger than the control parasite. And other thing what you see is that these lipid droplets are stuck close to the ER leaflet. So lipid droplets are formed between the ER leaflets, and the larger leaflet LDs could be due to a defect in lipid transport, a specific type of lipid transport, which we will talk about a little later. So now coming to Plasmodium falciparum. So far all the data which I showed you is for Toxoplasma gondii. But we wanted to study the role of Plasmodium falciparum. We generated knockdown parasite but did not see any knockdown of the protein. So whatever we observed with Toxoplasma gondii VMP1 is also true for Plasmodium falciparum VMP1. So what did we do? we epigenomally transfected knockdown parasite, which Plasmodium falciparum VMP1, as well as human VMP1. So human VMP1 is a very distant homologue, which shows about 30% identity with the Plasmodium falciparum and Toxoplasma VMP1. So this is the localization of TZ-VMP1, and here are the localization of PF-VMP1 and human VMP1. So the localization is correct. Both TZ-VMP1 and PF-VMP1 co-localized. So the localization not affected. Next, do PF-VMP1 and HS-VMP1 complement TZ-VMP1 functionally? That will be approved. So here we did plaque assay. So the knockdown parasites don't form plaque compared to the control parasite, but the PF-VMP1 complemented parasite do form plaque even when the TZ-VMP1 is depleted here. So is true for the human VMP1, which confirms that Plasmodium falciparum VMP1 and TZ-VMP1 are functionally similar. Not only that, interestingly, human VMP1, which is so distant, can also complement for the function of TZ-VMP1, suggesting that this family has certain common functions, okay, in different organisms. So we don't know what are all those functions, but some of those we know, which I will talk about here. That is one of the mechanism I will talk about. So I told you the phenotypes, then I told you the cellular structure affected by depletion of TZ-VMP1. Now what could be the molecular mechanism? So we evaluated these VMP1 proteins for lipid transport assay, and there are three types of lipid transporter. One which transport from outside leaflet to inner, the inner leaflet to out, and the scramblases which can transport from outer to inner or inner to outer. And the dead superfamily proteins are predicted to be scramblases. So we did an assay here. So it's a bit complex. We made liposomes here, and then these liposomes also contain a fluorescent lipid. So when diethionate, this is a membrane impermeable quencher, is added, so it quenches the fluorescence present on the outer leaflet of the liposomes. If you add tritone X100, the entire there is complete quenching. So now if your liposome contains the scramblase, so what we do is when you add diethionate, it will quench the fluorescence present on the outer leaflet, but then you have scramblase which would transport lipids from the inner leaflet to outer as well as outer leaflet. So it would cause near complete quenching of the fluorescence. In this case, of course, when you have tritone X100, so we have complete quenching. So in case when you don't have a scramblase, there will be about 50% quenching. In this case, it's nearly complete quenching. This is what you see here. So these are the empty liposomes. This is 100% fluorescence when you have diethionate. In case of empty liposomes, about 50% quenching, but you have scramblase here. In this case, TZ-VMP1, PF-VMP1, the quenching is about 90%, indicating that both TZ-VMP1 and PF-VMP1 function as scramblases, and it could be that function of TZ-VMP1 or PF-VMP1 which is required for the formation of different regions of the ER from its vesicles containing microneem or rhoptary or dense granule proteins arise and then form these organelles. That's our proposed mechanism so far, and this is summary. What I told you that the VMP1 is an ER resident protein. It has little lipid scramblase activity required for the biogenesis and secretion of different organelles, and it is knocked down. Okay, so that doesn't happen, which in turn affects motility, invasion, egress, and parasite development. Of course, this is a nice picture of my lab, and Srinivas is the one who led this work, finished his PhD. He was greatly supported by Kanika and Somesh here. And of course, thanks to our collaborators, the sources of reagents, and funding from various agencies. And thank you for listening to me.
Thank you, Puran, for the presentation. Time for one or two quick questions. Yeah.
Yeah, and Pushkar, go first. Very nice talk, Puran. So, a couple of just the obvious things. So, in the end, you were showing that scramblase activity. So, did you try the human VMP1?
Human VMP we did not try here, but there's a report in which human VMP has been already shown to have this activity. Actually, that was the clue for us to do. We did not.
All right. So the other thing I wanted to ask was, there you showed all these different, whatever the phenotypes with
Right.
the knockout.
Right.
You showed for invasion, you showed for
Motility
all
and
the
all that.
motility and all.
Yeah.
Did you try egress? Because egress is calcium
Egress was affected
with toxoplasma.
in toxoplasma. In fact,
But is it a calcium ionophore mediated
no,
egress?
The egress, it's ethanol, yeah.
Because you should try the calcium ionophore mediated.
Yeah. So in fact, what we found that actually that's very interesting question. So we also measured calcium levels in these parasites. So the knockdown parasites have higher calcium levels than the control parasites. So in fact, we were not expecting that, because one of the functions of human VMP1 is to regulate SARCA, which in turn regulates calcium levels. But in our IP also we did not find the toxoplasma SARCA.
Yeah, please, last question. Very interesting. So obviously, human cells do not have rhabdoids, micronemes, and so on and so forth, which are very toxoplasma specific functions. So obviously, I mean, since you're getting complementation, it has to be something to do with the biochemical machinery which interferon feeds. So that's a very different kettle of fish I can put.
Yeah.
I mean, there were no defects in apical organelles except dense granules, right? When you knock down TGVMP1, you said the formation, the rhabdoids are fine.
Rhabdoids are
micronemes are fine. Yeah.
Rhabdoids and dense granules.
So why specifically rhabdoid microneme? And affiliated question, you at the first place see differences in replication of the parasite, which is very different from rhabdoid related function, right?
Yeah.
So how do you?
So yeah, so I don't know if you were the reviewer. So these were the questions we got.
No, I was not. I can tell you.
Okay, but I think that's very important question, and very yeah. This is what we, so our, I don't have a exact answer, but so our expression for that was, so in the ER, the microneme proteins and the rhabdoid proteins are transported from different regions. Okay, to micronemes as well as the rhabdoids. So what we think, and this protein, which I didn't tell you, the VMP1, when we knock it down, the ER organization also affected greatly. So what we think, there could be two things. One, it might affect microdomains of the ER from vesicles containing microneme or rhabdoid proteins arise. That's one. Number two, as a lipid ascomplase, it may provide lipids which are required for formation of those vesicles. So that is, and it does have ascomplase activity. On the other hand, the the dense granule proteins and the vesicles, they are assembled in the ER itself. So probably because of that, they get stuck in the ER.
Toxoplasma does not.
So exactly. So that's what we are working on. So because we generated knockdown plasmodium falciparum VMP1 lines knockdown, two approaches, but didn't see any protein depletion. No. But we didn't get any knockdown. Protein depletion we didn't see.
Thank you. I think we can continue discussion during the tea. So with that, we end this session, and also thank you all the speakers from our side as well as from the organizers.
Thank you, sir.
Thank you.
Thank you, sir. May I request our chairpersons to facilitate our Puran, sir. So we thank both the chairpersons. So we are concluding this session. I request Dr. Shamal Roy to please felicitate our chairpersons. Thank you for your time, everybody. So we will now take a short tea break, and you all are cordially invited to join us for refreshments in CDRI lawn. The next two sessions will run parallelly at 5:20 p.m. Session 3 on immunobiology of parasitic infections will be conducted in auditorium, while session 4 on biology of parasitic infections will be conducted in hall 2. Kindly be back in time so that we may start promptly. Thank you so much. So I request you all to please be ready at 5:15 p.m. p.m. in advance. It's fine. Come back, please. Thank you, sir.
Good evening, everyone, and welcome to the session on immunobiology of parasitic infections. We are honored to have chairpersons Professor Mrinal Kanti Bhattacharya from the University of Hyderabad, a distinguished expert in malaria research, and Dr. Kalyaneshwar Mandal, professor at Tata Institute of Fundamental Research, Hyderabad, leading expert of synthetic organic chemistry and protein crystallography. May I kindly request both our esteemed chairs to please take over the session.
Good evening, everybody. I welcome you all to the evening session. Today we have 5 distinguished lectures in this session. The first lecture will be by Professor Shamal Roy. He is a known immunologist and did some seminal work with leishmaniasis. Currently, he is a Bose National Fellow at Cultivation of Science, Kolkata. So, over to you, Professor Shamal Roy. So, your talk will be for 25 minutes, followed by 5 minutes question answer.
Okay. So first of all, I thank organizers for giving me this opportunity to talk about some of the works that we have been currently pursuing. Over the years, I have spent my life at the Indian Institute of Chemical Biology and recently moved to Cultivation of Science, Calcutta. So I'm going to talk about one of the important protein, surface protein of Leishmania parasite, which is called the kinetoplastid membrane protein. In short, it is KMP11, as a new virulence factor for the Leishmania infection. So this protein favors the entry of the parasites into the host cells. So before I go into the subject proper, I thought I should talk about a little bit of history of Leishmania research that was done in Calcutta. And this is the unknown enemy in the Indian subcontinent, a long anguish of black sickness. So before the British Empire formally take over the British Crown, East India Company was in power, and East India Company actually established some hospitals, but those are basically for the people of the British Crown, and so hardly there is any treatment available for the common native people. So the entire Gangetic Plain and Brahmaputra Valley was the valley of shadow of death because of Kalajar, and there is no treatment available. And disease was not even diagnosed that time. People thought it could be some malaria because symptomatically those are similar. Now, the disease started actually in Jashore district in 1824, then moved to Bardwan, then to Garo Hills, to Naugan, Tezpur, Mongoldoi, Purnia, Naxalbari to Guwahati, and ultimately it started to spread all over the country. So, and this is the first patient of Kalajar who was afflicted, a British soldier, basically an unknown disease. And that patient was taken to the Royal Victoria Hospital in Natle, where the patient received the treatment. But patient died, and William Leishman was the physician who investigated the case and did some post-mortem investigation and found some tiny dots in the WBC or white blood cells of these patients. Now, in this 1903, Charles Donovan was a professor of physiology at Madras Medical College. Charles Donovan was born in Calcutta, but his father was a kind of British man working in some capacities, I don't know. So he did his schooling in Dehradun, Missouri, and then went to Dublin to study medicine and came back. And he wrote a letter to Ronald Ross in Calcutta, showing that some of the tiny dots in the screening aspirate of these patients and asking Ross what could be the new parasite. And similarly, Leishman also wrote a letter to Ross asking his opinion about these new tiny dots in the WBC of these patients. Then the controversy started, who will be the discoverer of this parasite. So Ross took up this investigation, Ross's laboratory in Calcutta, and he took up this investigation, Lady Ross, and he published the paper in British Medical Journal, giving the name as Leishmania donovani, the causative agent of Kala-Azar. And this is the first electron micrograph of Leishmania parasite was published by Calcutta University and School of Tropical Medicine in 1951 in Nature. So I'll talk about some of the surface protein of this Leishmania parasite, that kinetoplastid membrane protein, and this is the macrophage, the home of the Leishmania parasites, and these are the Leishmania parasites that bind to the surface of the macrophages, and then ultimately they enter into the host cells and where they replicate. And this protein, we have cloned and expressed this protein, and this is roughly about 2 million copies of this protein present on the surface of this parasite. And we cloned and expressed this parasite on the Leishmania, and this is one of the vaccine candidates that Dr. N.K. Ganguly was mentioning in the morning, and the paper was published in Science Translational Medicine 2014 as a potential vaccine candidate for the Leishmania infection. So this protein, KMP11 protein, many years we tried to really identify this protein in the serum of the patient, but we are fairly unsuccessful. We never knew that protein aggregates, and it actually, because of the aggregation, we could not really identify this protein in the sera of the infected patients. Now, this monomeric protein, they form oligomer, and ultimately we can see oligomeric KMP11 by AMF images, and it forms the pore, and we have some evidences that it forms the pore and ultimately allows the parasite entry into the host cells. And it has fairly strong affinity. This KMP11 protein has fairly strong affinity for the protein KMP11 that is present on the surface of any somatic cell. And look for binding of NBD cholesterol to KMP11 protein. And NBD cholesterol, we can excite this molecule at 469, and emission one can study at 537 nanometer. And we can see the binding of NBD cholesterol with the KMP11 protein, with K is not very high, but some degree of K with the binding affinity with the cholesterol. Next, we wanted to see that now we are convinced that cholesterol binds to KMP11 protein, and cholesterol also form oligomers, and it gets to the host cell surface, forms the pore, and allow the parasites to enter into the host cells. So membrane, we know that cholesterol is very important for maintaining membrane fluidity of the somatic cell surface, and if parasite takes up membrane cholesterol through KMP11, then macrophage surface will be very fluid. So taking different types of fluorescence probe, and here one of the fluorescence probe that is took, that is the DPH, and another is the TPA DPH, and it has an excitation of 350 nanometer and emission of 420 nanometer. And we actually, if we take these macrophages of the normal Balbstein mouse, and as opposed to the macrophages taken from the infected Balbstein mouse, and there is a distorted morphology, lot of blebs and fissure, suggesting that probably the membrane fluidity of these macrophages must have been altered. And we also showed that if we infect these macrophages with the Leishmania parasite, as a function of time, there is a progressive decrease in the membrane cholesterol. So by 24 hours, there is a significant decrease of the membrane cholesterol as opposed to the normal macrophages. And if we study the membrane fluidity in terms of fluorescence and isotropy, then fluorescence and isotropy and membrane fluidity is inversely proportional. So this is in the normal macrophages, and isotropy value is roughly 0.3, 0.35 or something around. But if we take the infected macrophages, we can see that the membrane fluidity and isotropy value is decreased. That means fluidity
of the infected macrophages has been significantly increased. So what we gain from here, that infected macrophages, that because of the decreased cholesterol, the membrane becomes more fluid, and there, which we can see from this scanning electron microscopy data. Next, we wanted to see that we can also put back cholesterol into the membrane by making liposomal cholesterol, and we make a liposome which is rich in cholesterol, and that this is in the when there is a less cholesterol, the membrane become very fluid. But if we put back cholesterol to the liposomal delivery, then we can see that it becomes rigid again. So by liposomal delivery, we can make macrophage membrane rigid. And basically attributes of the cholesterol from the liposome, that it is rich in cholesterol, so that favors the unidirectional transfer of cholesterol, and that simply follows the first order kinetics. And also it takes away cholesterol from the membrane. It also increases the lateral fluidity, lateral mobility of the membrane protein. So what we did, we transpect the macrophages with PLC delta, which is GFP level protein, and if we bleach it by a laser, what happens actually, we do that, we do some FRAP studies. Number one, that the fluorescent, green fluorescence one can see. Number two, after photo bleaching, we can see that hardly there is any GFP, and we put some time to go, and there is a recovery. The time recovery you need, if it is more fluid, you require more time as compared to rigid membrane. So this is, one can study the diffusion constant very nicely, the normal macrophages versus infected macrophages. The diffusion coefficient is significantly increased, and if we put back cholesterol into the membrane, then diffusion coefficient coming back to what we see in the normal population, normal macrophages. So it tells us that if we infect macrophages with Leishmania parasites, lateral mobility of the membrane protein is significantly increased, and if we put back cholesterol by liposomal delivery, then it is restored again. So we studied some of the mechanisms why cholesterol is significantly decreased in the patients as well as in the infected macrophages. And what we see here, the one of the microRNA, that is the pre-microRNA 122 is important, and which has indirectly promotes HMG-CoA reductase activity, and inhibiting microRNA 122, HMG-CoA expression is reduced, leading to lower plasma cholesterol level. So what we show here, that pre-microRNA 122, its maturation is inhibited by GP63, which is a surface protease and metalloprotease, and it cleaves the dicer, and therefore there is no maturation of microRNA, pre-microRNA 122, microRNA 122, and that leads to the decreased cholesterol level. So mechanism of cholesterol reduction in the patient, probably because of this metalloprotease GP63 that cleaves the dicer, and that prevents the maturation of pre-microRNA 122, microRNA 122, and therefore there is a decrease in the serum cholesterol in the patients. Now, then we used the recombinant protein KMP11 and wanted to see whether KMP11 can reduce or decrease the membrane fluidity. So we took here two fluorescent probe. One is DPH and another the Lordan. DPH, which is we can excite it at 350 nanometer, and emission we can study at 420 nanometer. And for Lordan, it is 340 versus 400 to 550 nanometer. And both the cases we can see FA value or significantly decreased as a function of KMP11 concentration, suggesting that whatever in infection, what we have seen, the decrease in membrane fluidity, probably it has been attributed by this membrane protein KMP11, that causing the decrease in the membrane fluidity of the host cells. Then we become interested to study which part of the protein, KMP11 protein, actually interacting with the cell surface. So what we did actually here, we did very simple thing. we did some tryptophan mutation of this protein, and there are four tryptophan, four tryptophan mutations at the site 5, then 48, 62, and 89. These 4 sites where we have done some tryptophan mutation, and then we made a liposome which is made with dehydroergosterol, and the tryptophan we can excite at a 225 nanometer, and emission we can study at 328 nanometer. And dehydroergosterol, which has a fluorescence property, we can excite it at a 325 to 350, and emission we can study at 375 nanometer. So it become a very nice freight combination to study the if it is within the Foster radii 10 nanometer, then if we excite the donor with 295 nanometer, and then emission we will be able to study at 375 nanometer. So using this kind of biophysical technique, so what we did, we studied this the donor accepted distance, that is R0. So at the when we did the mutation at the tryptophan mutation at the site 5 versus 48, 62, and 89, and we can see the least distance here when we have seen the mutation at the position 5, and also we see at the position 89, but rest of the part is probably away from the Foster radii. So based on this kind of biophysical approach, we propose that membrane protein KMP11 is interacting with the membrane is like this, that this part is somehow been interacting with the membrane, and this part is also interacting, C terminal also interacting in the membrane, and rest of these two parts are probably floating up in the membrane, and probably this is the way KMP11 actually interacts and forms a oligomer, and that forms the pore, and that allows the entry of the parasite into the host cells. So to show that it actually favoring the parasite entry, we took treated macrophages with KMP11 protein, and then infect with the Leishmania parasites, and we can see that if we treat the macrophages with the KMP11 protein, and we can see significant number of increased parasite burden in the macrophages, and as opposed to infection with the wild type LD parasites. So therefore it tells us that because of the increase in the membrane fluidity by this protein, probably that forms the pore and favoring the entry of the parasite into the host cells. And also we looked at the certain peptide part, whether it is interacting with the membrane and causing the increase in the membrane fluidity or not. And we show that LD parasite, if we take the peptide sequences from this KMP11 1 to 19, and we can see that instead of whole protein, if we take sequence 1 to 19, that also favors the parasite entry into the host cell, suggesting that only sequence 1 to 19 probably important for increasing the membrane fluidity and therefore favoring the entry of the parasites. So to come back, that KMP11 indeed is important for interacting with the host cells or not. So what we did, by applying CRISPR-Cas technique, we removed or knock out the KMP11 protein from the Leishmania parasite, and after, so if we remove KMP11 or knock out the KMP11 protein, and the parasite morphology is completely altered, and we can do some complementation studies where the KMP11 after introduction, its morphology coming more or less like the normal Leishmania parasites. So we did some functional studies with the wild type parasites and knockout parasites and the parasite that has been complemented with the KMP11 protein. And what we show here, that with the wild type parasite, number of attachment of the parasite to the host cells roughly about maybe 250 or something. So if we take out the knockout parasites, hardly there is any attachment of the parasites with the macrophages. And then if we do the complementation studies, then it is restored again. And also if we take one parasite which has been cultured for a long period of time 200 passes also loses its virulency. So therefore, this is also is significantly reduced if we take a very aged culture which has been passes for 200 times. So putting all these things together, it seems to us that the KMP11 is a protein which is a virulence factor, and it causes or increases the membrane fluidity, and it forms the pore, forms oligomer, forms the pore, and therefore it favors the entry of the parasites. And from these studies we could show indeed this is an important protein of the Leishmania parasite for the early attachment with the host cells. So in summary, we can show that KMP11 protein is through parasite is interacting with the macrophages, and it is attaching and ultimately entry, and KMP11 protein is getting into the membrane, and from this causes RAP disruption, and ultimately because of this RAP disruption there is a severe defect in the T cell stimulated ability, and etc. etc. So in our study, what it tells us that KMP11 is one of the important virulence factor of the Leishmania parasites, and probably it is important for the entry of the parasites into the host cells. So this is my people. Actually, I collaborate with many people. So Rajiv, Ochinto, basically many of this work done by Ochinto. Buddhaditya Mukherjee, my student currently at IIT Kharagpur, and Moumita Bhumi did a lot of this lateral mobility studies. Junaid Gibran at the Presidency College, University, and he is still continuing with me. And some of my collaborators, Krishnananda Chattopadhyay, he is doing a lot of biophysical studies together. Shubhendra, he was working with the microRNA, and we got some help from him to study microRNA 1 to 2 at a great detail. Pradeep Das, we actually collaborated through our European Commission project for vaccine development, and Sham Sundar, who is actually providing different parasite culture from his clinic at the Banaras Hindu University. With this, I stop here. Thank you very much.
Your talk was fantastic and well within the time, so we have time for many questions.
Sir, just wanted to know, at first I would like to say it's a wonderful talk. You have started with such a beautiful slides of the Ronald Rods and early history of leishmaniasis. So my question is that is the macrophage receptor is known for this KMP11 where it bind particularly?
KMP11 receptor, it's a cholesterol.
Cholesterol. So
if you remove cholesterol from the cell,
yeah,
then parasite actually it enters because membrane fluidity has increased. Okay. So it's independent of cholesterol.
And second question is that whether this is involved in after early infection, is there KMP11 has any implication in the amastigo stage or intracellular macrophage survival, the defense machinery?
Whether KMP11 knockout parasite, whether it transformed to amastigote or not.
No, not like that. That KMP11 is involved in other inhibition of macrophage function like ROS, nitric oxide, cytokines, MSC presentation, this kind of thing.
But KMP11 as a protein, if we immunize animal with the KMP11 protein, so it actually stimulate more of a Th1 type of response.
Okay.
Okay, and in turn it can drive superoxides and nitric oxide production.
Yeah, that's okay.
Yes.
So, sir, when also cholesterol can also enter the cell via the LDL, right? Like LDL cholesterol go to the lysosome cholesterol. So, why during infection the lysosome is not repairing the membrane?
No, ribosome does repair the membrane.
No, the lysosome.
Oh, lysosome. So your question is that why lysosome is
like,
not?
Yeah, like the LDLR through LDLR, the LDL cholesterol is going to the lysosome where the cholesterol will be released and it is there.
Okay.
So I understand that the pathogen is altering the cholesterol biosynthesis pathway.
Right.
But why the lysosomal cholesterol is not why the lysosome is not repairing the cell membrane.
So entire synthesis of cholesterol is reduced. Okay, if you go to patient serum, normally we have roughly about 200 values of cholesterol, normal value, but there are patients their cholesterol level is about 20. So overall cholesterol biosynthesis is significantly reduced. I do not know what is the status of LDL or HDL. So that I think some publications there LDL HDL ratio. I don't know actually. It was done from Patna. Okay. So overall cholesterol biosynthetic pathway is reduced.
Good evening, sir.
Good evening.
I have a query. The infectivity, the reduction infectivity for the KMP11 knockout, was that restricted to any specific type of the resistant parasite or not?
Whether we have done with any resistant parasites
Yes,
or not?
yes.
No, we have not done with the resistant parasites, but definitely I know that the KMP11 number is higher in the resistant parasites. So, but actually we have not studied with the resistant parasites.
Okay, thank you.
Okay, if there is no further questions, let's congratulate
Thank you
for an excellent talk.
Thank you so much, sir, for the wonderful presentation. May I now request Professor Bhattacharya to kindly felicitate our speaker as a token of appreciation.
No, it's not. Yeah, now it's fine. Yeah, so the next speaker is Dr. Sushanto Kaur. He's a senior principal scientist from IICV Kolkata. He's also known for immunoparasitology research. Am I audible? Yeah, I think so. At first, I would like to thank Misra ji, Dr. Satish Misra, or my beloved colleague when I was in CDRI. So, for inviting me in this esteemed platform of Indian Society for Parasitology to present my work. So, I am privileged always to come here in CDRI because I have started my research journey here as an independent investigator almost 14 years back. I spent a very good time here over a period of 12 years. I learned so many things from CDRI. I have lots of good friends, even like my family members, Dr. Meghang Srivastav, Dr. Amit Lahiri. Junior like Namit Lahiri is a virtue that I got this kind of junior and colleagues, also Vidyut. So coming back to presentation, I moved to IICB on 2023, and before going to the presentation, I'm just going to share you that. So this is just a basic slide to say that what is going on in my lab for last 10, 12 years or 14 years. So what I did, I just divided the whole lab's research program into two parts. I have joined CDRI. When I have joined, I have started the anti-leishmanial drug discovery screening program in my lab, which is the mandate of our institute also at that time. So basically, we have a very nice platform. Lucifer is expressing Leishmania parasite, which we use for anti-leishmanial drug screening. So this is the screening part, or direct, I named as a direct therapeutic approach. And if we got a good active lead in the in vitro model, depending on IC50 and selectivity index, we check them in the hamster model, which is a chronic model for visual leishmaniasis. And we also employ biopsy mice for immunological study. But the second part, this is a very brief and general presentation. This one, the indirect therapeutic approach, which is a basic biology part. In 10-12 years, I have mainly focuses on this TLR signaling and cytokine mediated STAT signaling, how the parasite inhibit these two pathways, host defensive pathway for disease progression. So this is to summarize in last 10 years observation. This is what we have found. If you see this negative, this red color protein, just like TOLIP, SOX3, epigenetic enzymes. These are the early negative regulators of host immunity. The parasite employs TOLIP to inhibit the TLR signaling at the early level, and epigenetic enzymes like histone lysine methyl transferase and DB-thylase to inhibit the M1 M2 to change the M1 M2 balance. It favors the M2 balance during infection. And the late phase, the parasite also induces SOX3, PGE2, prostaglandin E2 from macrophages, which bind to the T cell receptor and induces the cyclic AMP pathway that induces another negative regulator SOX3, which inhibit the STAT4 IFN gamma signaling. So this is how it inhibits the pro-inflammatory signaling. The parasite also induces an anti-inflammatory pathway JAG-NOT signaling, which in turn activate the anti-inflammatory TH2 response like IL-4 and IL-10 synthesis. So this is what we have done, and all these are host negative regulator. Now we are also focusing on what are the parasite derived factors that induces this host negative regulator, particularly LPG, GPC. LPG is a lipophosphogycan, which is a major abundant promasticode surface molecule, and GP63 is a zinc dependent metalloprotease. These two surface molecules are important for inhibition of various immune cell signaling cascades and macrophage function. And the third one is exosome, which is well characterized in last 7-8 years, that the parasite could secrete, Leishmania parasite could secrete exosome if you give the mammalian stress 37 degrees centigrade, and also the infected macrophage could also induces exosome synthesis. So this exosome, when this infected macrophage derived exosome, when incubated with the different immune cells like T cell, dendritic cell, and macrophage, we found that there is a preferential uptake by T cells with a robust IL-10 synthesis. So we are now working on the elucidate the mechanism of biogenesis and secretion of exosome in host, and also we have found that LPG and GP6 LPG has involvement in inducing the SOX1 induction. So this is, I am not going to discuss today the exosome biology part, and also we are interested in doing whether, because these exosomes are anti-inflammatory in nature, we have also already observed that. We hypothesize that whether we could exploit this exosome for as a anti-inflammatory therapeutic vesicle for type 1 autoimmune disease like inflammatory bowel disease or rheumatoid arthritis. So this work is going on in my lab, but in today's presentation I will restrict myself in this work how the parasite induces specific classes of protein arginine methyl transferase, which are specific classes of epigenetic modifier to induce M2 polarization. Now, why we have selected this epigenetic modifiers? There are 3 types of protein arginine methyl transferase: class 1, class 2, and class 3, which either employed asymmetric, symmetric dimethylation or monomethylation. There are 9 PRMTs in mammalian cells, and they are already implicated in disease biology like HSV infection or different kind of cancer. PRMT 5, 4, 6, 1 are involved, and among them the most well characterized PRMT is PRMT 1, protein arginine methyl transferase 1, which is well known to induces TH2 cytokine like IL-10. It inhibits TNF alpha induces NF kappa B activity. It induces PPR gamma, which is a major M2 polarized transcription factor. So these things are done by the PRMT1, and we hypothesize that the parasite may employ specific classes of PRMT to induce M2 polarization, because PRMTs are already well known for M2 polarization. So we relatively took an unbiased approach. First, we did the expression profiling of all PRMTs and found that PRMT1 was robustly upregulated in infected macrophages, where with a little bit of lower induction of PRMT4. This was also in the protein level. We got PRMT1 induction up to 48 hours of infection. Here, and the parasite could also induce the M2 genes like PPR gamma, ARG1, MRC1, and FIS1. And when we inhibit PRMT1 by pharmacological inhibitor furamidine, we found there is a specific downregulation of MRC1, PPR gamma, and ARG1 in infected macrophages, whereas FIS1 and Y1 expression remain unaltered. And if we block cells with furamidine, which is PRMT1 inhibitor, there is a decrease in parasitemia. We got almost 75% inhibition of parasite 1. So how this process is happening, how the parasite regulate this M2 transcription factor, MRC1, PPR gamma, and ARG1 is regulated by PRMT1. This is our next question. And we observe that since PRMT1 induces asymmetric dimethylation, H4R3 dimethylation in the promoter of M2 gene, we found that parasite could also induce the H4RT dimethylation at PPR gamma R1, MRC1 and FIS1 promoter. But if we block them, if we block this phenomena by furamidine, we found there is an inhibition of R1, inhibition of MRC1, and inhibition of PPR gamma binding, suggesting that these three transcription factor is under the control of PRMT1. Now, how these three transcription factor induce, and what is the specific involvement of PRMT in this process? So, to start with arginase 1, which is a very well-known M2 gene or anti-inflammatory gene. So, earlier study revealed that the arginase 1 could activate. Here, if you see that the parasite induces the arginase 1 expression at mRNA and protein level, and arginase 1 is for arginase 1 induction. It is the involvement of PPR gamma is also necessary. So we employ PPR gamma inhibitor GW966, and we found that arginase 1 induction is not under the control of PPR gamma, because blocking PPR gamma does not induce arginase 1 expression. Rather, we found there is an involvement of STAT6. STAT6 is also well known to induce arginase 1 synthesis in macrophages. So here, what we have to summarize that STAT6 could bind to the arginase 1 promoter in infected macrophages, which is independent of PPR gamma. And as we found that there is no inhibition with the PPR gamma inhibitor. But STAT6 phosphorylation, if you check the STAT6 phosphorylation here, it is getting compromised by the PRMT1 inhibitor, that is furamidine, because the asymmetric dimethylation of STAT6 is a prerequisite for its phosphorylation. So this is ADOX is an adenosine methyl transferase inhibitor. We observe that the phosphorylated tyrosine, the STAT6 phosphorylation is dependent on asymmetric dimethylation, and if we put ADOX, there is an inhibition of asymmetric dimethylation of STAT6. That led to inhibition of STAT6 binding to arginase 1 promoter. If you see here that when we employ PRMT1 inhibitor, there is less binding of STAT6 to the arginase 1 promoter, and also methyl transferase inhibitor could also inhibit the STAT6 binding to arginase 1 promoter. So this is suggestive of that the PRMT1 could induce the host arginase 1 synthesis by STAT6 dependent manner. Next transcription factor is MRC1. How it is induced? Now MRC1 is typically regulated in the empty polarized macrophage by the transcription factor CMYC. CMYC, not only MRC1, more than 40% gene of empty polarized transcription factors are regulated by CMYC. So we checked the involvement of CMYC. Here we found that CMYC could bind to the MRC1 promoter in infected macrophages. CMYC expression is also upregulated during infection, but when we block PRMT1, there is no inhibition of CMYC expression. Rather, CMYC binding to MRC1 promoter is compromised if we inhibit furamidine. So this is further validated by adenomethyl transferase inhibitor. CMYC binding to MRC1 promoter is also inhibited by ADOX, adenomethyl transferase inhibitor. And not only that, CMYC, we also observed CMYC asymmetric dimethylation by co-IP study, which is abrogated in presence of PRMT1 inhibition. That means arginine methylation by PRMT1 regulate CMYC dependent transcription of this transcription factor MRC1 or this gene M2 gene. Now the next question is how this PRMT1 is upregulated during infection. Now we don't have any literature support for that report, that exact signaling event that induces PRMT1 induction. So we took relatively unbiased approach. We take 25 to 30 anti-inflammatory transcription factor, and with the help of bioinformatics, I didn't put the bioinformatics slide here. My collaborator have used Algen, Promo, and different kind of software that could predict the binding of this 25 transcription factor, 20 to 25 transcription factor in the PRMT1 promoter. And accordingly, we ordered the cheap assay primer and did the all cell biological assay. And for this, among this 25 transcription factor, I found there is a binding of these 5: CREP, STAT3, CMYC, STAT6, and PRMT1 and CEBP beta. All these transcription factor, all these 5 transcription factors could bind to the PRMT1 promoter during infection. But if we block all these transcription factor either by siRNA or pharmacologic inhibitor, we found that there is a decrease in PRMT1 induction only in case of CEBP beta, marked reduction, and there is a little reduction in case of cyclic AMP PK inhibitor or CREB inhibitor, that is HAT. So CEBP beta is the master regulator, as it is evident from here, because when we inhibit CEBP beta by siRNA, there is a decrease in PRMT1 induction in macrophages, and also CEBP beta could bind to the PRMT1 promoter. Now the question is that how this CEBP beta is induced during infection.
So for that, already there are reports that CB beta could be has some kind of role with the cyclic AMP-PK pathway, and the parasite we found that leishmania induces cyclic AMP-PK pathway, that is phosphorylation of PK and phosphorylation of CRAB, and CB beta could also go to the nucleus, and CRAB binding to CB beta promoter is also observed during infection. Unfortunately, when we block this pathway, cyclic AMP-PK pathway, by inhibitor H89, which is a specific inhibitor for PK, we found that there is a reduction of CRAB binding to CB beta promoter, and CB beta expression is downregulated by this PK inhibitor. And these events happens up to 18 hours of infection, and if we can rewind that the CB beta could upregulated up to 36 hours of infection. So we hypothesize that there must be some other regulator that that regulate its long-term induction of CB beta. So here, if you see here that when we employ H89 could inhibit the CB beta induction, PRMT1 induction up to 12 hours of infection. That is cyclic AMP. If you block cyclic AMP-PK pathway, we got PRMT1 induction inhibition up at the early level. But the second, when you employ the TGF beta inhibitor, there is no changes in the PRMT1 induction in the early level. But if you see here at the 24 hour and 36 hour, the TGF beta inhibitor SB40 could downregulate PRMT1 induction at the late hour of infection. So this TGF beta is also induced by the parasite leishman. And here also the TGF beta inhibitor could inhibit the CB beta expression at 36 hours of infection or 48 hours of infection, and CB beta binding to the PRMT1 promoter is also compromised in presence of SB, that is TGF beta inhibitor. This is suggested that TGF beta signaling somewhat regulated the PRMT1 induction at the late level of infection. So the final question is that what are the parasite derived factors? So initially we started with these two main parasite derived factor, LPG and GP63. As I mentioned that these two parasite derived factors are well known to inhibit different macrophage signaling cascade. So if you see here that we use 110 phenolphthalein, which is a zinc dependent metalloprotease inhibitor, could inhibit the GP63 also, and we have LPG knockout parasite, leishmania parasite, which we are maintaining for a long time. So we found that when we employ GP63 inhibitor, there is no inhibition of PRMT1 induction, but employing LPG knockout parasite inhibit the PRMT1 induction, and also the CP beta binding to PRMT1 promoter is inhibited by LPG knockout parasite. Then the upstream signaling event, the phosphorylation of CRAB binding to CB beta promoter and CB beta binding to PRMT1 promoter is also compromised if we employ LPG knockout parasite. And this LPG mediated inhibition of CRAB also led to inhibition of COX2 and PGE2 release, because if we externally supplement with 10 nanomolar PGE2, then LPG mediated knockout of inhibition of CB beta binding to PRMT1 promoter is restored here. If you see here that it is a restoration if you employ 10 nanomolar PGE2, even 10 nanomolar PGE2 which rescue the biological effect of CRAB COX2 could also induce PRMT1 activation. So this is the summary of the story so far, that what we have observed, that I didn't put here the PI3 kinase AKT pathway data which you have found to inhibit activate the phospho-CRAB at 6 to 12 hours of infection. So leishmanial LPG activate PI3 kinase AKT pathway for phosphorylation of CRAB, and CRAB then induces COX2, and COX2 can increases PGE2 release. This PGE2 then bind to the cyclic EP2-EP4 receptor in the macrophage and activate another sort of cyclic AMP-PK pathway and CRAB mediated CB beta induction. This CB beta in turn activate the PRMT1 induction. Also there are involvement of TGF beta signaling pathway. So all these are done in either in GH1 cell or in J774 macrophage. So this experiment is also going on. What is the implication of this PRMT in the in vivo disease progression? We are now the study is going on, and also this structure of PRMT1 is known. We are also trying to synthesize this PRMT1 inhibitor by in silico approach, and we have ordered some in Medchem Express, and we'll check their efficacy in the experimental model of VL very soon. That's all. So this is what the PRMT story and the collective summary of the presentation. So I would like to acknowledge all the funding agencies. So far I have received CSIR, DST, DBT and INSA grant, and this is my last lab in CSIR-CDRI. All this PRMT work was done by Anuradha, and other works like this Leishmania, this epigenetic work and the SOX work were done by Pragya at that time. In the IICB, I don't have so far very good. I recently published in Journal of Infectious Disease, and this is the IICB group from 2023. They are the pillar of my lab. So thank you so much for your patience.
Thank you, Dr. Kaur. Very nice work. So the floor is open for questions, maybe 2, 3.
Sir, very interesting presentation, very insightful work you have done. So my query, basically it's a query. In case of this work, M1 to M2 polarization, which you have found, so is there, have you checked the negative regulator of cytokine signaling SOX1, 2, CIS?
In context of what? They are upregulated during infection.
In context of PRMT1.
Yeah. If you are asking that whether PRMT1 could regulate other
negative regulator of cytokine signaling or not.
See, PRMT1 is an epigenetic modifier. If particular, although PRMT has some non-histone, you are right, the non-histone target also. It could methylate the protein also and regulate its stability. So to answer your question, right now I didn't check the level of this methylation in Tolib, SOX3 and other proteins.
Okay, sir.
Hello, sir. This side.
Oh, yeah.
Very nice talk, sir. Actually, you also talk about that Leishmania derived factor, that is GP63.
Yeah.
Like, is it like, I know that it is a metalloproteinase kind of.
Zinc dependent metalloproteinase.
Okay, so like, and it is a glycoprotein also. Or
yes, it is. I think it's a glycoprotein also.
Okay, so
in the promasticode surface, it is a glycoprotein.
Glycoprotein, huh? So like you have extracted it in your lab or like?
No. I didn't extract and neither do anything. Because
okay.
I employ the inhibitor of GP63.
Acha. Okay.
Also, you can say that is not sure shot inhibitor because that is 110 phenanthrolene is it's a zinc chelator. So
okay.
It's a zinc dependent metalloproteinase. We have ordered some GP63 inhibitory peptide, and we should validate the last slide with this peptide. So.
Okay. So like okay. And second question is that like in that case, in your case also like you have explained about PPR gamma.
Yeah.
And this kind of thing. So like have you checked about like how it affects the autophagosome formation or the autophagy?
No. For this work I didn't check whether it is involved in autophagy or autophagosome. Why you are asking? Because PPR gamma has a role in autophagosome formation.
No, kind of like because it is going to the anti-inflammatory also, and sometimes when that PPR gamma and PMRT that you have discussed now.
Yeah.
So in some of the literature review that I have studied, so this pathway also can go towards autophagosome, like autophagy.
But Leishmania inhibit autophagy of macrophage. If PPR gamma is involved in autophagy activation, then I don't think that we don't see that. We will not see that. If it inhibit autophagy, then there are some scientific evidence.
Okay, thank you.
All right. So if there are no more questions, let us thank Dr. Kaur one more time.
Thank you so much, sir, for the enlightening presentation. Now I would like to invite Dr. Bundle to please felicitate our speaker as a token for appreciation.
So our next speaker is Dr. Pankaj Sharma from NCCS Pune. Yeah, please come.
Okay, am I audible? Yeah.
Good evening, everyone. It's actually a pleasure to be back at CDRI after 8 years. I see my mentors sitting over there, and I have to say that I'm actually overwhelmed. So I did my PhD from CDRI in 2017, and after that I went for postdoc at Boston Children's Hospital. And I recently joined NCCS Pune, and I'm starting my lab. But today I'm going to talk about some of my postdoc work, and I'm still following up on this. So we had several interesting talks from morning about parasites. So I'm going to shift that lens a bit, and we'll talk about fundamental mechanisms of B cell responses that certainly have implications for vaccine development for many parasitic or infectious diseases. So this is my lab brief overview. So I work on various aspects of B cell biology, and of course infection biology has a major focus there. But in B cells, I'm interested in germinal center responses. So what are germinal centers actually? So these are temporary microanatomical structures in lymph nodes where B cells undergo selection, proliferation, and eventually differentiate into mammary and plasma cells. So upon pathogen encounters, which actually pathogenic antigens which are drained through lymphatic vessels or lymph fluids to lymph nodes, these pathogens or these antigens are encountered by these naive B cells, and depending on their specificity at this stage, they enter in this germinal center reactions where they undergo extensive proliferation. So these are different anatomical locations which are found in these germinal centers. They are called dark zone and light zone, and they are strictly controlled by different cytokines and chemokines at these locations. So in dark zone, these B cells, they are called centroblast, where they undergo somatic hypermutation and extensive proliferation. And after that, these cells migrate to the light zone, where they capture these antigens which are deposited on follicular dendritic cells. And if these B cells are able to bind these antigens here, they get fast survival signals. Otherwise, they will undergo apoptosis. And after phagocytosing these antigens here, they present these antigens on MSC class II molecules to follicular T helper cells. And if they are able to present, or if they can, if there are T cell receptor reactivities on this side, these get another survival signal. So they essentially need these two survival signals for their differentiation into mammary and plasma cells. So if you isolate germinal center B cells from lymph node and stain them for apoptotic marker, you will see most of them are apoptotic, or many of them are apoptotic. So this is a very stringent mechanisms, and my lab is interested in understanding what controls and dictates the selection. How can we actually manipulate B cell responses at this stage so that we can control antibody responses or vaccine responses to various infectious diseases. As we know, of course, all vaccines do not need high affinity antibody. Sometimes we need diversity. How can we actually dictate these responses for informed vaccine design? So my postdoc work was focused on a lipid. It has a very tongue twisting name, globotriaosylceramide, but we abbreviate it as a GB3. And its structure is very simple actually. If you see it, it has a ceramide base, which means a sphingosine base attached to a fatty acid chain, which forms ceramide, and it is attached to only 3 sugar molecules. This is glucose, galactose, and galactose. So when it is attached to glucose, it's called glucosylceramide, glucosylceramide, which has been studied in context to NK T cell because this is displayed on CD1D molecule, and it can prime at least activate some of the NK T cells. And this is lactosylceramide, and when it has 3 sugar molecules, it is called GB3. The structure is very simple, but biologically what is interesting is if we look at its expression on all immune cells, it is highly expressed by germinal center B cells. If we take naive B cells or differentiated B cells, it's not displayed on membrane. So we were interested what is this lipid doing during this transition or during this proliferation stage. So what we did actually, we used loss of gene function approaches. So if you look at metabolic pathways of its synthesis and degradation, it is synthesized by alpha-1,4 galactosyltransferase. It is called A4-GALT, and it is degraded by alpha-galactosidase A. So the mice which lack alpha-galactosidase A, they have higher accumulation of GB3 on germinal center B cells, and mice which lack A4-GALT or GB3 synthetase, they completely lack GB3. So this actually gives us different titration. We have physiological levels of GB3 overexpression setting, and GB3 deficient setting. So using this model system, what we did a classical immunization experiment where we take NP OVA. NP is a heptane. It is adsorbed on, or it is conjugated to ovalbumin, and it is combined with alum to study the immunological responses. And what we see here is when GB3 is highly abundant, we get higher germinal center responses, higher frequencies of germinal center B cells. In contrast, when GB3 is lacking, we have relatively lower germinal center responses. We can hardly see actually or poor organization of germinal center in the splenic tissue or in lymphoid tissue. But more importantly, when we look at the antibody responses, the antibody titers, of course, they somehow match with the germinal center responses. But if we see antibody class switch from IgG or IgM to IgG2C, which is an important isotype, that is completely lacking or is compromised in GB3 deficient mice. We did clonotype analysis to look at molecular level what is their selection or if there is any compromised affinity maturation. So after immunization, we took purified B cells, all B cells, and sequenced B cell receptors. So these are different clones actually shown by different shades. What we did was we picked 3 commonly expressed clones and look at the mutation which guides the which is a high affinity mutation for NP heptanes. So what we see here is, I think it's relatively slow. When GB3 is high or when GB3 is lacking, we lack the affinity maturation, which clearly shows that the GB3, its abundance in germinal center is important for antibody affinity maturation. So the question was, how is a lipid dictating antibody affinity maturation? So coming back to the germinal center, like I explained, these are 2 different anatomical selection sites. We looked at somatic hypermutation that was not compromised when GB3 was lacking. So we hypothesized that GB3 somehow interfere between recycling from dark zone to light zone because the high affinity mutations are selected when they go to this light zone in the selection zone. So to study, of course, dark zone and light zone populations, we used CXCR4, which is a marker expressed by dark zone centroblast, and CD83, which is expressed by light zone B cells. So what we see here is by immunostaining as well as by FACS analysis is that when GB3 is high, we get higher centroblast to centrocytes, or we get higher centrocytes or higher migration of the cells to the light zone. And when GB3 is, of course, lacking, we have first of all lower germinal centers, and these cells are stuck in the dark zone or proliferation zone. So which kind of indicates that GB3 is essential for migration or placing these cells in the T cell area for their efficient selection. So the question was, mechanistically, how is GB3 facilitating it? If we look at B cell receptor signaling, in B cells, BCR signaling is what dictates actually the differentiation throughout, from bone marrow to lymph node and even in periphery or even in recall responses. So what we see here is CD19, which is a co-receptor of B cell receptor, and it has been shown that for their migration, it is the phosphorylation of CD19 and activation of CD19 downstream signaling, which actually degrade FOXO1, and FOXO1 is a transcription factor which induce their proliferation and rest the cells in the dark zone. So what we did actually, we just looked at CD19 downstream signaling, and what we can appreciate here is that the activation of BCR downstream kinases, SYK and LIN, it was not compromised in all these settings. But if we see the phosphorylation of CD19 downstream signaling, it was clearly lacking when GB3 was lacking. So that made us hypothesize that GB3 somehow interfere with CD19 downstream signaling, and it's not directly involved in BCR. And that makes sense also because the proliferation in the dark zone was not compromised in when GB3 was lacking. So how is the lipid actually doing it? If we look at BCR signaling in general in germinal center and non-germinal center B cells, there is transient activation of SYK. BCR or kinases downstream of BCR, they show just transient activation. That means if there is so basically actually SYK phosphorylates CD19, and for this efficient phosphorylation, and SYK phosphorylation it itself is transient, they have to be in close proximity. That was our hypothesis at this point. So we proposed and we checked actually if GB3 somehow facilitates CD19 translocation to IgM micro or signaling nanodomains. For that, we used proximity ligation assay, which is an in situ hybridization assay, where of course two antigens are probed with antibodies conjugated to oligos, and then of course signal is amplified by rolling circle PCR, and then signal can be detected by any DNA binding dye and quantified by FACS or immunofluorescence microscopy. In our case, we did immunofluorescence microscopy, and what we can see here is when GB3 is lacking, we see CD19 association with CD81, and when GB3 is high, we see its association with B cell receptor. That means CD19 is somehow facilitating translocation of CD19 on membrane. So this was our working hypothesis, that when GB3 is high, we see this translocation of CD19 from CD81 resting complex to the activation complex, which leads to amplification of downstream signaling, and which eventually place these cells in the T cell zone for efficient selection. I will not go too much into this data, but we did some of the binding experiment to see whether it is sugar or the lipids what leads to the binding. And of course, sugar is critical. This trisaccharide structure is critical for CD19 binding, but of course lipids length also determines their integration and assembly in the BCR nanodomains. What was more interesting for us was we revisited our B cell receptor or BCR sequencing library to see affinity maturation is one aspect, but antibody diversity is another thing, to see if GB3 somehow is also boosting the overall antibody repertoire. So this is rarefaction analysis. We see what is the clonotype diversity. So when GB3 is overexpressed compared to wild type homeostatic condition, we get higher BCR diversity. And when we checked reactivity of these BCR diversity, is it able to break actually immune dominance, right? That is a problem for vaccine development also, because most of the epitopes or antibodies are generated against dominant epitope. So this actually serves a very nice model where ovalbumin, which is a carrier protein, it serves as immunosubdominant, and NP-heptane it goes dominant. All responses are mounted against it. So if we see reactivities by flow cytometry staining of NP and OVA on germinal center B cells, we get higher OVA reactivity or subdominant epitope reactivities only when GB3 is overexpressed. So similarly, we also looked at follicular T helper cells and T helper cells reactivities. Here also we can see that TFH cells, or these are OT2 cells which are reactive against ovalbumin, they are also amplified when GB3 is highly expressed. So this is actually something that we are working right now, very, very preliminary data. We are exploring GB3 as an adjuvant, right? So this was all the genetic manipulation of GB3 in different settings. But what we are doing is we are incorporating GB3 in different lipid nanoparticle formulation. So this is data from model antigen ovalbumin, but of course we are exploring it for different infectious diseases, cancer, and even malaria. So what we are doing is lipid nanoparticle containing GB3. What we see here, OVA has some of the epitopes which are dominant and some of the epitopes which are cryptic in nature, and we see reactivity for CD4 T cells to get the overall diversity response. And what we see when GB3 is there, we get response not only for dominant epitope but also response for the cryptic epitopes. So this actually with this I've picked GB3 as a potent adjuvant that can boost immunological repertoire or can break the immunological dominance. So this is what I showed today, that when GB3 is high, it translocates CD19 from its resting complex to activation complex, facilitate downstream signaling, leads to degradation of FOXO1, which is critical for its migration to the T cell zone, where it promotes antigen presentation, and both these are critical for boosting antibody affinity and diversity. So we have influenza data already published in our manuscript, where we tested GB3 for cross reactivity using influenza as a model, but it is working very well for other diseases also, including cancer and even malaria. So because I see lots of young students here, I want to pitch a concept that was discovered in 2025 earlier this year, and this is an oil on canvas painting. What it depicts actually is that this is representation of germinal centers actually. This is primary repertoire entering in germinal center reaction and giving rise or giving birth to the specificity. And it is what is highlighted in this manuscript is this is new concept called antibody affinity birth. It shows that no matter what the T cell reactivities are, B cells by using somatic hypermutations can also select antibodies very efficiently against diverse epitopes or cryptic epitope. So there is a need to understand B cell biology in more detail for informed vaccine design when it comes to various infectious diseases. So with this, I stop and acknowledge everyone listed here. So this Florian is my postdoc mentor, and Fred is the one who actually trained me in B cell biology. At NCCS, I have different collaborators. I'm thankful to, I have 2 PhD students and 1 Tech Dr. Mahadev Goran. And NCCS has excellent transgenic animal house facility. So with this, I will stop and will take any question. Thank you. Thanks.
Yeah, maybe here first.
So, what are the other mechanism for the lateral translocation of CD19 to activated B cell?
Yeah, actually, it is especially in germinal center B cells, it's not studied that much. So there was one paper from Michael Rett that showed that this translocation is required. But what are the other mechanisms which flippase and other molecules are involved? That is not known yet. But with GB3 in context to this co-lateral migration, now there is another study coming up that shows that for co-locomotion of CD19 to BCRs, it is GB3 mediated, at least in germinal center B cell. In naive B cells and in differentiated B cells, CD19 doesn't co-localize with BCRs. So it stays, it doesn't go close to BCRs.
Here the lipid and protein interaction is must for this.
Lipid and protein interaction is must, yes.
So nice talk. So what happens if I take on T independent antigen?
Yes, that's interesting question. That was a referee's question for us. Yes, so for T independent antigen, GB3 doesn't play much role. It is germinal center restricted.
Now you get antibody repertoire?
Yes, we get antibody repertoire. That was not different when GB3 was lacking and it was abundant. It doesn't play a role in it.
And in the context of affinity maturation,
yes.
So how actually, because you get antibody response and after that the affinity maturation starts?
Yes.
As a mutation at the antigen binding site. So how this lipid is influencing your affinity maturation?
Yeah, so actually what it does, it increases clonal competition in the light zone, right? Light zone means T cell zone. If there is a strong competition there, that leads to better affinity selection also, right? So what it is doing, it is just shifting these cells from dark zone to light zone, increasing competition for antigen as well as TFH cells there.
All you are trying to say that the number of B cells from one zone to another, that has been restricted.
That has been restricted when GB3 is lacking.
So what is happening to the other B cells? Those are not going to...
They undergo apoptosis. We checked, we fate mapped them, tracked them for their longevity.
Any specific way of explaining this, which clonal population will move, which will not?
Yeah, it is tough to actually track which cells are moving and not. There are 2 photon microscopy that people did to track these cells and their selection in light and dark zone. But it is essentially driven by clonal competition, because when we reconstitute and increase antigen doses, right, or do adaptive transfer of follicular T helper cells, we can rescue it. We can still get affinity maturation even GB3 is lacking. So it is somehow clonal competition in the light zone. That is what GB3 is dictating.
Thank you.
Is there any other question? No. So what is the importance of those 3 sugar moieties there? It binds to the lipid, right?
It is, it is attached to lipid, but it binds to CD19.
Right. So suppose you have 3 galactose or 3 glucose.
Yes.
Then what do you expect? It still
binds but doesn't signal, because it has to be on membrane to translocate it. So we did try sugar as a ligand studies to see if it can still promote CD19 downstream signaling. It doesn't. So it has to be on membrane in BCR signaling nanodomain, lipid raft. That is what translocates CD19 to.
And it has to be glucose, galactose.
Glucose, galactose. Even if we change the bondage from alpha 1,4 to some other bondage, it loses. Actually, I could not explain that slide. We just changed GB3 to IGBP. Only one linkage is missing or is different. We completely lose the signaling. Yes.
Nice. Very nice.
Thank you.
Thank you so much, sir, for the insightful presentation. May I now invite Professor Bhattacharya to kindly felicitate our speaker as a token of appreciation.
Okay, so the next speaker of this session is Dr. Suprabhat Mukherjee. He's an assistant professor at Kazi Nazrul University, and he is working on lymphatic filariasis.
Thank you. So first of all, thanks to the organizer for giving me this opportunity to present at this platform. So till the last speaker, and actually from the beginning of the day, we are listening to the different talks that are actually emphasized on the protozoan parasites, namely malaria, leishmania, and toxoplasma. So I'll be talking on a multicellular parasite, that is the lymphatic filarial parasite. So why? Because lymphatic filariasis, it is a global problem. So you see that currently 657 million cases are still active in the all over different countries in the world, but in India it contributes more around 62% of the total global cases of the lymphatic filariasis. So the currently the three drugs, ivermectin, albendazole and doxycycline, these are having a numerous side effects and they are having limited efficacy. And also in the filaria endemic population, there are so many taboos are there because they don't want to take these drugs, because after taking the drugs they face several different types of side effects. So I joined Kazi Nazrul University in 2018, and with the help of my previous supervisors and also different friends and collaborators from Kolkata and outside West Bengal, so we tried to find out some novel targets by which we can either develop chemotherapeutics or immunotherapeutics. So when I joined this university, so as my background was filariasis, so we wanted to find out what is the prevalence. So initially we got one project from UGC under STRIDE scheme, and we have found that the region in and around Kazi Nazrul University in a radius of 50 kilometers is a highly prevalent for lymphatic filariasis, because it is a coal mine zone and the coal miners are generally underprivileged, and this particular parasite, it affects the underprivileged population, and the prevalence is as much as more than 10%. So the first target that we fixed, we wanted to study the mechanism of immunopathogenesis and how to treat the immunopathogenic mechanisms. So this parasite, it is a vector-borne parasite. So when it comes in the human body system, it interacts with the multiple cells, including the macrophages, monocyte and macrophages. So when the macrophages are interacted with this parasite, mainly the surface proteins or surface antigen, macrophage changes their polarity from M2 to M1. So M1 are highly, we know that these are the pro-inflammatory subtypes. So now these M1 macrophages are related with the number of the immunopathological consequences associated with this disease. But as I said, that this disease, it mainly affects the underprivileged population, and the pharma company, they are not also interested to make drug against this disease. So only a few laboratories in India they are interested working on lymphatic filariasis. So so we wanted to isolate the antigen that is actually involved in that is involved in the M1 macrophage induction. So what we did, we isolated the parasite from the human blood, that is a nocturnal blood, because it comes in the blood only in the night, and from the parasite we isolated the surface layers. It was done through repeated cycles of fish thawing and sonication, and this surface layer, when we added this crude lysate, protein rich lysate, or the ultra filtered fraction, we have found that there was upregulation of TNF alpha, and based on this TNF alpha upregulation, that we set as a biomarker to isolate the pro-inflammatory protein. So this lysate was purified subsequently purify the protein that is most likely to be involved in the M1 macrophage generation. So how did we purify? So this protein, it is a surface protein, and we know the surface proteins are mainly the lipophilic in nature. So we use first the gel filtration chromatography, and after that we use the paraminophenyl phosphorylcholine chromatography, which is it is a specific matrix for purifying the membrane proteins, and we have purified this protein. And when we did the mass spectroscopy analysis, we got the sequence, and when we did the blast analysis, we have found that it is similar to a particular protein that is present on the surface of the C. elegans, because in C. elegans genome has it is a highest degree of similarity with the filarial genome, and most of the antibody that we use in the filarial research are actually developed from C. elegans. And this purified protein, when we added in the raw macrophage culture, it is a macrophage cell line, so there was upregulation of the TNF alpha as well as the other two inflammatory cytokines. And when we develop antibody against this particular protein, we have found that it is located on the surface of the parasite. Okay. And it is a typical surface protein because the bioinformatics analysis suggests that it contains the transmembrane helices. So after that, we started understanding the mechanism by which this protein actually induces inflammation. So these three cytokines, we know these are the product of NF-kappa B signaling. So we studied the NF-kappa B signaling, and herein we have found that NF-kappa B it was found activated, but when we added TLR4 siRNA, because NF-kappa B in most of the cases is signaled by the TLR4, and when we knock down the cell with TLR4 siRNA, the signal was not found. So the question was that whether this particular protein from parasite, so does it bind to TLR4? For that, we label the protein with FITC, and in another set we block the surface TLR4 with anti-TLR4 antibody, and we added this FITC level protein, and we measured the fluorescence. So this fluorescent micrograph is suggesting that this protein is primarily binding to the surface TLR4, and it was supported by again the co-immunoprecipitation. When reciprocal immunoblotting was done by pulling down the complex with either anti-TLR4 or anti-MFP antibody. And finally, we did the surface plasmon analysis. It was done in the RCB. Here you can see that this protein is actually interact physically with TLR4. And lastly, the bioinformatics analysis with the crystal structure of the TLR4 that is found in the PDB, and the model structure of MFP, that is the microfilarial protein, it is docking or it is binding at the extracellular surface. So that means our hypothesis is that the protein may be the particular agent that is actually inducing the TNF alpha response. But in filariasis, another thing is that another peculiarity is that unlike malaria or leishmania, this particular parasite is deposited on the skin first, and the parasite they secrete some protease to enter into the body system of human. So whether this particular protein has any role in activating dendritic cells. So we studied the maturation and activation marker of dendritic cell, and we have found that the maturation marker like CD80, CD86 and the activation marker like CD54 and the co-stimulating molecule CD40 all were upregulated. But and it is not due to LPS. Why? Because another peculiarity of the filarial parasite is that they also contain a bacterium, that is Olbachia, and that also secretes LPS. So the particular lysate we use, we checked with LAL assay for LPS contamination, and there was no LPS contamination. So that is solely due to this particular protein. And so alongside the dendritic cell marker, activation marker, the pro-inflammatory cytokines all were upregulated. So we wanted to study again whether it is TLR4 dependent or not. So we use the classical TLR4 blocker, that is CLI-095, and the signal was gone down. So that again supports that this protein binds to TLR4. So again at the cytokine level, it was also found that if we block the TLR4, then there was no inflammation. So, so the binding of MAP to dendritic TLR4, it was also studied by the other assay that I have already shown for macrophages like immunofluorescence. And so when the parasitic antigen is exposed to the dendritic cell, obviously dendritic cell is an antigen presenting cell, so it will present the antigen to whom to the naive T cell. So what we did, we educated the monocyte derived dendritic cell in vitro, and these antigen educated cells were co-cultured with naive T cell, and we have found that there was expansion of the Th1 and Th1 T cell subset and Th17 subsets. So that means, and also it was also supported by IFN gamma and IL-17A. These are the respective markers of Th1 and Th17 pathways. So taken together, we can say that the protein that we have purified, it most likely to cause the maturation of the dendritic cell and activation of the dendritic cell, and these antigen educated dendritic cell may expand the Th1 and Th17, which are actually related with the filarial immunopathology. So, so this protein appears to be a new and meaningful target for chemotherapy or immunotherapy that I will show. So the question was that, can we target this MAP TLR4 interaction, because either MAP or TLR4 both are actually. So what we did, we searched the literature that are already published by our previous mentors or by other researcher, and natural products were found to have significant antifilarial activity. So, and most of the compounds that have been reported for antifilarial activity either are quinolones or sulfonamide. So one idea came to my mind, and I discussed with a chemist collaborator from Bose Institute, Kolkata, and they eventually gifted me a series of different quinolone fused cyclic sulfonamide, and from this sulfonamide we have found this structure, and we name it 8L because it was the 8th molecule of the L series among 96 compounds, and it is having activity against the adult parasite, the microfilaria, and the oocytes. And the LD50 was very low, 4.8 microgram for oocyte, 46.38 for microfilaria, and 78.16 for the adult parasite. And as I told that filaria also contains a bacterium, and very interestingly, this bacterium, it regulates the reproduction, pathogenicity, and survival within the host of the parasite. So we checked whether this particular compound have any anti-olbachal action or not, and this was done in collaboration with the University of Bonn. And we have interestingly, when we studied this molecule in vitro using human cells or the mouse liver or spleen, after giving the oral administration, we have found that it appears to be a benign molecule. So this particular molecule, that is a quinolone fused cyclic sulfonamide, it can kill the parasite at different developmental stages. But the thing is that we wanted to develop a molecule that can have anti-parasitic action as well as it can have some anti-inflammatory action. For that, again, this molecule was modified for its side chain by the chemist collaborator, and this 3, that is the 3,5-dihydroxyphenyl derivative, it was found highly active in reducing the inflammation caused by the LPS induced TLR4 activation, and it binds at the same place where the LPS bind in the TLR4. So this particular compound, it can be used, or it can be utilized for further studies to present this molecule for antiviral chemotherapy. But as we are working with this protein, so can MAP used for targeted immunotherapy? One interesting observation I want to share with you, that when we develop antibody against this particular protein and we treated the microfilaria with this protein, when we treated the mouse intraperitoneally, and we have found that the level of the cytokines were very less as compared to the live Wucheria bankruptcy parasite. So that signifies that this protein it is having some immunotherapeutic potential. And when again we checked with the infected serum, when we treated the rat with this particular protein, we collected the serum, and using this antibody derived from the serum, when we use, we can see that it can cure the microfilaria in rat model. Alongside, it can also reduce the inflammation. So our ongoing work is going on, and collectively we are working with to see whether this is a prophylactic or preventive kind of agent, and for that we are also working with some in silico analysis by extracting the epitopes, and we are trying to develop a multi-epitope vaccine. So what I was telling, that the MAP, that is the immunoregulatory antigen. So apart from these antigen, there are so many others antigens are there that can also be synthesized in the Uchira bank of T. So if we look at the life cycle, so when the parasite it deposits the larvae, that is the L3, okay, that's the infective stage. So this L3, upon discharge in the human body, it quickly undergoes through a fast molting that is called that is the L4. Say, and this L4 after molting 3, it forms the adult. So now this particular molting stage is very key, because during when this L3 larvae, when it is present in the blood, it takes the steroid hormone from the patient, and this steroid hormone, it binds to the intercellular steroid hormone binding receptor of the parasite, and that causes expression of some genes, and these genes are actually expressing some of the proteins that are involved in the immune evasion strategy. So we wanted to purify this particular protein, and using the C. elegans DAF1 antibody, we have that we have set as a biomarker for isolating the intercellular steroid hormone binding receptor. So using the activity guided purification, we have purified around a 43 kilodalton intercellular steroid hormone binding receptor, and this appears to be the first report where we have characterized this particular protein, and using AlphaFold we have seen such kind of a chicken like appearance of the protein. So currently we are working with this protein, because these proteins is actually likely to be involved in synthesizing many of the genes that are involved in the immunoevasion. So we have developed the antibody. We have found that this is also present throughout the surface of the parasite. So I'll just finish, because recently we have got one project from DBT, because filarial patient they are having a different kind of immunoregulatory status, because in leishmania or malaria you can see they are highly pro-inflammatory, if I am not wrong, but in filaria, during the stage of infection, when the infection occurs, the Th1 level is very high, but in chronic stage it is Th2. Again, when the parasite in dead condition or dying state, then again the Th1 is established, and during the middle time, that means during the immunocompromised state, there is a chance of secondary infection, and actually secondary infections happen and they causes the lymphangitis, and this stage is reversible. But when this lymphangitis again that means the bacterial antigen together with the filarial antigen, they causes the lymphedema. This stage is also reversible, because you can treat the patient with antifilarial drugs, because still the parasite is in the larval condition. But when the parasite matures to the adult, the entire antigenic set or the repertoire is changed, and together with the secondary infection, they form the elephantiasis, that is irreversible. So we wanted to characterize what kind of co-infection is there, and in the 4 districts that are nearest to us, that is Paschim Bardhaman, Birbhum, Bangpur and Purulia in West Bengal, we have found that fungal co-infection is there. And very interestingly, when we perform in the laboratory regularly, that is the diagnosis of the infection through PCR based method, repeatedly we have found these 3 bands that does not belong to filarial parasite. So when we sequence that, we have found that this is belongs to a particular fungal isolate, that is Candida gulenmundi, and we have identified this through the 26S RDNA based amplification, followed by the pathogenicity marker, that is RPS0, and currently we are working with the isolation of the immunoregulatory antigen from this particular fungal pathogen. I wanted to see whether MAP together with the fungal antigen, what kind of immunogenicity is found. And so these are some of the publication that have been that we have generated, and these are the collaborator, because without them it was very impossible to work out the problem staying in the 200 kilometers away from Kolkata. So, and also I am lucky to get funding from different funding agency from India and abroad. And this is our campus. This is Kazi Nanjul University, established in 2022 by the government of West Bengal. So thank you so much.
Thank you, Dr. Mukherjee. So we don't have much time, maybe one or maximum two questions. Okay, let me start with that. So the crystal structure that you shown with TLR4, what was the other counterpart, the ligand?
That is the filial antigen. We have modeled the structure with modeler software, and the crystal structure that is already present in the protein data bank, and we did the molecular document.
Oh, acha. Okay. So it has 3 transmembrane domain, right?
Yes.
But then which domain?
4 transmembrane domain.
4 transmembrane.
Yes,
so which extracellular domain that is actually interacting?
Actually, the extracellular domain of the TLR4 and third transmembrane domain of that particular parasitic protein.
Okay.
Yes, sir. No, sir. The lipoprotein that is synthesized from steroid hormone signaling. I wanted to know what kind of steroid hormone receptor is present within the parasite. That actually responds to human steroid hormone. And when it receives the signal from the human steroid hormone, it synthesizes some of the genes, out of which MAP is one of them. Yes, sir, that we have characterized.
All right. So let us thank Dr. Mukherjee once again.
Thank you.
Our sincere thanks to the speaker for an engaging and enlightening talk. I would now like to call upon Dr. Kalyaneshwar Mandal to felicitate our speaker.
Now let me invite the last speaker of this session, Dr. Mridul Mohan from NIMR, New Delhi, and he will be giving us some ideas on the molecular markers from the drug resistance parasites. You have 6 minutes plus 4 minutes.
Okay. First of all, I want to thank the organizing committee, and I'm very delighted to be here because I was the student of CDRI, and right now I'm presenting my work at CDRI forum. It's a very proud moment for me. And we're coming to the presentation. Title of my presentation is Molecular Analysis of Anti-Malarial Drug Resistance Markers in Nancowry Group of Islands of Andaman and Nicobar Archipelago. So if we look at the site, we have to go into the history. Andaman Nicobar historically endemic for malaria, and in case of if we look at the site of our study, that is near to this place, means the Nancowry Tehsil. So Nicobar Islands first occupied by the first European colonial power was the Danish. So Austrian basically colonized it, but they couldn't succeed to keep it long time. They have to abandon due to malaria. So why we chose this site for our study? So it was the reason because in Andaman there are 3 districts, and Nicobar is the only district where the malaria is endemic. And in Nicobar, Nancowry Tehsil is the only tehsil where is the very high API of malaria. So that's why we choose this. It is a hard to reach island. This Nankauri Tehsil is comprised of 5 different islands of population 10,000, and these particular islands are hard to reach and malaria endemic and geographically isolated. That's why we conducted a study here. We have done the mass blood survey of the 10,000 population, and we dig out the malaria positives, and then we look at the what is the status of anti-malarial markers, molecular markers. So anti-malarial drug resistance markers. First one is we looked at the PF dihydrofolate reductase gene. It is the target of sulfadoxine paramethamine SP partner drug. Basically, the paramethamine is the drug of choice, and PFDHFR is the target of paramethamine. Another gene we looked at the PF plasmodium falciparum dihydrofolate synthase. This gene also involved in the folate biosynthesis, and it is the target of SP drug sulfonamide. We have also looked at the PFMDR1 gene, multi-drug resistant 1 gene, and we looked at only one point mutation, that is N86Y. We also looked at the PF-CALS30, the CALS propeller region of CALS protein, which is the point mutation which are associated with the drug resistance against artemisinin. So looking at if you look here, the point mutation which we have found, it's a very interesting data. We have found around N51I 93.48%, C59R 91.3%, S108 N is 100% mutant, and I164L, isoleucine 164L leucine 97.83%. Parasite population, collected parasites were found mutant. We're looking at the DHPS point mutations. Then S436A 57.5%, A437G is a very crucial one. The resistance against pyrimethamine basically conferred by this particular point mutation. And if we look at the another point mutation which also potentiate the drug resistance against pyrimethamine, that is K540E, which is equally means the 92.5% parasites having these two point mutation together. Then the A581G we found around 37.5% parasite isolates are mutant for that particular point mutation. Then we analyze the haplotype, and in haplotype analysis, you can see we have not found a single isolate which has the wild type haplotype. We have found double mutant. Here you can see around 2.1%, because RN and were the two types of haplotypes of double mutant we found. RN in this case for 2.7%, while in case of ANCNL, so for this around 4.3%. Triple mutants 4.34%, but the quadruple mutant of DHFR, it's a very interesting finding. We have found 89.13% of the parasite isolates which are having quadruple mutation, and that actually tells us if the parasite has the quadruple mutation, then it means it is resistance, it has the SP drug failure in that case. Similarly, we have conducted the haplotype analysis for dihydrofolate synthase gene, and in that case you can find, if we look at, because shortage of time, I'll summarize it, approximate 87.5% of the parasite isolates are having triple mutant. Then we have conducted, we have analyzed the data with the haplotype analysis in combination for DHFR as well as DHPS, and we have found almost, if we look at the data, here you can see The septuple mutation we found is the 3144. Around 80% of the parasite isolates are having the septuple mutation if we look at the these two genes in combination together. For MDR1 gene, we have looked at the N86Y, which confers the mutation against the quinoline-based drugs like lumefantrine and all. So almost 50% of the parasite population have this particular plasmodium falciparum isolates having the mutation against this particular point mutation. Coming to the takeaway, as I said, we have found a very high frequency of quadruple mutation in PF-DHFR gene as well as triple mutation in PF-DHPS gene in the majority of the plasmodium falciparum isolates received from the Andaman Nicobar Islands. Data seems, based on the data, we can say the mutation has been fixed in that particular population locally, and we have not found, fortunately, we have not found any mutation in the PF-Cal13 gene, which is a good thing. And this very high prevalence of DHFR and DHPS in combination mutant suggests there is a compromisation of SP drug, and it actually warrants the therapeutic efficacy study and potential policy review in that particular region. But there is a logistic challenge. As I said, these islands are very hard to reach, and conducting such clinical trial therapeutic efficacy studies are not feasible in these islands. So therefore, we are proposing molecular surveillance can serve as a valuable tool for guiding drug policy decisions. These are Priyanka, Kanika are my students. Dr. Bharti is a colleague and collaborator. I would thank Dr. N. Sriram, Dr. Manjurai from Vector Control Research Institute, Dr. I.P. Sunish for the providing logistic support during the sample collection, and Director Health Services Sri Vijayapuram for the other logistic support, and ICMR for funding support. Thank you very much.
Interesting finding. We have time for a couple of questions.
Thank you, Dr. Mohan, for your presentation. I have one question regarding your conclusion about the molecular surveillance. First of all, I would like to know the epidemiology of malaria in this archipelago. Is there any PF in this study site, or PV is also there?
Yeah, very interesting. Question. Yeah, there is a PF as well as PV, but almost 60% of the cases are the PF, and the 40% of the cases were PV.
Okay, because my concern is about the molecular part. For PV, we just have a putative molecular marker just for PV, but for PF it's already validated. But how we can do for PV?
Yeah, but as far as my knowledge is concerned, for PV the drug distance is not as problematic as for the PF. So it is the one question, and the if we look at the antimalarial treatment for PV, it is so far we have not found any drug failure in India.
Okay, maybe Krishanpal.
Okay, very nice talk. So my question is, do you have clinical information of these patients? Because that's where it will make much more sense to say we do not have any Kelch mutation. See, unless we have association, can we say this is really important or not? Do you have any information about drugs not working that much?
Yeah, your question is quite interesting, but yeah, we have not found because we have not found any drug failure as of now, because most of the, if we look at the ACT, artemisinin based combination therapy, most of the parasite burden has to be cleared by the artemisinin or the artesunate or any derivatives of artemisinin. The partner drug actually has to remaining residual actually eliminate the residual malarial burden. But if the data is what the data is telling us, if we have not revised the drug policy change as of now, because for that you are saying we have to do the therapeutic efficacy trial, and which I am telling you because I went there.
No, I'm not saying trial. You can just have the means clinical information, whether they are cured by 2 doses, 3 doses, those kind of things.
We tried, but see, the population is tribal, and keep tracking of them. It is not
Okay.
as easy. Yeah.
Another thing, just a comment. When you say 0 wild type, do you think it can be a problem of the referencing also? Like we are looking at the genome, which is not really native.
Yeah, so I'm talking about those particular point mutations.
Yeah, means it can be because we have the wild type. Means that whatever sequence we think is not there in this population. It's not wild type for us.
Yeah. So that particular, which I'm trying to tell, is that particular wild type, or wild type. We can say wild type of wild type sequence are not present there.
Okay, thanks.
Okay. Any other questions? So I have one question for you. What I found very interesting was that your quadruple mutant frequencies are much higher than the single, double, and triple.
Yes, sir.
Right. So if, let's say, for single mutant, if the frequency is 10 to the minus 6, then the quadruple it will be 1 in 10 to like 10 to the minus 24, right? But you are finding that frequency is much higher than the single. So how do you explain this?
Sir, if we look at the how many of the parasite, I mean, if the parasite population, suppose parasite isolates were the 100, so out of 100, how many were the quadruple mutant were having those particular mutations in together? So we found out of the 100, around 89 or 87% of the parasites isolates were having those particular mutations.
Now, do you think they acquire this mutation sequentially?
Yeah, they acquire sequentially. And there is, I said, isolation factor can also be work because due to the island, so isolation factor can also amplify this.
Okay. Any reason that you did not check for PFCRT mutation, the chloroquine mutation there?
Yeah, sir, it's also very interesting question. Actually, initially plan was not for even for MDR1. We were looked at the SP, means the DHFR, DHPS, as well as the PFCAL13 only. So the amount of DNA, because the DBS has to shift from that to NIMR, it took a lot of time. Sometimes the quality of DBS was not good. The isolation of DNA was not enough to perform the experiment for PFCRT.
You already asked one question.
Yes, but I have not finished my question.
Yeah. No, you can continue with him later on because we need to really wind up this session. So thank to all the speakers of this session. So now we conclude this particular session.
Thank you so much, sir, for a wonderful presentation. Now I would like to invite Professor Bhattacharya to felicitate our speaker as a token of appreciation. We extend our gratitude to the chairpersons for their valuable time and support in conducting this session. Now I would like to invite Dr. Anuradha Dubey to felicitate our esteemed chairpersons. Very well. Our next session of the Flash Talk series will begin shortly. Thank you everyone for your presence and participation. Good evening everyone.
Welcome to the Flash Talk session of 33rd NCP. It is our privilege to have among us our esteemed judges. We extend a warm welcome to all our judges and the participant present with us today. As you know, the Flash Talk format is designed to challenge the speaker to present their work clearly, concisely. Each speaker will have a total of 5 minutes. A reminder buzzer will sound at 4 minutes and the final buzzer at 5 minutes. And also there will be no question and answer for this session. So we request all the 10 participants to please conclude your talk within time limit. We may now call upon the first participant, Ms. Disha Shah.
Do I start? Okay, yeah. Good evening, everyone. I am Disha. I am a PhD student working with Dr. Krishan Pal at CSIR Pune. And I'll be talking to you today about the role of H3K36 methylation and associated writer enzymes in Plasmodium falciparum. Okay. Yeah. Okay. Right. So now when you look at the malarial parasite Plasmodium falciparum, you see that there is very tight transcriptional control across the different stages. And there are certain peculiarities for this parasite, such as an unusually AT-rich genome and a relative scarcity of specific transcription factors. And this has led to researchers in the field exploring epigenetic mechanisms of regulation. Now, we see that there is a dynamic and state-specific distribution of histone marks also across the Plasmodium life cycle. And epigenetic control has been implicated in survival as well as virulence in the parasite. So now if you look at the histone modification landscape of Plasmodium falciparum, activating marks are present in abundance and they are present across the genome. However, if you look at the repressive marks, this is not the case. So the heterochromatin is restricted spatially and also it houses only a very small fraction of all the genes, and these are clonally variant multi-copy genes or virulence genes and are enriched in marks such as H3K9 trimethylation, H3K36 trimethylation. But if you look at the rest of these loci and the rest of these genes, which is the major bulk of the genes, it is unclear how dynamic transcriptional regulation is happening here. So, but what we think is that based on ChIP-seq analysis and RNA sequencing analysis, is that the amounts of H3K36 dimethylation and relative activating marks decides the transcriptional status at these loci. Now, to answer the question, is K36 dimethyl a global repressive mark in falciparum? We looked at the histone lysine methyl transferases in Plasmodium. Now, these are histone lysine methyl transferases typically belong to two families, which are set domain families or .1 domain. And these can either be processive or distributive in their mechanism. Now, Plasmodium HKMTs are a group of 11 enzymes, but product specificities for many of these are undefined and there are conflicting reports. Now, knockout of the predicted K36 trimethyl writer, set 2, it increases K36 ME3 at these virulence loci, causes an upregulation of these genes, but does nothing to K36 dimethyl levels. So the dimethyl writer in Plasmodium is unknown. Hence, we decided to screen the PF-set proteins for K36 methylation activity. Now, we purified these proteins from E. coli and performed histone methyltransferase assays and detected activity using byproduct substrate detection, histone PTM specific antibodies, as well as mass spec. Could not detect K36 methylation activity, however, and we think that the bacteria purified enzymes are either inactive or weakly active. Now, this was a setback, but we made a comeback. Now, we looked at histone methyltransferase in Cerevisiae, where SET2 performs mono, di, and trimethylation. And in a background where you have SET2 delta, which will have no K36, PF-SET6 was actually able to rescue K36 methylation, and specifically mono and dimethylation. Now, this encourages us to then look at PF-SET6 in our system of interest, that's Plasmodium. And this is a 55 kilodalton protein with a split set domain. And these proteins are typically K4 methyltransferases, but sometimes also have K36 methyltransferase. We see that overexpressing this protein results in increased K36 dimethylation levels in the parasite, leads to a higher growth rate, and upregulation of invasion-related genes and downregulation of virulence genes. So to summarize, the bacterially expressed PF-sets were either weakly active or inactive, but the yeast complementation assays revealed that PF-set6 might be a potential K36 mono and dimethyl writer. And we also see that K36 methylation activity in Plasmodium as well. Yeah. Thank you.
Thank you. I request all the flash talk participants to please line up near the backstage. Moving on, our next participant is Ms. Himadri Shukla.
Good evening, everyone. I'm Himadri Shukla. I'm here to giving my flash talk, which entitled Understanding the Role of Tyrosine Kinase-like Protein in the Plasmodium burgeri. So Plasmodium has a complex digenetic life cycle which comprises stage-specific transmission. So during this transmission, protein kinases play a vital role in the signal transduction and many cellular processes. So here we target the unique protein kinase, which is tyrosine kinase-like protein 3. So in silico analysis revealed that TKL3 is a evolutionary conserved in among epicomplexan species, and it has a complex structure, and it having a specific domain organization which is SAM domain, which is known for sexual stage differentiation in the model organism. To check the expression of the tyrosine kinase-like protein in the Plasmodium life cycle stages, we generated a transgenic parasite line, and we checked the expression via the Western blotting, and we checked the co-localization study with the anti-HSP antibody mCherry expressing co-localizes with the cytosolic marker, which confirms TKL3 expression localizes with the cytosol. To demonstrate the role of TKL3 in Plasmodium life cycle stages, we generated a knockout parasite line by the double cross of homologous recombination strategy to check the effect of TKL3 knockout parasite line in the intraheterocity development, and these results confirms that lack of TKL3 has an impact of the fitness of the parasite in the blood on the blood stages. After that, to assess the effect of TKL3 disruption in the mosquito stage transmission, and we observe severely reduced number of oocysts inside the midgut and markedly reduced number of size of the oocyst. Taken together, we observe very fewer number of midgut sporozoites. So, moreover, majority of the oocyst within the small size produced reduced sporogony and resulted in a negligible sporozoite inside the salivary gland. So to examine the TKL3 phenotype was due to the defect at the ukinete level. That's why we performed time course analysis. So in the time course analysis, wild type GFP ukinete differentiate into stage 1 to mature ukinete. But in the case of TKL3 knockout parasite, it arrested from the stage 1 to stage 3, which is confirmed by the mature ukinete evaluation. So to block at the ukinete level, we was resulting from the defective gamete. That's why we performed genetic cross experiment with the wild type M-cherry and male and female deficient line. So when we crosses the TKL3 knockout with the wild type M-cherry and female defective line, where male is fertile, the parasite was recovered, which confirms that TKL3 is essential for the male gamete fertility. To further check the sexual stage development of the parasite in the knockout and wild type, we got normal development of the activated male gamete and activated female gamete. But when we evaluated the zygote number, we found significantly reduced number of zygote in the knockout. So further, we detailed analysis of the free microgametes after X flagellation, and we check the nucleus aggregation of the microgametes nucleus aggregation of the microgametes, and we found that majority of the TKL3 knockout microgametes were DAPI negative or host negative. That's why this is the reason for the which is further confirmed by the IFA using antitubular antibody. Here in the case of TKL3 knockout, the host was absent in the knockout microgamete. Whether the DNA intensity is the similar as compared to the wild type in male gamete and zygote. To further check the sporozoite infectivity in the TKL3 knockout in the C57BL6 mice, we observed 2 to 3 days delay in the prepatent period, which is further confirmed by the blood growth analysis. To find out the reason behind this. To find out the reason behind this delay, we perform in vitro analysis of the TKL3 knockout, and there is no liver stage development was observed, which confirms that the delay in the prepatent period was due to the slower blood growth rate propagation. So in conclusion, we conclude that in the case of TKL3 knockout, after the 12 to 15 minute post activation, when the microgametes was released from the basal body, in the case of knockout, the majority of the microgametes are without nuclei. So it failed to fertilize the macrogametes and hampered the overall development process. But in contrast, the wild type parasite developed normally throughout the stage specific manner. Thank you.
Thank you. Our next participant is Ms. Parul Mehra.
ये माइक निकाली भैया, ऐसे से हाथ हो रहा है, कर्सर भी ऐसे से हो रहा है,
चल रहा है, बोलिए।
Previous slide. Good evening, everyone. My name is Parul Mehra and I'm from IIT Mandi. And today I'm very excited to share with you the story of novel PA1-like protein homologue from Taenia solium, a molecule that quietly reshapes the way we look at host-parasite interactions. So over the next few slides, I'll be walking you through how we discovered this molecule, what it does inside the cells, and how it links metabolic suppression to immune modulation. Before zooming into the molecule, let us first look at the scale of the problem that we are dealing with. Taenia solium is not just a parasite, it's a menace. What you're looking at on the screen is the larval and the adult stage of the parasite. And this is the entire clinical spectrum of the disease. Now, what happens is when these larval cysts get lodged into different parts of the body, it causes cysticercosis, including neurocysticercosis in the brain, subcutaneous cysticercosis in the lymph nodules, and ocular cysticercosis in the eye. And these are the MRI scans of the brains of the people suffering from NCC, wherein you see that the cysts are lodged in different parts of the brain. And this also sometimes gives us the multicyst starry sky appearance. Now, neurocysticercosis is the leading cause of acquired epilepsy worldwide, affecting millions of people across Africa, Latin America, and India, South Asia mainly. And this disease has a highly variable clinical manifestation ranging from mild seizures to severe epilepsy. And that depends upon both the host and the parasite factors, including the socioeconomic status, the host immunity, and the number and the position of cysticerci as well.
However, neurocysticercosis is a neglected tropical disease, and the molecular toolkit with which it manipulates the host immune system largely is unknown. And it is in this unknown that our work exactly begins. But the most fascinating thing about this organism is its magical secretome. And we do have a few previous reports from our lab in which we try to decipher what is in the magical secretome of this organism. So in the first report, we saw that there are clusters of immune modulatory molecules, which some of them tend to have an anti-inflammatory potential, while others have a pro-inflammatory nature. And in the second report, we showed that how the exosomes of Taenia solium cysticerci are interfering with the PI3K AKT mTOR axis, thereby interacting with the macrophages and acting as strong immunomodulators. And it is in this secretome that we also found our PA1-like protein molecule. However, to verify it as a standalone protein, we first needed to purify it. And after we had enough protein in our hand for further downstream biochemical and functional assays, we decided to check if it is even going inside the cells or not. And what we found was strikingly surprising, that the protein was getting uniformly localized within the cytosol of macrophages. So this data suggested us that PA1-like protein is a bona fide parasite expressible protein which is actively internalized, and it is setting its stage for downstream metabolic pathways. But what is it actually doing was still unknown to us. And here is where the most interesting part of the story lies. We found a striking reduction in the glucose of the macrophages. Our fluorescence microscopy images reinforce the flow cytometric results, wherein we see lesser fluorescence. Here, we see lesser fluorescence in the PA1 treated macrophages in comparison to the treated macrophages. Now, since we are talking about the interference in the glucose metabolism, we also did a docking analysis to check the binding interface between the GLUT1 and PA1 molecule, and we found that it was pretty stable. Our results were validated by GLUT1 and GLUT3 molecules, which are the glucose transporters in macrophages. And it is not just the glucose metabolism that is being interfered with. This PA1 molecule is actually deleting mTORC1, and downstream of mTORC1, when we found an impairment in the autophagic flux as well. But this is happening non-canonically. So overall, PA1-like molecule is pushing the macrophage into a state of metabolic deprivation, acting as a strong immunomodulator. So overall, I'd like to end my slides by saying something close to home, that at IIT Mandi we say mountains shape the size, and this work is exactly that. How it is related to the bigger picture, how it fits in there, the cellular rewiring is the rest of the story, which awaits you at post number 68. You're all welcome. I'd be very happy to interact with you. Thank you.
Our next participant is Dr. Shrikant Nema.
Good evening, everyone. So this is my work. This is a part of a project which I have done in NIMR. So it is all about molecular surveillance of Plasmodium falciparum, histidine-rich protein 2 and 3 gene deletion in high anemic states of India. This study was conducted in 2023 and 24. So as we all know that malaria is one of the most pressing public health challenges, and if we look at the fact and figures, in 2023 there are around 263 million global cases with around 6 lakh deaths, which is quite high, which is 11 million rise from 2022, and 94% cases and 94% deaths occurring mostly in Africa. And if we talk about India, in the same year, this was reported of around 2 million cases and around 3,500 deaths, which reflects nearly 70% decline since 2007. If you look into the bar graph, there's a massive decline from 2027 to 2022. So how do we track these cases? How do we diagnose? Mostly in the field setting, there are two major tools, which is microscopy and RDT kits. So microscopy has its own limitations. It needs expertise. So national program recommends that if microscopy is not feasible, we can go to the RDT. But RDT has its own problem, like false negative, false positive. False negative is due to the gene deletion, and false positive comes due to persistence of HRP2 protein in the blood after the parasite get clear. And WHO also recommend that the continuous monitoring should be done for the gene deletion, and if the deletion level reaches up to the 5%, then we need to change the strategy. So our study is to assess the extent of these deletion across 9 malaria endemic regions in India. So sorry. So these are the 12 sites in 9 states. We have used 2 approaches. One is the pragmatic consideration, and another one is the geostatistical modeling. So modeling give us the 4 sites, and we already had established sites and resources and through pragmatic considerations. So we have chosen these 12 sites around, in the 9 states, and these 3 colors highlights the endemicity level of the malaria in India. So we have enrolled around 10,290 patients in all these 9 states, and 10,254 were we have RDT and microscopy results, and out of that, microscopy confirmed P. falciparum monoinfection was 1,025, and out of this 1,025, we had only 19 RDT negative and HRP2 gene deletion, confirmed HRP2 3 gene deletions. So this is the data of these 19 samples. Yeah, and we have like different transmission pattern, and among these 19, only 1 was found complete deletion. Other were partial deleted. So in these 9 states, we have Longtaling Mizoram, which is reported higher deletion, which is 5.1%, and South Tripura 4.6%, and West Singhbhumi Jharkhand was 3.2%, and in Telangana 0.96%. And other 8 sites have no deletions. We have also done the meta-analysis in India. So, and using a different terminology in a PubMed, we have found some 130 papers, and out of the 130 papers, we have only 8 papers which were included in the meta-analysis. So that data suggests that in Odisha, Chhattisgarh, and Madhya Pradesh, we have over the period of time, the deletion was 0, and in Jharkhand, Mizoram, this was quite high in the last 6 years. So in overall finding, we say that Mizoram have reported progressively increased deletion over the past year, and 0% in 2019, and this study observed 5.1%. So there's a need of continuous monitoring of HRP2 gene deletions, and one of the key limitations, we can say, the timing of sample collections that potentially influenced our findings, and regular monitoring is must. And but despite these low deletions, there is a need to search the alternative biomarker, because as we are approaching the elimination goal by 2030, so we should have some backup plan. I would like to thank all the collaborators and funding supports from the Bill and Melinda Gates Foundations. Thank you.
Our next participant is Mr. Nirdosh.
Thank you so much, organizer, for giving me this opportunity. So as we all know that the cardiolipin, which is the integral part of the inner membrane complex of the mitochondria, and the mitochondria is the part of the every eukaryotic cell, and we also know that it also acts as a powerhouse of the cell. So this cardiolipin is synthesized by the cardiolipin synthase. So we want to check its phenotypic characterization and its expression. So that we performed the IFS study, in which we found that its expression is in the blood stage and the liver stage, and co-localizes with the mitochondria with the PCC's value of 0.787. So for the phenotypic characterization, we perform the gene deletion by the double homologous crossover recombination, and simultaneously perform the complementation for the gene specificity of the phenotype. And after the successful transaction, we initiated the cycle by the administration of the 0.5% parasitemia blood, and observed the slides by the gene sustained blood smear, and we found that the knockout parasite has significant reduced blood stage propagation rate, and the gametocytemia was also reduced. So whether this phenotype is correlated with the mitochondrial function, we performed the mitochondrial membrane potential assay, and we found that the knockout parasite has drastically reduced membrane potential in comparison to the wild type and this is the reason why the knockout parasites are living longer than the wild type parasite, the wild type parasites are dying in between the 7 to 10 days in comparison to the knockout, which are living longer till the 22 days. Next, after the successful, we performed the mosquito stage transmission, where we observed that the knockout parasites are comparatively able to grow in the mosquito stage propagation of the parasite. Then we performed the pre-patent period analysis by the inoculation of 5,000 sporozoite into the BL6 mice and performed the Giemsa stain to check the first appearance in the blood stage propagation. So the pre-patent period was drastically, significantly reduced in the case of the knockout parasite. Here we can say by the 3 to 4 day delay. To find out the delay, the reason behind this delay, we performed the in vitro stage analysis, and we found that both the knockout and wild type parasites were able to successfully invade the HB2 cells, and the knockout inside and outside assay invasion study also reveals its normal functionality, and the early and mid stage liver stage propagation of the parasite and development of the PVM membrane was also normal. But surprisingly, we found that all of the cardiolipin synthase knockout parasite arrest at the cytomere stage, and their nuclei number also reduce. Next, to check and in comparison to the wild type, which grow and mature in the 65 hours, while they arrest at the 72 hours in the cytomere stage only. So next we perform the merosome assay, and we found that there is no detached cell in the supernatant or in the medium of the merosome assay medium. So we collected the all supernatant and injected into the cells mice to perform the merosome assay. So surprisingly, found that CLS knockout parasites were not able to develop the blood stage infection, and there is the failure behind this is due to the so the failure behind this delay, there is failure in the prepatency was because of the mitochondrial branching and apiculous biogenesis and endoplasmic reticulum. The branching and apiculous organelle biogenesis was drastically affected in the knockout parasite in comparison to the wild type, and the PVM membrane was intact, which correlates with our previous study because most of the parasites are arrested at the liver stage, cytomere stage. That's why the immaturation leads to the intact PVM membrane. So in conclusion, we found that the cardiolipin has several stage specific phenotype. So firstly, it has a decreased blood stage propagation of the parasite because of the low oxygen consumption rate. Data was not shown here. The mitochondrial membrane potentiality also reduced, which is resulting in the ablation of the branching of the mitochondria, and this leads to this defect in the organelle biogenesis leads to the immature merosome formation. In counterpart, the host lives longer and shows no patency. These are my publication. Thank you.
Our next participant is Dr. Pankaj Mosale.
Hello. Good evening, all. Today I'll be presenting mitochondrial genome variability and potential drug resistance related to Theileria annulata from Indian cattle. So here, bovine theileriosis is nothing but the tick-borne protozoal parasitic disease, which is caused by the parasite of Theileria annulata, and which shows a prevalence around 30 to 40% in India. On the right hand side, we can see here, this is the global distribution of this parasite caused through the different vectors involved in the transmission of this disease. In the middle, we can see the life cycle. It requires 2 hosts to perform the life cycle, and globally around like 250 million cattle are at risk contracting this parasite infection. Now moving on to this particular cytochrome B protein, and this is the important gene, like in considering the malaria, looking at the autocoin resistance that targeting the cytochrome B gene. There are previously reported studies, proven studies, which shows a resistance in autocoin in malaria parasite. Like that, I'm looking after the this Theileria parasite, looking at the bupropion drug resistance. Here we can see these are the studies which are previously reported to cause buprenorphine resistance across different countries. Till today, from India, there are only 2 reports. One was from Nehra et al. And another from my study. So we reported these mutations causing having a good relation, having the buprenorphine drug resistance in case of Theileria parasite. So buprenorphine is nothing but the hydroxynaphthquinone, which is a structural analog of autocon. So there is a clear linkage considering the malaria as a parasite of interest. Today I am presenting one of the case study. While doing so, during my PhD thesis, I came across a set of a sample from a set of sample collected from same farmer. So there were like 5 samples collected from same farmer, and here we can see first round of sampling was done in November 21, and after like 6 to 8 months, we repeated the sampling. Based on the analysis, we came to know that there are multiple mutations observed in the cytochrome B gene of this parasite. So in second round of sampling, we observed the same pattern of mutations. I'll briefly explain about these things in the next slide. So here we can see these are the 5 cattle samples here, and as compared with the reference strain of Ankara, which is a worldwide strain, and this is the Indian isolate. While comparing these are the mutations which we observed across the profile of the cytochrome gene from the Theileria parasite. So in order to look, we have done performed the repeat sampling, what exactly the scenario. So in the repeat sampling also we observed the same pattern of the mutations in the cattle. So here this is round 1, round 2, and here we can see the round 1 and round 2 mutations observed similar or conserved in the both round of sampling. While in case of cattle 3, the cattle was observed to be died, and in case of cattle 5, the mutation which was not present previously, later we observed in the second round of sampling, we observed the same pattern, these cryptic mutations we observed. While during my PhD, I looked at 3 important genes of Theileria parasite, that is cytochrome B, deoxyribonucleic PIN1. The other 2 genes were behaved normally, but in case of cytochrome B, why these mutations are there? So in order to look further, what exactly the scenario? So we tried to look for the complete mitochondria of this particular parasite. So we targeted these 2 approach. One is the complete whole genome sequencing, and the other approach was multiple fragments from this mitochondria. So we attempted and we succeeded in this particular mitochondrial genome sequencing, and we came to know that these cryptic mutations, variations, not only restricted to the cytochrome B, but also it is present in the other genes also, like the COX1 and COX3, which is a part of mitochondria. As like I said, like cytochrome B is a part of mitochondria. So what exactly the scenario? So for that purpose, we have done this particular study, and we came to know that more than 5% variability at the nucleotide level observed in the complete mitochondria. Looking at this hypergenetic variation, this confirms that the variation is not confined to cytochrome B. And another ongoing study we are performing with the Tamil Nadu. So we are observing same set of cryptic behavior in those samples as well, considering the mitochondria. So the phylogeny of this mitochondrial genome variation and its prevalence needs to be further investigated. Thank you. Thank you.
The next participant is Ms. Ritika Rajput.
A very pleasant evening to one and all. I am Ritika from NIAB. Today I am going to talk about an interesting parasite, Theileria annulata, which is not only known for its infecting cattle, but it does manipulation in host also. That's why I gave the title Masters of Manipulation, Unmasking the Theileria secretory proteins in host cell transformation. Before going into work, I would like to give a quick introduction about Theileria annulata, which belongs to phylum Apicomplexan, genus Theileria, and species Annulata, which is transmitted by tick species of Hyaloma and causes bovine tropical theileriosis, which shows symptoms like enlargement of lymph nodes, lacrimination, anemia, and untreated animals will die within 3 to 4 weeks. The T. annulata mainly infects B cell, dendritic cell, and macrophages in host, It directly infects cattle, but it indirectly impacts human due to economic losses in livestock sector. The only available drug is Parvacurine and only available vaccine is Rakshavac T vaccine. Coming to the life cycle of Thilarian lata, I would like to draw your attention to this stage where the parasite infects host leukocytes and hijacks the host cell machinery, drives uncontrolled proliferation and creates a cancer-like phenotype. This stage is called microcyzont, and we are culturing this stage in our lab named as ANA 2014 cell. This uniquely thing indicates that these parasites encodes many key proteins which may have a role in host cell manipulation. In my work, I am trying to find that proteins. So to search of that, I began with in silico analysis, and I retrieved the sequences from Thilarian lata proteome, removes the duplicate, then predict the subcellular localization and GPI anchor and signal sequence. Whatever protein we got, we removed the sequences by checking the homologous to Bosterus, that is host, and after that we checked the orthologous to T. parva, that is another Thilaria species, transforming species. Whatever protein we got, we characterized into hypothetical and putative. My work mainly focusing on the hypothetical protein. So today I am going to present one of that hypothetical protein, that is TA11900, which is having a faint domain which is uniquely present in a Thiliaria species. So I started with cloning that gene into a PET28A vector using NCO1 and XH to one side. I expressed and purified by nickel NTA beads by using affinitochromatography and detected in Western using anti-stack antibody. With that protein, I raised the polyclonal antibodies in biopsy female mice. Following this protocol, after 6 booster doses is collected the separated sample, we tested the antibody titer using ELISA, and we found a robust antibody production. Also, we have checked and detect the target protein in Western blot in ANA cell lysate, which is infected cell line. After that, we wanted to check the subcellular localization. So we did the IFA, and we found that our protein is localizes in the parasite surface as well as in the host cytoplasm. Pre-immune serum and uninfected cells we have taken as a control. After we have known that our protein is secreted out, we wanted to check the interacting partner in host. Then for that we did the pull down assay with my protein followed by mass spectrometry analysis, and we found some protein which is uniquely present in elution. But one protein draws our attention, that is PPIA, which is already having a well-known role in cancer cell progression. So we to validate this interaction, we did the East2 hybrid analysis, and for that we cloned our thilarial protein in the bait vector and the host protein in the prey vector, and co-transformed into Y2H gold strain, and found growths found in different selective media. Blue colonies represent that strong interaction between these two proteins. Followed that, we did the serial diluted spot assay also, and we found the strong and direct interaction between these two proteins. P53 and LAM we have taken as a positive and negative control respectively. From these data, we can say that our protein TA11900 interacts with host PPIA cyclophilin, which is also known as cyclophilin A. So I concluded my work that I have found one protein with in silico analysis. We cloned, expressed and purified that protein. Polyclonal antibodies were generated, and then we checked the subcellular localization. PT-BPIA protein as a interacting partner we have found through pull down assay, and then after that we validate this interaction with these two hybrid analysis. In future we will do the functional characterization assay. With that, I acknowledge all this. Thank you so much.
The next participant is Mr. Yangala Sudhir Babu.
Respected dignitaries, very good evening to everyone. Currently, I am working on alpha-synuclein and its mediated neurodegeneration during Toxoplasma gondii infection. Coming to the background of my study, T. gondii is an intracellular protozoan parasite, causes toxoplasmosis by showing serious neuropsychiatric symptoms of schizophrenia, paranoia, along with the common symptoms of headache, fever, and fatigue.
and swollen lymph nodes. Coming to the neurodegenerative disorders, these are the major threats for causing defective central nervous system. There are various factors are influencing for causing these NDDs. Parasitic infections are also one of the important factors for causing NDDs. Alpha-synuclein is a neuronal protein present abundantly in the central nervous system. There are various factors involved in it for dysregulation of this protein. According to previous report, there is a report that alpha-synuclein induces neurodegeneration due to dysregulation caused by the infectious agents and a small fold change in its expression leading to wide range of neurodegenerative phenotype. We reviewed literature and understand that there are no studies related to the alpha-synuclein dysregulation caused by the infection with the apicomplex parasites. In this study, we are going to investigate the role of alpha-synuclein upon dysregulation during Toxoplasma gondii infection. These are the two objectives we framed for these studies. First one is analysis of alpha-synuclein expression levels upon T. gondii infection in vitro, and second one is overexpression analysis of synuclein during T. gondii infection and elucidating the effect of alterations in synuclein is along with its associated gene expression profiles. Apart from alpha-synuclein, according to literature, we selected few genes due to their role in neurodegenerative pathogenesis along with the alpha-synuclein. They are of GTP cyclohydrylase 1, C2N1, STAT3, PARK2, etc. This is the experimental methodology we followed for this study. We selected two cell lines. First one is vero cell line for maintaining T. gondii tachyzoids, and second one is mouse hippocampus, that is HT22, as a best model for neurological studies. Here, after getting appropriate cell confluency, we detached the cells followed by the counting. Then we seeded into appropriate cell density in two different types of culture plates. First one is 6-well and second one is 24-well plate for RNA isolation, cDNA profiles synthesis and expression profiles analysis, as well as immunofluorescence studies. Here we can see the swimming of parasites after 4 days of infection. These are the results we obtained for this study. Here, in earlier infection under lower infection rate, we observed a slight upregulation of synuclein followed by the downregulation of it. Similarly, in longer infection hours, we have observed the downregulation of alpha-synuclein. Apart from synuclein, we observed its associated gene expression profiles also. Here, except L-galactin S1, we observed the downregulations of all alpha-synuclein associated network. Here, for observing the T. gondii infection rates, we analyzed the T. gondii 18S rRNA. Further, we analyzed the expression patterns of similar genes in longer infection hours also. Here, we observed the downregulation of certain genes. They are including STAT3, annexin 2, interleukin 1 beta, along with the monoamine oxidase A. With this observation, it is suggests that T. gondii infection is having important role in dysregulation of alpha-synuclein along with its associate network, leading to neurodegenerative phenotype. Further, for validating our results, we did immunofluorescence based studies. Here we can see the alterations in alpha-synuclein expression levels when compared with the control. Here in control, there is an abundance of alpha-synuclein levels followed by the infection. This is the experimental methodology we followed for our overexpression study. Here we followed the similar methodology earlier as I discussed above, but here we did transfection with 3 different types of alpha-synuclein variants before infection. After transfection, we analyzed again the alpha-synuclein expression levels upon T. gondii infection. Here, when compared with the wild type overexpression neurons, we observed the slight increase in alpha-synuclein expression levels in mutant A53T alpha-synuclein overexpression neurons along with the infection. So further, we analyzed that T. gondii infection is getting susceptibility towards to mutant alpha-synuclein expression neurons when compared with the wild type. With this, I would like to thank NCP.
Our next participant is Dr. Krishnendu Kundu.
Good evening, very good evening to respected judges and ladies and gentlemen. Actually, this is a very small part of the initial work which we have initiated as a part of the project. So, this is regarding Babesia gibsoni, which is a protozoan parasite that infects dogs. So, Babesia, just like malarial parasite, Plasmodium is epicomplex in protozoa. The difference is that it comes altogether into a different group called as Pyroplasmida, because they are transmitted by tick vectors, arachnids, and secondly, they don't form the hemozoin pigment. Now, you can clearly see the importance in India because of the high prevalence. It is prevalent throughout the globe, but challenges are in India are specifically different. Now, what are the challenges? First of all comes the diagnostic challenges. The parasite, actually altogether there are 7 known species of Babesia that parasitize dogs. In India, there are 2 main species. One is gibsoni, another is Vogeli. Now, both these species are transmitted by a common vector, and that's why this particular tick vector is responsible for transmitting. Now, the another problem comes is the difference in the treatment. Now, the drugs to which this Babesia vogeli respond or other forms of Babesia respond, this Babesia gibsoni does not respond to the same treatment protocol. So, one is the diagnostic amoeba. Third most important thing is that if you go for sensitive tools like PCR or qPCR, they are not available for field. So, we need to get a assay which can be tried in the field. Now, so the field mainly the dogs are employed as a part of the security forces, the army or the paramilitary or the police. Now, the increasing pet population. Now, the one of the reason for endemicity is not only this Babesia is transmitted by the tick vectors, but also they are transmitted by during the vertical transmission. So, it is well documented that Babesia gibsoni can be transmitted from the mother to its offspring through the transplacental route. Now, why this particular antigen? Now, a lot of antigens have already been screened. Now, most of them are merozoite surface proteins. So, merozoite surface proteins, we can only use them for detection of antibodies against these proteins. Now, through the literature search, whatever literature is available, 2 proteins have been identified as of date. One is secretory antigen 1 and another is secretory antigen 3. Now, secretory antigen 1, though it is highly conserved, but again the problem is the detection of antigen is less sensitive as compared to the antibody detection assays. So, this secretory antigen 3, number one, it is a multi-copy gene, it is intronless, and it has not been explored for antigen detection assays. So, so what we did, we have screened the samples from the clinical cases from the veterinary clinics in the private or in the government veterinary clinics or college clinics. So, we need to have samples which are clearly positive for Babesia gibsoni or Babesia vogeli or negative. So, thorough screening of the samples was done based on microscopic examination as well as nested PCR. Now, then since this is an intronless gene, it was amplified. Now, different isolates from different parts of the country, they were collected, they were available, and so we sequenced them. And on the basis of the sequences, there is only one sequence available in the database, that is from Japan. So, this is the first sequences we are generating from India. Now, in that we find at the N terminal large number of peptides are present. So, we go for the expression. It is expressed as a protein of 56 kDa just because of the thyroidoxin tag. Now, in the DOT ELISA and the ELISA studies, they show that the sensitivity, specifically this is a antibody detection ELISA. So, we can see a reactivity with the Babesia gibsoni serum and a clear distinction from the negative serum. Now, the next challenge that comes here is that we have to generate the polyclonal antibodies against this particular antigen in heterologous system, rabbit or mice, and then we have to detect whether we are able to detect the antigen in plasma or serum. So, that is how we can detect it. So, there are Along with this, we have done the literature search, and we have found that the peptides that are available, they have some homology with some other hypothetical proteins which are available in the database. So we are going for the amino acid sequencing and then peptide synthesis. Thank you. So I acknowledge DBT, Central Military Veterinary Laboratory, which are our collaborative partners. Thank you very much.
Our last participant is Mr. Karthik Ramalingam.
Good evening, everybody. My title for today's flash talk is Exploration of Therapeutic Potential of Dipeptidyl Carboxypeptidase Inhibitor and Amphotericin B against Experimental Visceral Leishmaniasis. VL is a very severe fatal form of disease with no effective vaccine available and limited drug options to treat. There is always an urgent need for new drugs. Before jumping into the topic, I would like to advocate and defend saying that this neglected tropical disease is not over. This disease always strikes back. Whenever there is a treatment failure, it leads to relapse and in the form of dermal leishmaniasis, challenging the elimination efforts in India. To support this, there has been report of 350 VL cases with 10 deaths and 250 PKDL cases up to 31st August in RMRIMS Patna, which is the WHO Center for Leishmaniasis. There are three challenges towards sustainment of disease control management. They are cyclic outbreak, vicious cycle, and non-endemic new pockets. If you see the pattern of case outbreak in India from 1977 to 2023, every 15 years there has been increase in the number of cases. So if we are happy saying that by 2023 the data shows the cases have gone down, we cannot relax like that. Definitely it is going to bounce back. And it is surprising that recently CL cases have been reported in non-endemic states like Jammu Kashmir, HP, Himachal Pradesh, Rajasthan, Kerala and Punjab. And 5 to 7% of VL patients in India were identified with co-infection of HIV and TB, forming a vicious fatal cycle. And shockingly, asymptomatic filariasis have also shown to progress clinically visceral leishmaniasis in Bihar patients. Now coming to my work, since a combination therapy of liposomal amphotericin B and meltifosine has been successful in Kala-Azar elimination program, in my study I took a CDRI oral compound 98/288, which is an LD-DCP inhibitor reported from my lab with amphotericin B as a new treatment option for VL. And this graphical abstract shows various in vitro and in vivo studies that we planned. Coming to the output or results, the in vitro drug interaction between the combination dose exhibited a synergistic effect in both the stages of the parasite, in both promastigote and as well as intracellular amastigotes. And the same thing was observed in the in vivo drug interaction study in hamster, chronic hamster model, where more than 90% of inhibition was achieved in all the three target organs of the treated hamster groups. Though the percentage of inhibition seems to be moderate, which is around 20% in all the three target organs, they are statistically significant. And in active VL, is generally characterized by loss of body weight, splenomegaly and hepatomegaly. So we tried measuring the clinical alterations and we found there were no significant difference in body weight and liver weight in case of combination therapy compared to their monotherapies on 28-day post-treatment, but there were significant difference in the spleen weight between the treated and untreated groups, which clearly indicates the onset of splenomegaly. We also quantified the Th1, Th2 cytokines and inducible nitric oxide synthase level by RT-PCR analysis, and the results showed that in INOS and Th1 cytokines, there were a twofold increase in the level of expression in the combination treated groups compared to their respective monotherapies, whereas in Th2 cytokines, there were twofold decrease in the combination treated group when in comparison to their respective monotherapies. The other two parameters for assessing the cellular immune response are lymphocyte proliferation assay and nitric oxide generation estimation. And both these experiments we carried out from the lymphocyte cells isolated from the mesenteric lymphodes of the treated hamsters, and saw a result of twofold increase in the combination treated groups. Similarly, we assessed the biochemical parameters like reactive oxygen species and nitrogen species with threefold increase in ROS and fivefold increase in RNS of the combination treated group. And this combination also seemed to enhance the phagocytic activity. And when we studied the survival kinetics, our combination treated group survived more than 300 days, similar to that of a normal healthy control. From this, we conclude that this combination therapy exhibited synergistic effect in the in vitro level, and in the in vivo level it had a potential immunostimulatory effect, and thereby this study establishes that the combination therapy is much more effective than the monotherapy due to its immunotherapeutic activity, and may be more safer than the existing combination. Thank you.
Well, that concludes the first day of 33rd NCP. We sincerely thank all the speakers and participants for their active participation. And I also request all the speakers and participants for tomorrow to be prepared with their PPT or submit it in Hall 2. On that note, dinner is served now, and we look forward to your presence tomorrow for another day of engaging session. Thank you.